Thank you all for coming. Good morning and welcome everyone. We are honored to introduce the Ambassador and Permanent Representative of Finland to the UN to open this symposium today. So please, if we can give a warm welcome to Ambassador Heidi Schroderus-Fox. Thank you again.
Good morning, everyone. A great honor to be here. Really pleased to see you all, ladies and gentlemen. My name is Heidi Schroderus-Fox. I'm the Ambassador and Permanent Representative of Finland here in Geneva. We are taking care of the Finnish relations with all the different international organizations, for example, that are based in Geneva, and there are more than 40 of those. So we are working on security policy, arms control, humanitarian affairs, human rights, trade policy issues, and so on. And so I would like to really thank the MEG for inviting me. It's a great pleasure and really an honor to welcome you all to this MEG in Pharmaco Symposium. Your host today, MEGIN, the global leader in magnetoencephalography technology, originating from Finland. As the Finnish ambassador, it gives me great pleasure to open this symposium and really appreciate the more than 30 years of hard work that has gone into the MEG technology and by all the different scientists and leaders in this field. As a diplomat, one has to learn new things every day, be it from foreign and security policy to international trade, negotiations, and the last, I would say, 4 months, I have also been co-chairing the UN Human Rights Council, which is based here in Geneva. And it is the highest organ looking into the world of human rights. And this is a quite challenging task, I have to say, especially during these very turbulent times. But they say that learning new things keeps your brain healthy. And people in this room could probably put the same in slightly more scientific and precise terms. And again, I've tried to learn a new thing coming here, and that is how to pronounce the word magnetoencephalography. So within a few hours, I hope that I will also hear and understand a little bit better what it all means. So, ladies and gentlemen, MEGIN is a global neuroscience technology company focused on developing innovative solutions for functional brain mapping for pre-surgical evaluation. Originating from Finland over 30 years ago, and now with offices in 4 countries, MEGIN is committed to advancing the standard of patient care worldwide in emerging translational clinical research and advancing clinical applications. And I'm honored to welcome you to this first MEG Center in Switzerland, powered by MEGIN. It is wonderful to bring international translational research and pharmaceutical industry experts together today to share knowledge and further this field with innovative technology. This group of key opinion leaders represents some of the most influential research with MEG in this space. So with these few words, I look forward to seeing the advances that this group can make together in the future. Thank you to MEGIN for inviting me to open this event, and thank for Campus Biotech for hosting the symposium in your beautiful premises and enabling these fruitful discussions that we are and you are going to have in the next few days. I wish you all a very good day, and thank you again.
Thank you very much, Ambassador. That was a great introduction. It was a great honor to have you here to start this meeting off. For those of you who don't know me, my name is Krish Singh. I'm the head of the MEG lab in Cardiff, in Wales. And it's my honor to get this party started. So thanks, first of all. I want to start with some thanks to Ambassador Schroderus-Fox for kicking off our meeting, Megan, obviously, and all the people I've listed for organizing the meeting. It's very generous support for this important topic. Campus Biotech and all the staff who are in this room who've helped put this meeting together. I know how much work it is, so we really appreciate it, and our speakers for today. Now, for it's nobody's fault, but we've started a little bit late, and I've done my usual trick of having far too many slides for the slot that I've been allocated. So please forgive me if I skip over a few slides. I'll cut my talk on the fly, but hopefully you'll still get the gist of what I wanted to talk about. I also wanted to thank our speakers for today. We have an amazing program. I'll skip over this because it will become obvious through the day who's speaking, but we've got leading speakers from both industry, clinicians, and MEG experts talking about a variety of topics that are all within the translational and pharmacological sphere. So we're going to learn a lot, and I'm really excited about the program. So thanks to all the speakers for their efforts to get here. So what I'm going to do is I'm not going to talk too much about the pharma space and target engagement, because people better qualified to talk about that will be coming after me. I thought I'd do a brief introduction to MEG and how I see what its strengths and weaknesses are, and then describe some of the work that we do at Cardiff in pharmacological MEG. So I'll just skip over this for the sake of time. Okay, so very complicated slide. I think it's always sobering and important to talk a little bit about the complexity that we're up against. So this diagram here, which I always tell my undergraduate students they don't have to memorize for the exam, is from a famous paper by David Van Essen from 1991. And it was a deep reconstruction of everything we knew about the macaque visual system at that time. So every box is a known functional brain area, starting at the retina and going all the way up to the top to the hippocampus. And every line is a white matter pathway. So this is what we're up against. We want to understand how the brain works. And remember, this is just the visual system. We need to understand everything about this. And we want to probe it non-invasively with high repeatability and robustness, non-invasively in human beings. And obviously this would be relevant to clinical and pharmacological studies. So if we wanted to design a perfect neuroimaging tool to do that, we need to be able to measure all of these parameters. We need excellent spatial localization, amplitude, temporal sequencing, frequency, connectivity, transient connectivity, and how these things are modified by disease and, for example, pharmacological action. And of course, no single methodology can provide an ambiguous measurement of all of these parameters. Now, the thing that I want to concentrate today is on oscillations and connectivity, because it's become clear over the last few years that the way the brain works in terms of routing information through different brain areas to support cognitive processes is through the integration of oscillatory rhythms in the different frequency bands. And it does this, the brain uses this and creates these oscillatory networks in a transient basis, millisecond by millisecond. So we need an imaging technique that can measure those oscillations and characterize them and look at how these networks evolve on the scale of thousandths of a second. And those of you that work in the field will be familiar with some of the key physiologists and theoreticians working in this area. Pascal Fries with his communication by coherence hypotheses around the visual gamma oscillation, around the gamma oscillation, which says that the synchronization of gamma is the way that areas transiently become connected to each other to pass information and then disconnected when they're no longer needed to communicate. So you can see here spiking rates are maximized when the oscillations are in phase in two brain areas. And similar and complementary to that are theories around alpha, the 10 hertz rhythm as an inhibitory gating rhythm. This is a paper by Ollie Jensen and Ali Mazahira, showing that you can suppress and activate brain regions on the scale of milliseconds by modulating the size of this 10 hertz alpha rhythm. So this just shows that, these electrical rhythms in the brain are critically important, and we need to be able to measure and characterize them. So my take-home message one is that electrophysiological signals, whether you're about understanding normal brain function or brain function in disease, or using some kind of target engagement measure in a pharma study, you want to be measuring electrophysiological signals, non-invasively and in humans, and not just indirect hemodynamic markers such as fMRI. And we have two main techniques for non-invasively measuring human electrophysiology. We have the classic EEG that I know many of you work with, which is cheap, millisecond time resolution. You can measure and characterize oscillations, but its localization, it can be rather poor, and it's particularly sensitive to muscle artifacts. Then we have MEG. Most of us are currently using SQUID MEG. Has all of the advantages of EEG, but has better spatial resolution, high throughput because there's no electrode prep time. You can just put somebody in the helmet and start recording. Less sensitive to muscle artifacts. But it's expensive to run. It's expensive to buy, and it's expensive to run. And because we have a fixed helmet size, which is usually sized for adults, it's not suitable for young children and babies. But the new kid on the block, and there are many of you who are working in this area, some of you will sell systems based around this new technology of optically pumped magnetometry, is a wearable room temperature system that has all of the advantages of MEG, much cheaper to buy and run, and is an up-and-coming future technology that we are getting to grips with a little bit in Cardiff. And here you can see one of our participants wearing the OPM MEG helmet and having a fine time doing an experiment for us. For the purpose of this talk, I'm going to completely ignore the distinction between SQUIDs and OPMs, and I'm just talking about MEG as a technique. But you could do everything with either of these techniques. So MEG measures the magnetic field that comes from neuronal populations. Where does that come from? Well, it comes from the synchronous firing of the excitatory pyramidal cells. And for various technical reasons, we know that the signal principally comes from the lower frequency, sort of less than around 150 hertz, local field potential. So I think that the way to think about once you've source localized MEG and EEG signals, you see it as a macroscopic version of the local field potential that you could measure if you stuck an electrode into that brain region. And that's one of the powers of these tools. They're completely translatable from the animal recordings with invasive electrode recordings. One interesting thing that's not been particularly well studied yet is in the higher gamma band, where you have power that's sort of broadband and then increasing with task above around 80 hertz. There's some evidence that that might be a surrogate of the spiking rate and firing rates of neurons. So we don't normally think that we can detect, well, we know we can't directly detect individual firing rates of neurons or even bulk spiking rates, but this higher gamma band activity may be a proxy for that. In the interest of time, I'll skip over this a bit, but basically just to say that MEG is mostly sensitive to the external neocortex, and it's harder to measure from deeper areas. But there's a paper down on the right there by Brian Cornwell back in 2008, I think, showing that you can measure and characterize hippocampal theta oscillations in a navigation task. So MEG, with appropriate design and source localization, can measure from deep activity, and there's technical reasons to think that OPMs may give you even better sensitive to this deeper activity. Okay. So why use MEG and EEG as a functional imaging tool and not fMRI? And the reason I wanted to address this is that many of you will know that fMRI is a really common tool that pharma companies tend to like in early phase trials for looking at target engagement. And one of the points of this meeting that I hope will come across is that MEG has a lot of unique information to offer here that has significant advantages over fMRI in many applications areas, but particularly in this one. I don't see them as exclusive competitors. In our research, we use both MEG and fMRI. So I published a paper in Neuroimaging back in 2012, which was just a short review article about some of the issues around fMRI, and I published this very complicated figure in the paper, where I tried to summarize the whole issue around the kinds of things that we want to understand as cognitive and clinical neuroscientists, which is the blue boxes on the left, and how the brain creates these percepts and cognitive processes. And then in pink are the electrical signals that the brain uses and the things that we can measure that tell us about these processes in blue. And then on the right-hand side, the boxes in orange are all the things that control the signal that we measure with fMRI, this hemodynamic blood oxygen signal. It's a very complicated neurovascular coupling system that produces these beautiful spatial localizations. But getting from the blue to the orange is quite difficult, and in some ways is a non-unique inverse problem in terms of the information that we need to be able to solve it and to properly understand what the fMRI results are telling us about brain function. At the top in green are modulatory effects, things like aging, disease, relationship to structure and variations in neurotransmitter concentrations like GABA, and obviously pharmacological manipulation would be one of the green boxes at the top. So take-home message number 2 is that the standard bold signal has this amazing spatial resolution, but its neurovascular coupling function is only partially understood. And that itself can change with age and with drugs. So if you give somebody a novel drug compound, do you know that it's the brain activity that's really changed? Or could you be directly systemically affecting the neurovascular coupling by that drug? And there's some really nice papers coming out just recently showing that the hemodynamic bold response that we measure with fMRI in the default mode network and the hippocampus is really weird. It's not the standard HRF that we know about. And in fact, in many cases in the default mode network and the hippocampus, there's some emerging evidence that the hemodynamic response function might be inverted. So actually you see a negative signal when the neural activity goes up. And obviously I'm not going to go into this here, but there are some papers here that I've enjoyed reading recently about the weirdness of the fMRI signal in the default mode network and the hippocampus. So if you're interested in that, I'd suggest you having a look at those. So all of these signals are potential measures of target engagement. If you're interested in pharmacological manipulation, these various different electrical signals and phenomenology, they're all potentially accessible with invasive recordings in animal models and with EEG and MEG with appropriate source localization. So one sort of nice review paper about which provides a framework around the relevance of oscillations in the human brain is this paper by Donner and Siegel that came out a few years ago, where they talk about the distinction between local processing and connectivity, which is mostly relevant in the kind of high frequency gamma band, and long range networks across the brain, which mostly seem to communicate in the lower frequencies in delta, theta, alpha, beta. So in our lab, we probe the high frequency gamma variability and task modulation using simple passive grating, viewing of high contrast grating stimuli to generate a visual gamma oscillation. And like most people, we use resting state MEG connectivity to look at lower frequency band activity and connectivity. So if we just talk about the resting state for a moment, the key thing I want you to take away from here, if you're not familiar with the field, is that by doing a simple 5 or 10 minute resting state recording with MEG, then doing appropriate source localization in different frequency bands, you can get patterns of both activity and connectivity from the same data using a variety of common analysis approaches. And you can do that with either a static connectivity, where you just look at connectivity over the course of the whole run, or you can do it dynamically, where you look at how the connectivity updates every few 100 milliseconds, for example. And one really important thing to say is that it's recently become clear that these patterns of oscillatory connectivity are really individual. They're robust, repeatable, and they're like a fingerprint in a given individual. So this is a really nice paper that came out a couple of years ago from the Montreal lab. They showed that if you did a resting state MEG recording and broke it down into different frequency bands, you could train a classifier, a system to then re-identify people in a second run with over 90% accuracy. So we know that these resting state patterns are really robust, repeatable, and are specific to you as an individual. And that makes them really good candidates for pharmacological studies and target engagement studies. Now that happened to be just in terms of patterns of static connectivity, but we can look at how these networks evolve over hundreds of milliseconds. You can use various different algorithms for extracting these time varying networks. And obviously the ability of drugs to modulate how fast networks switch in and switch out is another area of opportunity for pharmacological studies. And when you do these sort of network decomposition methods to find out what the relevant stable networks are that we keep repeating in the time series, in the EEG, you get these really robust, what are called microstates, which tend to have very simple topologies. And usually you would break it down into only sort of 4 or 7 microstates. And these are really ubiquitous and repeatable across many different labs and studies. My colleague, Dr. Luke Tate from Cardiff, he published a paper a couple of years ago showing that you could do this similar kind of analysis with MEG. And with source localization, you can get much more of a neurophysiologically informed picture of where these microstates are represented. You also tend to get more microstates from the MEG data than you do from the EEG data. So in our lab, we use resting state MEG and principally static connectivity to look at network hyperconnectivity and hypoconnectivity in diseases such as schizophrenia and risk of schizophrenia. So here you can see this is a very simple static connectivity analysis, really easy to do from resting state MEG data, showing alpha and beta posterior parietal and default mode network disconnectivity, which is these blue lines here, and gamma hyperconnectivity in the frontal lobes down at the bottom here, adults with chronic schizophrenia. At the top here, adults carrying particular genetic deletions, which give rise to added risk of later development of schizophrenia. And here in children with a rare genetic deletion called 22q11 deletion syndrome, where these children also have much higher risk of later developing diseases such as schizophrenia. And you can see there's kind of a convergent pattern of hyperconnectivity and disconnectivity. So switching away from resting state and talking about the visual gamma oscillation, which is a simple task-based experiment where you just show people these black-white bars, and it elicits this high narrowband gamma response in the visual cortex, which you can see there. This kind of horizontal line is the sustained gamma oscillation. You can see there's an initial gamma spike that's also induced. And these have been seen in macaque monkey. So this is an LFP recording from a macaque monkey. This is a source localized response from our MEG lab from a human participant. So although it's not a very interesting cognitive neuroscience experiment, it's about the simplest experiment that you could do. What I like about this and why we publish so many papers using this type of paradigm is it's directly translatable from the animal literature. And different people have different gamma oscillations. These are different individuals. So if you look on the right, you can see the frequency changing. And what's really nice about these oscillations, we know exactly where they come from. They come from the superficial layers of the cortex, superficial pyramidal cells and inhibitory interneurons. And this recent paper studying macaque monkey from the Bastos lab has shown that this kind of superficial gamma oscillations, deeper layers, alpha and beta oscillations is ubiquitous across the cortex. So there's a real difference in the different layers of the gray matter, which layers are generating these oscillatory dynamics. So take home message 3, by source localizing frequency-specific oscillations, either in resting state or task, you're actually getting insight into layer-specific cortical activity within specific types of neuronal populations. And that's really important and useful. And my colleague Suresh Muthukumaraswamy showed that these oscillations are really repeatable across individuals if you measure them every week, they're identical in a given individual. This paper here again from Pascal Fries's group showed that identical twins have identical visual gamma oscillations, so it's highly heritable. And we know what causes these visual gamma oscillations. It's an interaction between inhibitory interneurons and superficial pyramidal cells. And if you tweak the balance between excitation and inhibition in this network, you change the frequency. So when different people have different frequencies, we think that's because people have different balances between excitation and inhibition within these superficial layer 2-3 networks. And I think in the interest of time, I'm going to just skip ahead a little bit, but we've done quite a lot of work using a method called DCM. That's my colleague Alex Shaw. And we showed that you could basically build a computational model, which you can see on the right there, which is of the primary visual cortical column. And you can solve for all of these parameters and connection strengths in the model. And as I said earlier, individual variability in this gamma oscillation is predicted by the strength of the drive from the inhibitory interneurons in the superficial pyramidal cells and the intrinsic synaptic properties. So the key thing that you have to know, and there are many people in this room who work with this kind of technology, is MEG can source localize neurophysiologically relevant oscillatory and activity patterns from task or rest-based data. But you can get really useful added insight into receptor-specific neurophysiology from modeling this data using techniques such as DCM or HNN. So HNN is another toolbox that come from Stephanie Jones's lab over in the US that's definitely worth a look as well. And all of these approaches could be deployed in pharmacological studies. And I think the effect that drugs have on the parameters of these fitted models, and I would predict that James and or Laura are probably going to talk about that this afternoon in more detail, is really giving added insight into this approach, into the pharmacological action of drugs using pharmacomeg. So I'm going to finish in the last 5 minutes or so talking about what we've been doing in Cardiff over the years. We've been doing pharmacological MEG for maybe 10 to 15 years in our lab. In CUBRIC, in Cardiff, is a university in Wales. And we have a really nice facility there. We have a clinical research facility, which is basically capable of running a clinical trial. It's proper ward level control. We have the MEG lab right next door. We have a 3T Prisma next door. We have wet labs, and we also have EEG. So we can run a whole clinical trial just in this part of our building. And we've done work on the left. You can see lots of different drugs that we've studied across a variety of receptors, GABA, NMDA, AMPA. We've also done work on psychedelics like psilocybin and LSD. And then we have a group led by Neeraj Saxena and Shamila Ko, who are looking at the action of anesthetic agents like propofol, dexmedetomidine, remifentanil, and midazolam. So I said we've been doing MEG at Cardiff for sort of 10 to 15 years. I wanted to name check my former colleague, Suresh Muthukumaraswamy, who started the pharmacological effort in our lab and was the kind of lead on most of the studies that I mentioned on the previous slide. And Suresh published this nice review article about pharmacomeg in 2014. Suresh is now working he has his own EEG lab in Auckland in New Zealand. So it's a really nice review article, but I was slightly dismayed to see that it's 10 years old. And I think it's fair to say there are not many labs that have taken up pharmacomeg since then. And I think this is a really untapped need and potential that as a community we should really be evangelical about. MEG has so much to offer in this space that there really needs to be more work in this area. And this meeting is partly about trying to be evangelical and drive that far forward. Let's skip over the slide in the interest of time. I just wanted to say one thing about the philosophy of our experimental design. In both our pharmaco, MEG, and clinical studies at Cardiff, we tend to use very simple experimental protocols. So we don't tend to use complex cognitive designs. We use simple tasks like the visual gamma, 40 hertz auditory stimulation, mismatch negativity, and resting state, mainly because we can guarantee that all participants can do them. Some of these signals are actually effectively automatic. So engagement is not strictly necessary in some of these tasks. And in challenging populations, particularly kids with maybe some kind of diseases or genetic risk factors, that's important. So because we see oscillations being linked directly to underlying synaptic physiology and pathophysiology, we want tasks that tap into that. So the tasks that we do are not necessarily linked to the cognitive impairments in the target disease. And I think that's important. So here is some of the work that Suresh led on ketamine. So unsurprisingly, when you give people ketamine, you get really profound oscillatory changes within the brain. So you can see over here, ketamine versus placebo in sensor space, you see profound suppression of the delta and alpha and beta rhythms across the whole brain. Surprisingly, rather limited effect in theta, and you get enhancement of gamma. And when you source localize this, which is really straightforward to do, you see you get really added insight into which brain regions are creating these sensor space topologies. And just quickly, we also did a study I should say that ketamine is of interest because it's an experimental model in animals for schizophrenia. So it's quite often animals, rodents will be given ketamine to try and mimic some of the symptoms of schizophrenia. So that's why it's of interest in the clinical domain. We also studied perampanel. So ketamine is an NMDA antagonist, and perampanel is an AMPA antagonist. It's an anti-epileptic drug. And you can see that the center topologies are really fundamentally different to ketamine. So this is showing us the benefit of looking in these different frequency bands. You can see each drug has its kind of own unique fingerprint, even in a simple resting state experiment. So even though these two receptors are a part of the kind of glutamate pathway, AMPA and NMDA, the signals and responses that you see are completely different using the two drugs. We've also done quite a lot of work with an anti-epileptic drug called tiagabine. So tiagabine is a GABA reuptake inhibitor. So if you give people tiagabine, the amount of GABA within the synapse will rise. So it's boosting GABA inhibitory availability. And in our lab, we looked at both resting state data using tiagabine and this visual gamma oscillation. And what happens with the visual gamma oscillation is that the frequency drops when you give people tiagabine. Actually, both the alpha, beta, and gamma oscillation drops in frequency. And when you model that, we found that the biggest model parameter using DCM that changed during the administration of tiagabine was a lengthening of the inhibitory time constant on the iPSCs. And actually that's been shown in vitro in this paper that I've shown here. So the MEG source localization DCM modeling shows the same behavior within this parameter that's been seen in direct recordings from these types of neurons when you put tiagabine on them. In the resting state, when we gave people tiagabine, we saw patterns of changes again along delta, theta, alpha, and beta. Frontal increases in the power of these rhythms, and posterior parietal and posterior default mode suppression of these rhythms when we gave people tiagabine at different time points. And the reason I wanted to show this is working with Alexander Hammers, who's in London. Alexander Hammers gave us some flumazenil binding PET data, which says where are the highest regions of GABA A receptors on average in a human brain. And I don't really have time, sadly, to go into this in detail, but what we found in this paper is that those regions of the brain that had the highest GABA-A receptor binding were the ones that showed the biggest modulation of the oscillatory responses. So you can see, for example, at the top there in the delta band, on the x-axis of these graphs is the amount of GABA-A binding in a particular brain region, and each dot is a brain region, is a brain voxel. And on the y-axis is how much the oscillatory power was changed in that voxel. And you can see that the highest changes were seen in the regions of the brain that had the highest GABA-A receptor binding. And there was a really nice paper looking at this type of effect of mapping neurotransmitter densities to oscillatory and functional and structural brain parameters across individuals. Again, came out of the Montreal group. This paper by Justine Hansen and colleagues a few years ago, where they took available maps of many different receptors and looked at that. Okay, I don't really have time to do this story justice, but we've also got a group, as I mentioned earlier, who've been studying the effects of anesthetics, sedatory drugs on brain function. And this is about trying to understand how anesthetics work. Anesthetics, we've all had them. Well, we've all had sedation, but mostly we don't know how these work. We know something about their mechanism of action. So remifentanil targets the mu opioid receptors in the brain. Midazolam is a GABA modulating, a GABA-A modulating sedative. And using resting state MEG and source localization, this team have been looking at the patterns of modulation that you see with these different drugs. So at the top row here, this is midazolam. And down here is remifentanil. And you can see there's this nice pattern of increases and decreases in resting state oscillatory power under these two sedative drugs. Remifentanil didn't induce as many changes as midazolam. One kind of funny thing is you can see there's a really strong response in the thalamus. So the alpha and beta rhythms, when we source localization, we saw really big. Now, we don't really believe this because MEG isn't supposed to be able to localize effects in the thalamus, but it just turns out that the part of the brain that has the highest mu opioid receptor density in the brain is that area of the thalamus. So we've got a result which is perfectly expected, but not expected, if you see what I mean, because we shouldn't be able to measure signals from that region. What we are more excited about is we saw theta reductions in the hippocampus with midazolam, and that's exciting for two reasons. One is midazolam is known to have an amnesiac effect, so patients often can't remember things after administration of midazolam, and that theta suppression has been seen in mice with invasive recordings and when people have been given midazolam. So finally, the big study that we're currently involved in with Pharmaco MEG and Pharmaco fMRI with my colleague Neil Harrison, who's over here, is a phase 1 clinical trial. So in Cardiff, we're really lucky that we have an institute that develops new drugs called the Medicines Discovery Institute. And they've developed, this is run by my colleagues, Simon Ward and John Atack. And they've developed a new drug for potentially treating the cognitive impairments in schizophrenia. And we have got ourselves linked into this study. This is the Medicines Discovery Institute, led by Simon and John. And the drug is an AMPA-PAM. It's a positive allosteric modulator of the AMPA receptors. And the hope is that by giving people with schizophrenia this drug, you'll improve cognitive performance through this upregulation of glutamate release. And this drug has been in development since 2013, originally in GSK. Wellcome have then funded the MDI to take this forward, including recently a £6.7 million grant to do the clinical trial. It's through using a clinical trials company called Symbeka Ryan, based in Wales, near us. And they gave the drug to the first human participant in March 2023, which was successful. And that meant that we could then join the study And participants are brought by Simbec Orion to our clinical trials unit. They have fMRI, which is led by Neil, and they have MEG, which is led by me, and Rasha Haider, Natalie Jones, and Abby Rees. Natalie and Abby are in the audience. They're the people actually doing the work. So this is an amazing study because it's very rare for a university neuroimaging lab to get involved at this early level in a first-in-human trial. There's lots of governance and organizational issues to sort out. But the guys are doing an amazing job, and we're running people through this trial now. So they come and we're doing, we'd scan participants as they're part of the trial at Simbec, and then we're also going to be doing a placebo crossover design of the drug later on. And I should say that clinical EEG is also being done by our colleagues at a company called The Science Behind, based in Cardiff, and Tonya and Sophia are also in the audience from The Science Behind. And I'm going to have to skip over this, but basically the design of the clinical trial was first in human safety testing, a single ascending dose study, a multiple ascending dose study where people are being looked after by Simbec for quite some time. And at the start and the end of that process, they come to CUBRIC and we give them MEG and fMRI. And then we will have our own crossover design is due to start in about a month, where people will have the drug and have it within CUBRIC. And then will be immediately scanned with MEG and fMRI. Okay, so I'll wrap up now. I've covered these take-home messages through my talk, but the thing that I like about MEG and why it's useful in this context is understanding individual variability, fundamental neurophysiology of disease, translatable from animal models, sensitive early markers, target engagement of receptor-specific profiles, and you can use MEG in challenging populations. And then I just want to finally thank the funders that over the years have funded all of the work that I presented today. And that's me done. You'll be pleased to know. Thank you very much for listening. Neil is an academic psychiatrist. And as I said before, he's mostly working with fMRI, and he leads the fMRI pharma efforts in CUBRIC. Thanks, Neil.
Thank you, Krish. Yeah, thank you very much. Thank you, Krish, and thank you, Nicole, for that invitation to speak. Now, I feel a little bit of an interloper here because I'm really not a MEG researcher, or at least not yet. But I've been working with Krish, as you've just heard, on one project. We're beginning to work. Well, we have started last week on a second project. I'm going to talk a little bit about that. And I suppose I want to give a little sort of story into why I think MEG may be particularly useful in psychiatry, and trying to address this difficulty of, if you like, this crisis in drug discovery within psychiatry. So why do I think there's why is psychiatry in crisis? So these are a few quotes. This is some quotes from Tom Insel, who ran NIMH, the big US government facility for psychiatry for 13 years. But essentially what he's argued is that we've poured enormous amounts of money into psychiatry research, particularly within imaging, within genetics, but what have we got back? How has this transformed the patient journey? How has this translated into new drugs and a new understanding that actually benefits patients at the bedside? And I think we probably all appreciate, and especially when we get a little bit grey-haired and we're a bit further through our clinical careers, we begin thinking, what has our generation accomplished for patients? And I think within psychiatry, sadly, I don't think we've really accomplished a great deal, at least within the development of novel therapeutics, pharmacotherapeutics. And indeed, as you can see here, there really haven't been any mechanistically novel new therapies in psychiatry for more than 50 years. So we used to tease our colleagues in neurology for not having new treatments. Sadly, they've overtaken us, and we're really still using these old drugs that were developed 50 odd years ago. And of course, medics like to reflect on other specialties, and just, for example, looking at oncology, for example. I mean, how has oncology fared? Well, again, over the last 20 years or so, oncology has really been transformed. We've moved away from treatments that were targeted on the tissue of interest, say breast or prostate, and really now entered this world of precision oncology, where patients get their individual tumors phenotyped, and then we can begin to use specific treatments that target that patient's individual tumor. And this is really not where we're at in psychiatry at the moment, and this is somewhere that we would like to try and get to. So why are we stuck here then? I mean, essentially really is still stuck at the starting line. What is the diagnosis and how can we begin to move forward from here? And probably the coalescing sort of opinion at the moment is really a critical part of the problem is we're still making all of our diagnoses based on symptoms. So this is unfortunately innovation within psychiatry. I mean, we moved from 2013 to 2022. What are the big innovations in diagnosis? Well, they're a modification of the DSM. So a modification of the Bible used for making clinical diagnoses within psychiatry. So we're still lacking biomarkers and we're still very much reliant on patient symptomatology for making diagnoses. Now, why is that problematic? Well, one area of problem here is that these diagnostic syndromes are incredibly heterogeneous. So, for example, just taking, for example, depression as one example, if you look at the ICD-10 and you look at all of the different ways that a patient can be diagnosed with depression according to ICD-10, there are literally hundreds of different ways that different patients with completely different profiles of symptoms can end up with the exact same diagnosis. So just as an example here, too much sleep, too little sleep, eating too much, eating too little, reduction in energy, etc., etc. So we're really in a difficult space, and if you like, analogous to where oncology was 20 years ago. So we're coalescing all of these different symptom profiles into the same sort of diagnostic label, and then we're treating them all exactly the same. So we're not treating symptoms, we're treating these diagnoses which are highly heterogeneous. The other difficulty, and this is something that was raised a decade ago by, Sir Michael Owen, he's another psychiatrist based at Cardiff, but was really this difficulty that psychiatric genetics now have clarified that the diagnostic categories we're using don't really map on terribly well to the underlying biology. So if you look at the genetics of psychosis, for example, there's a broad overlap between genes involved in schizophrenia and those involved in bipolar, and in fact even in those in things like psychotic depression. So they don't breed true. The diagnoses we use within psychiatry don't breed true, and the diagnostic criteria and the classifications we got don't map terribly well to the underlying biology. The other challenge within psychiatry is we don't really have any biomarkers, and I suppose if I was being strictly honest with you, we do actually have one, and that's within anorexia nervosa. We use BMI as a diagnostic biomarker, but beyond that, we really do not have any biomarkers within psychiatry. And again, this is a paper written by John Crystal and various other sort of researchers from the States, really just demonstrating that we're really hampered by this lack of robust, reliable biomarkers, and which can aid objectively measured diagnosis, stratify patients, or even have prognostic biomarkers. And, as Tom Insall noted, we have done an awful lot of work identifying potential biomarkers. We've identified numerous different potential biomarkers, but none of them have really proven sufficiently reliable to be taken into the clinic. And there is now really a plea to try and invest in really trying to solidify the evidence for specific biomarkers. And the reason I wanted to emphasize this is that increasingly the biomarkers that appear to be emerging are electrophysiological biomarkers within psychiatry. So the N170 here, there are also others like mismatch negativity and other electrophysiological biomarkers, which seem to be popping up as perhaps some of the more reliable markers within psychiatry, not just within psychosis, but across other psychiatric disorders as well. And also this raises the tricky difficulty of perhaps needing to identify novel classification methods. So do we need to completely revamp the whole classification structure within psychiatry? You might think that one way to solving this problem is just to look at the drugs that we currently have. And so fine, we know that we have a whole range of different drugs within psychiatry. Why do we just not look at the mechanisms that they're targeting and therefore get a better understanding of the mechanisms that are the biological mechanisms underlying those specific disorders? But the problem is, again, many of the drugs we use were developed serendipitously. So essentially, a lot of the drugs we've got, this is just an example for the monoamine oxidase inhibitors, but you can see the same for each of the other classes in psychiatry, is that each of these drugs was developed by chance, if you like. So here, the MAOIs actually span out of some of the very early efforts to develop anti-TB medications. So this is potentially problematic. They weren't developed from a mechanistic bottom-up approach. They were discovered by chance. And the problem there is that even our best drugs, so for example, in schizophrenia, the antipsychotic drugs which block D2 receptors, if you dig down and look at the genetic evidence underpinning these, there really is very little evidence to support there being a deficit in dopaminergic neurons as being the root-seat cause of schizophrenia, for example. So even the best drugs we've got, okay, they certainly improve patient symptoms and have transformed to some degree patients' lives. But when we dig down and try and understand mechanistically, does this inform us about mechanism? The answer is no. It seems to be that we're improving symptoms, but not getting at the root cause of some of these, some of the most serious psychiatric disorders. And even when we think we do know the underlying mechanism, we probably got it wrong. And this is, I think, some really fascinating work looking at how many of the antidepressant medications work, the SSRIs. And we've always assumed that this is really based on serotonin, for example, and these drugs have been developed to target serotonin. But it would appear that, in fact, probably many of these drugs are actually working on BDNF. So this may actually be the mechanism through which these drugs are working. So again, we're really in a bit of a state within psychiatry, I'd suggest, at the moment. And if you like, psychiatry is failing to carve nature at its joints. We're not really understanding what the root mechanisms are underlying each of our most severe sort of disorders within psychiatry. And that's making it very difficult to advance new therapies, because if you like, we're stuck in this circular system where we're testing drugs for disorders which we know don't reflect the underlying neurobiology. So we're really getting very stuck. So how do we get out of this this impasse and try and move things forward? And I don't know how many people in the audience are familiar with RDoC, but this was again an initiative that came out of the National Institutes of Health more than a decade ago now, 2010, Tom Insel and Bruce Cuthbert. But they recognized this difficulty, as did a number of other people in the field, and suggested we move away, at least for research purposes, from these diagnostic manuals, instead move more to a symptomatic or dimensional type approach. So begin looking at units of analysis and, for example, looking, at electrophysiological measures, neuroimaging measures, cognitive domains, and taking this much more dimensional approach to psychiatry, and then develop our therapies and our mechanistic understanding from this kind of approach. And what has been the sort of the upshot of this? I mean, where have we got to after 10, 14 years of RDoC? Well, I mean, again, the consensus seems to be there's been much criticism about RDoC. It was never designed to be a diagnostic tool, but instead as a way of trying to get into this impasse of understanding mechanism. And I think the broad conclusion is that the RDoC has been an impetus for driving this precision psychiatry approach. So recognizing there's massive heterogeneity within psychiatry, and beginning to address that heterogeneity by collecting data across multiple different domains. So that would include electrophysiological data in this particular context, but also genetics, and using that to try and stratify patients into biotypes that may be more reflective of the underlying biophysiology than we are currently doing. And has that been successful? Well, there have been a number of approaches to try and achieve this, and probably the most famous of them is this initiative called the BSNP initiative, which is the bipolar and schizophrenia network for intermediate phenotypes. But essentially what this group of investigators in North America has done is they've collected about 800 patients with a range of different psychotic disorders. So this is schizophrenia, bipolar, schizoaffective disorder. So rather than try and classify people by these DSM measures, they've just taken patients who have broad different presentations with psychosis, and then collected a range of different measures on them, ranging from physiological measures, electrophysiological measures. So in this case, this is EEG, not MEG. There's also MRI and fMRI in here, and cognition, and then use machine learning approaches to try and identify what they call biotypes. Can we classify patients, forgetting their actual DSM diagnosis, can we classify them based on these different domains of data? And they claim to have been fairly successful in doing this, largely based, again, on electrophysiological and cognitive measures. So they've described these 3 different biotypes that have different electrophysiological and cognitive signatures. And now they're beginning the first trial where they're using a antipsychotic drug, so clozapine, and targeting that to one of these specific biotypes. So it doesn't matter whether you've got schizophrenia or bipolar. In this trial, they're going to be targeting individual biotypes that they predict will show a better response to this particular drug than some of the other biotypes. So this is the first indication of perhaps this new approach coming through into a drug trials. So moving forward then, so that was BSNP. So moving forward, what data should we be collecting in these data-driven approaches? What is the ideal data that needs to come in here? And I would argue that it needs to be informed by a number of different sort of bits of knowledge. So firstly, we need to be informed by neuropathology. So what do we know about schizophrenia? We know that there are changes at the level of the synapse. So there's a reduction in synaptic boutons. We know that pyramidal cells are smaller. We know that interneuron, there are deficits in interneurons, particularly parvalbumin-associated interneurons. So we need to be informed by the underlying neuropathology. So the data that we would certainly need to collect, at least from the imaging side of things, would be MR measures that would attempt to capture these changes in perhaps cortical thickness that we know exist, and diffusion measures that may capture something of these neurite density and possibly even these sort of synaptic densities. What about electrophysiologically? I mean, Krish has already given us a very nice overview of this already, but essentially we know that within schizophrenia, there are changes both within local oscillatory activity, but also connectivity. So again, if we're going to adopt these data-driven approaches, then we need to be collecting this rich electrophysiological data in these patient groups. Now, to date, with BSNP, this has just been EEG data, and you'll see in a moment that what we're beginning to do is to go and do a much deeper dive, this time using MEG data, to really capture information like gamma frequency, beta frequency, and some of these connectivity approaches that Krish described so nicely in the previous talk. The other thing that I would argue that we need to be informed by is by genetics. And this is very, very small. I don't know how well you can see this up on the top right, but essentially, this is chromosome 6. This is the MHC region, a rich immune region within the chromosome. And we now know that schizophrenia particularly is associated with a whole range of hits within this MHC or this immune region. Why is that relevant and why is that interesting? Well, we also now know that immune cells, microglial cells, are involved in synaptic pruning, and schizophrenia increasingly appears to be a disorder of the synapse. So again, it seems plausible and reasonable to assume that capturing immunological information would also be critical to try and understand what are the real biotypes, what is the physiological process that's going on underlying these disorders. So what are we doing in Cardiff? So what we started this year is we're developing something called the South Wales and South West Mental Health Hub, and we're going to be doing something similar to the BSNP initiative that I just told you about. But this time we're really going to go and do a much deeper dive in trying to understand and try and get information in 600 patients with psychosis on various electrophysiological and neuroimaging measures. So we're going to be collecting rich neuroimaging data on MRI in this patient group, and we're also going to be capturing and using the battery of MEG tasks that Krish has already gone through in the previous talk, so doing induced and evoked oscillatory activity and also some of the network analyses, as well as collecting rich immunophenotyping in these patient groups, with the aim of really trying to understand what are the underlying mechanistic processes within these patients' groups, and can we classify them and separate them based on this electrophysiological and brain microstructural activity. So this is one approach to sort of trying to break this impasse within psychiatry, collecting data and trying to re-understand and reclassify patients, re-stratify patients according to underlying electrophysiology, neurobiology, etc. But one other approach that we're adopting, and this is one that Krish has already commented on, is this challenge of drug trials. So within CNS drug development and drug discovery, pharma has largely withdrawn or had largely withdrawn from this space because of the enormous costs and risks associated with developing drugs for CNS indications. And what is one of the key reasons for that? Well, one of the key reasons is we lack target engagement measures, in vivo target engagement measures. So a drug will be designed for, say, for example, for use in depression. We won't have any good measures of target engagement in early phase trials. So we don't even know necessarily that the drug gets into the brain. Okay, if we do CSF, we determine it does, but does it actually do anything of relevance within the brain? Often we don't know that in phase 1 studies or even phase 2 studies. So these drugs will be advanced into very expensive phase 3 studies and then fail with clinical endpoints through being underpowered or having small effect sizes, and then that whole drug program is closed down. So what the NIH has proposed is this thing, the Fast Fail initiative, and what they've argued is that what we need in psychiatry is to be able to fail our studies quicker, and the way they argue that we do that is we need to identify biomarkers that can demonstrate target engagement or not target engagement of these individual drugs at a very early phase study, so for example, a phase 1 study. And there have been a number of studies. Andrew Christos, for example, has written this very nice review here just describing this process, and it's been undertaken in a number of different trials within the US. And this is also now what we're beginning to do in the trial that Krish was just talking about with the MDI. So to my knowledge, this would be the first time that MEG, for example, is being used in a phase 1 study in humans. So this is an AMPA-PAM drug that Krish has already described being developed. And what we're wanting to do is to use MEG and fMRI in this very early phase 1 study to try and identify, is there a signature of target engagement? Does this drug get into the brain? We can determine that, yes, using CSF measures, but does it do something at this stage? Does it change electrophysiological activity in a way that we'd predict from the biology of this drug it may do? And then can we use that information to target this drug in future studies? So, for example, patients that show a particular electrophysiological phenotype. And the way Krish again has already gone through this, so I'll be fairly quick, but the way that phase 1 studies normally run is they consist of a single ascending dose and then a multiple ascending dose studies, and that is it. And really the key aim in these phase 1 studies is to determine whether this drug is safe and to look at the kinetics of this drug. However, what we're doing in this study is beginning to use this opportunity in a relatively small number of patients to try and understand, is there target engagement with this drug at this stage? And so we've added on an additional what we call a Part C study here, which is a crossover design. So once patients have gone through the SAD and the MAD and we've demonstrated safety of this drug, we can then begin to administer this drug in the imaging environment in a crossover sort of design to try and determine, can we see target engagement in that particular context? I think we're probably getting fairly short of time, but I know most people here are not MR researchers, but just to give you a very broad overview of what we're planning on doing in the MR. So there are going to be 3 areas that we're going to be looking at. And this, again, we've heard about Natalie and Abby, who are in the audience, who are going to be working on this. But there are 3 broad measures we're doing with the MRI, trying to capture seed-based hippocampal connectivity and a range of other connectivity measures using resting state fMRI. I think I'm going to skip this or go over it extremely quickly. I just wanted to use this slide to show that there are other ways that MEG or fMRI or PET can be used in this early stage target engagement. So another approach that we've been doing, working with Janssen in this case, is to try and work out how can we demonstrate target engagement in the neuroimmunology space. So within the brain, there are these microglial cells. What about, which have become increasingly interesting in the context of depression and psychiatric disease? How can we determine whether novel immune-targeted drugs show target engagement? And the approach that we've been using is this approach using endotoxin, where we take healthy individuals, we give them endotoxin, which we know results in a shift in microglial morphometry, and we've shown that with TSPO PET and also with something called diffusion spectroscopy, and then use that as a model to determine whether you use you bring in a new drug of interest that is believed to block that activation. Do you see inhibition of this LPS-induced microglial activation signal? So this is another approach that we've been working on with Janssen as another potential mechanism to trying to get at that target engagement in a super early stage in clinical trials before advancing this into expensive phase 2 and phase 3 studies. I'm going to skip this one. This is just to show that other companies are using these electrophysiological measures such as ALTO for their own clinical studies. And I think that is me done. So thank you very much. Thank you for accommodating a non-MEG researcher in this meeting, and I'm very open to questions later on after the break.
So I'm delighted next to introduce Dr. Emily Rogers. I'm sorry, we haven't met yet.
Nice to meet you.
Nice to meet you. So Emily, they did their PhD at University of Wake Forest. Yeah. And I was pleasantly surprised to see you were one of the first people to do MEG in awake primates.
Yes.
That's super cool. Yeah. And so welcome. Thank you so much for coming, and we look forward to your talk.
Yeah, so I'm a little bit of an anomaly here where I did my PhD in MEG and then took my talents to industry into clinical development and medical affairs. So I'm a little bit removed from it now, but I met with this group at a couple conferences and wanted to talk a little bit about my work, and they, were gracious enough to let me come here and nerd out with you guys for a little bit. And let me say that it was great to kind of like kick the cobwebs and dust off of thinking about this work. Okay, so again, thank you guys for having me today. Today, we're going to talk a little bit about how we can best leverage and expand MEG to address unmet needs in industry. And so this is kind of going to be a two-part talk. But first, I'll say that I have no conflicts relevant to this talk. It's my work, my opinions. I am a current employee of Neurelis and past employee of Jazz. So as I touched on, I left academics in 2021 and joined medical affairs. So I kind of have a different perspective on how clinical research can take place, as that's kind of part of my job right now, is to help facilitate that. And we've heard a lot of talk, and we'll hear more talk about biomarkers, but I'd like to talk a little bit about biomarker kind of paradigm that I worked on in grad school, using a technique called optogenetics and non-human primates. And the idea behind creating this model was to, number one, kick the tires of some of the questions we had about MEG, specifically deep source localization, but also create a robust model for drug discovery and an attempt at understanding different drugs and how they affect the body. And then the second part, we'll be talking a little bit about things that I've learned that I think would be useful to share as far as from the process from my side that would kind of help expand things into industry, or from academia into industry, and get more MEG into clinical studies. So without further ado, let's get going. So a lot of us talk today about important biomarkers in humans, but we're going to talk a little bit about non-human primate models. So preclinical trials in animals are critical for FDA approvals, and also for drug discovery. And some of them are from a pharmacokinetic, pharmacodynamic perspective, but also from an outcomes perspective. There's certain things that we know and can study in primate behavior across lots of different kinds of diseases to kind of understand both naive animals and animals that have different procedures or different drugs on board. And compared to paradigms like neuronal cell lines and things like insects and rodents, the contribution of life from non-human primates is significantly smaller. So it's only about 20%. So this isn't a ton of research, but it's really important research. But one of the great similarities is how similar the anatomy and physiology and things like social structure and cognitive capacity non-human primates are to neuroscience and to humans in neuroscience. So there are a lot of different, excuse me, primate models of brain pathology across different kinds of diseases. Most laboratories these days, you'd be familiar with macaques, but today I'm going to talk a little bit about the vervet monkey, which is a similarly used animal to study, but macaques are probably more common overall. And so kind of as I mentioned before, one of the things that I wanted to do was combine a really cool new technique called optogenetics. So if you're not familiar kind of with the basic science side of cell culture and things like that, you may not be familiar with optogenetics, but there has been some really interesting work of optogenetics and fMRI that I'll talk about later, so maybe you have heard of it. So primate research has started to incorporate optogenetics, which is essentially this technique that takes this light activated protein from an algae, this algae that's kind of lighting up right here, and you can inject it into brain areas and inserts this channel onto the membrane so that when you shine light, it depolarizes the cell and causes it to fire. Or if you are using an inhibitory, it could cause it to stop firing. So you have this really interesting control over what is going on. There's lots of different viral vectors that you can use to inject and transfect this onto the membrane, and it can be done in whatever region of the brain you choose, as long as you get really good at surgical targeting. The far right graphic here kind of demonstrates that there are some variety of the promoter regions. So you can target it to specific, like the one that I'll talk about, we did CAM kinase 2 alpha promoters. You can target different parts of the cell to make it even more specific. And it allows for this precise and repeatable and acute change to the brain activity that is dependent on whatever question you want to ask. So it was a really great tool for us to kind of say, okay, we've got some questions about MEG, we've got some questions about drug development. Let's see if we can combine those. And this is not the first time that people have wanted to do this and study it in primates. So there have been some studies looking at optogenetically induced behavioral and functional changes in fMRI. And so Gerardus and colleagues here performed a series of experiments using macaque monkeys with an optogenetic virus in the frontal eye fields and looked at changes during a visually guided saccade task and looking at differences in the bold signal because of that. And I'm not going to kind of go over this too much here, but this was really the first and only study of its kind. You know, I'm a little bit out of the game, so maybe there's been something new, but as of a couple of years ago, looking at imaging networks in awake and behaving primates. And so as we've started to talk about already today, functional networks are a really hot topic right now. So kind of understanding this constantly evolving field and how our brain functions through functional networks is really exciting. And these results were really exciting and open up a lot of questions, especially for someone or, you know, groups like us who say, okay, fMRI, what about MEG? But enter MEG, which is everyone in this room, you know, we really like this technology, but there are some pitfalls of its own too that we need to address, especially concerning, excuse me, that the magnetic fields are really small. And if we're trying to get at areas, you know, I'm coming from an epilepsy background. So if we're talking about things like hippocampal sclerosis causing seizures, temporal epilepsy, there's a lot of questions about resolving those kinds of deep signals and the appropriate ways to do that. So some of the questions that we wanted to ask was, we do know that MEG has some improvements when we're talking about spatial localization, but most importantly, the temporal resolution being superior to fMRI. Now we know that it has similar to EEG, but the fact that we can record on faster timescales is really important when looking at acute drug intervention or even seizures, because we know how the seizure and epileptiform discharges change so quickly that having something that is a little bit quicker would be really important for these models to kind of just see how they work. So what does that mean? So we've talked about primate models being very useful. These optogenetic primate models are really cool and of special interest, but only opto-fMRI at this point existed. We do know that MEG does have some advantages over fMRI, but it also has some pitfalls. So what do? We use optogenetics and MEG to address concerns in the abilities of MEG that we were having about spatial resolution. And we attempt to improve non-human primate brain mapping initiatives through this combination. Okay, so hopefully this is resolving pretty well up there. So what did we do? I took the first iterations of this experiment were started before I joined the lab, where they took anesthetized vervet monkeys that we had already injected the light sensitive virus into the CA3 of the hippocampus. And you can see here these MRIs which are a little bit darker than I would like for this screen. But there are arrows that are also distorted because of this, but we're going to go with it. We implanted a stimulating electrode, which we were able to record depth LFPs at the site of injection and transfection. So we had a true kind of north recording of what was going on down there. And we directly next to it, we implanted a little fiber optic cable that allowed us to shine light down there. This allowed the animal to be, in its cage normally, and then we would bring it into MEG anesthetized and kind of do some experiments. So we chose hippocampus here because, as I was talking about before, it is theoretically the worst case scenario here. These vervet monkeys are about the size of a large house cat. So we're talking a very, very small brain. And it's, kind of far from the sensors, but also very deep in the brain. So we wanted to kind of use this to make it as hard as humanly possible. The animals were brought into the MEG and they were sedated with ketamine and dex and then maintained under propofol. So this is being the pre-experiment, we knew we wanted to load with anesthesia as we figured out how to do the awake, but eventually I will get to the fact that we did do this in awake primates. So what we did is we did 100 flashes of light that were 10 milliseconds each, delivered with a 5-second interstimulus interval coming through that implant into the brain. And it was in the transfected region of the hippocampus. And then we used dual state SAM beamformer, which if you're familiar with the SAM beamformer, it is basically creating a statistical parametric map, which allows us to see kind of statistically relevant areas of activity. And just kind of looking at these full bandwidth recordings and these time signals or in these time epochs and seeing, what areas popped out during the stimulation period. And surprisingly, but not surprisingly, because the animal was anesthetized, we found that we ended up with the peak in the beamformer, as you can see in the right panel there, right exactly where our transfection site was. And this is confirmed when we euthanized and looked at histology on this animal, that it was essentially right there with 0.5 millimeter cubed resolution, exactly where we had transfected the site at. So it was really interesting and really exciting that we could deliver this tiny pulse and recover it with MEG in this anesthetized animal. But, that's I called it kind of like setting yourself up for success, because how often are we putting anesthetized people in the MEG and wanting to record that, or especially for drug development studies? So what I did was said, okay, I come from background of single unit electrophys in monkeys in frontal eye fields and colliculi. So let's take that implant and put it on the monkey and put the monkey awake into the MEG. And so that's what I did. So we used, you can see here, the 3-point fiducial system. This is what the animal looked like completely sitting up in the chair. The head is fixed. All of this is completely magnetically transparent, which was absolute nightmare. Trying to get all of these pieces to not break, but also be able to go into the MEG. She would sit with her head, she would sit perfectly quiet with her head straight, and we would have really, really great resolution. Really great sensor coverage, and she could just hang out there for as long as the recording sessions were. Usually it was about an hour and a half, depending on the situation. So unfortunately, because we don't have a lot of time, I just kind of want to skip forward a bit. But you can see here that this is before we actually added a secondary net plate to bring her up higher into the helmet, because the fiducials were having a little bit of an issue. But again, this was, what it looked like just for those purposes. And so once we had the experimental prep knocked down, and it was kind of important that we address how long this virus stays in the brain, because that's one of the things we're talking about is, you build this model, how long will it work? And one of the things we know about AAV viral vectors is sometimes they degrade. So you can see on the top left panel here, these are essentially a diagram of showing what the LFP is recording during this response. When a full bandwidth light was flashed, and it's kind of graded out by different light powers, just to show that it's similar in the awake and the anesthetized. So it was almost the exact same prep is eliciting the same response in the brain versus awake and anesthetized. And that response on the right, on the top right panel, is similar, again, awake and anesthetized. And then the bottom panel is just kind of showing how long that these recordings continue to go on. So the day 0 is the first day that we implanted, and we would test every week to see when we would have a response in that LFP. And we continued to have responses that didn't really start to degrade until over 1,000 days after surgery. So this monkey I was able to do recordings for years, and that's really important when you're spending a lot of time on something like this. But kind of getting to the good stuff, what was I able to do? And so we have our monkey in the MEG. We stimulate via that implanted electrode and record the data and analyze it using SAM. And this is kind of what we found. So these are 3 examples of this optogenetically evoked localization in the left hippocampus using the dual state beamformer. So every stimulation, we used a pre-stim baseline of its own baseline to then create these parametric maps. The MRIs have kind of been mirrored here, where left is true left, just for ease of viewing. And all of this data was collected on different days. So each row here indicates a different stimulation condition. So the top is a 10 hertz sine wave, the bottom is a 20 hertz sine wave, and the middle is 20 hertz and the bottom is a 30 hertz sine wave. So this is a little bit different than the anesthetized data that I was looking, which was just pulses. This is sinusoidal stimulations that were on for 1 second. And then you can see here in that chart, these are kind of the different beam forming windows that we looked at. So one of the things that I wanted to do was let the beamformer tell me what it could find. And that's what I did. So that was a really big result, that we were able to kind of successfully localize a really difficult region in an awake monkey under multiple stimulation conditions. And one of the things that I cut out of this was, because for the sake of time, which I'm doing better on time than I thought I was, but the second chapter of my thesis is using kind of where these peaks in the map were, and using that as well as default mode network coordinates that we know from Vervet Monkey Atlas, and doing functional network analysis and seeing how these stimulation patterns changed different functional network metrics like clustering and things like that, based on the stimulation parameter. So if you want to talk about that later, we definitely can. And then furthermore, we kind of wanted to examine how these tomographic maps were affected by that stimulation. So here are the results of a kind of full width half max maps for each stimulation condition. And as you can see, there's kind of considerably less downstream network activity at the 40 hertz stimulation compared to the other two. The difference in the coloring there, as well as the how dark it is. The 10 hertz oscillation definitely created a higher T-score than the other ones, but it was also more localized to the area of stimulation for the same time epoch. And the 20 hertz stimulation seems to kind of just rip through the network very quickly, which when you look at the literature and you look at these kind of bandwidths and how they are affected in the different areas, this physiologically makes sense. So it was really exciting to see that what we knew to be true in the literature was kind of being mirrored here in our primate model. So the conclusion of this part is that OptoMeg is a viable method for brain mapping studies of and in non-human primates. I also just really like the name OptoMeg. I thought it sounded really cool. Head motion and movement can be minimized with implant considerations, because that was probably the first thing that a lot of people would ask is, how do you get this animal to sit still? I can't even get humans to sit still for that long. But the recordings, that because of that, the way that the head post was, any movement then brought back to center. So if you cleared out that, little bit of movement, you were always going to end up at the same place. You couldn't kind of move and shift the way that humans do when they're getting uncomfortable. We did show that MEG can localize deep sources in small awake brains under optimal circumstances. So this is also really important. And we've talked about it, appropriate experimental design, appropriate, considerations when doing this. And three of them were, we had a really low MEG noise floor at Wake Forest. It was incredibly low. The CTF people, every time they would come, were just really excited about that. We used axial gradiometers, and that was really important to be able to measure that depth activity, and using the dual state SAM beamformer set us up for success. So shifting gears a little bit to kind of the last part of this is, number one, you may be asking, like, what's great, or that sounds great, what's been going on with that work? And unfortunately, I can't say that there's anything new going on that work. Once I left the lab, it kind of died down, but hopefully maybe with new invigoration, we can be convinced to get it back started. I took my talents to industry when I finished, and have been on this medical affairs side ever since. And one of the reasons that I joined this panel, as I talked to you before, was to kind of bring my past, my present, and what I hope the future looks like together for MEG research, and kind of how we can do that together. And so we know MEG is great. That's why we're sitting here in this room. But we know that it can be used in a lot of ways, but how do we convince the powers that be to spend the money and invest in the development, and even make it a tertiary endpoint in a trial at this point? And so thinking about that, there are some concepts that I think should be highlighted more when we're talking about industry-sponsored collaboration research with, I'm coming specifically from an anti-seizure medication side, so this could be a little bit different for drug device development. But kind of these things for collaboration trials, also investigator-sponsored trials, which there are differences in. So if you want to talk more about that, just let me know. So number one, the more data that the funding body can extrapolate from a single source, the happier they will be. A lot of people don't know, I've never heard of MEG, and outside of potentially your area's medical affairs representative or someone like me doing that research on you before they come to have a meeting with you, they may never have heard of it before. So continuing to preach the gospel, or someone mentioned earlier, like be evangelical about it, can do relatively quickly and clearly to generate a ton of data. Many have only heard of EEG before, so knowing that you can do both at the same time is something that you have to that needs to be reiterated. Especially coming from an epilepsy perspective, they know EEG, they've likely never heard of MEG. So saying, oh, you can do that both times, maximizes cost effectiveness. From an epilepsy perspective, as we move into the age of anti-seizure medications looking to benchmark and improve more than just seizure outcomes, things like quality of life and cognition and different mapping initiatives can be really important to include in these trials, because some of these, especially rare diseases, seizure freedom, while it's the goal, is very unlikely. So they're looking at other kind of secondary comorbid things to benchmark their studies. And it could be really interesting to look at this through the MEG lens. We also know that MEG has a speed advantage over fMRI, and so medication changes can be tracked by sequential recordings, both acutely and chronically. And we know that we can also still see the epileptiform areas of interest, even if the patient is on medications. I know this from my time working on the surgical pipeline, that even patients coming in on meds, you could still see the epileptic zone. So you can track that, as well as the difference in the functional connectivity of that brain. So, and as I kind of said before, cost effectiveness is really key here. The studies that are set up for success in the beginning are the ones that get funded. We know that from the NIH, we know that from other federal funding agencies. So as most people here, your MEG centers already have these pipelines in place. Sometimes people will submit for investigator-sponsored trials and they'll say, I want to look at your drug or this disease state for whatever. And they'll say, okay, I need a statistician, I need this, I need that. But most of these, our MEG centers and our groups in this collaborative way that we go about research with MEG already has that in place. So you can get things turned around very quickly and in a very efficient manner. And that it's also a positive that likely, at least in the US, when it comes to MEG centers, are usually affiliated with neurology departments. And when we're talking about clinical studies, they have a patient population that they can pull from, especially for epilepsy groups. Some patients may not be able to pay for an MEG, so this is another positive to a recruitment tool, the same way that patients who want access to medications enroll in drug trials. And then a few other brief things on it are that these studies can be done in geographically separate areas, both US and internationally, for a more diverse sampling, and can have a full spectrum of age ranges from children to adults. And so that can be very appropriate depending on what kind of drug they are looking for, or what kind of intervention, or what kind of biomarker we're looking for. So we're kind of set at a really good place with MEG already. So kind of in conclusion, we've talked about, how MEG is appropriate for both preclinical and clinical models. And, we'll continue to talk about that today. We know it works, we know it gives great data, and we know it's non-redundant, and that it can be implemented appropriately to give us a really good outcome. And importantly, just kind of leveraging what we know about MEG to educate companies on the usefulness and reliability and cost effectiveness is really key. For those of you who are sitting here that are not the epileptologist yourself, or in close collaboration with an epileptologist, that may be a great place to start, just because of that kind of pipeline. But you almost certainly work close with them, at least in the US. So that's kind of my angle that I'm coming from. So I know it's a little bit different over here. And they are the ones who are likely participating in clinical research or investigator-sponsored trials. So maybe there is an opportunity that they didn't know about or you didn't know about, that they are participating in, that maybe an MEG biomarker, an MEG surrogate is something that could be really useful. There are usually people like me who can help facilitate that for different companies, for different drugs, for different devices. So usually if you want to answer that question, reach out to someone in medical or research if you have that information. And, Sometimes it just doesn't even have to be using the drug a company does. If the company is studying a disease state or a special kind of epilepsy or a special, neurodevelopmental disability, they could be looking to just expand the research on that side, whether or not it has to do with that drug. So it could be a really great use of funds and resources that could benefit both. So quick thank you to all of my colleagues who helped with my PhD work and the current job who let me take off time to be before a huge conference in the US to be here. So thank you guys, and I'm happy to answer any questions afterwards.
Does anybody have a quick question for Emily? I think it's only fair, seeing as Emily kept beautifully to time, and it was not their fault that we were started a little bit late. Does anybody? Yes.
Hi, I was saying, beautiful talk. Thank you so much. This might be a bit of logistic and a bit of dumb question, but access to MEG in terms of bringing patients in and families and support and transport, so on. Have you, do you have any insight of that?
Well, I think it's different from place to place. You will need that resource without the within budget. It just really just depends. My industry experience is mostly in rare disease, so it's, assume that there is a burden on the patients and the families and getting caregivers and transportation, stuff like that you factor into the budget. And that's just as much as I know. Usually I'm just part of the facilitation of the study side, because what it is with a industry or investigator sponsored study is an investigator has an idea, they come to the company and say, here's my idea, give me money for it. And the company will say, that's a great idea, here's money, you have full control over that. It has nothing to do with them anymore. Then there are collaboration studies where they say, this is a great idea, we want we want to work together on this. And then there's a more heavy hand from the funding side. So it really just depends which mechanism you go through and how much support you get one way or the other. Thank you.
Okay, let's thank Emily again and for an amazing talk. And our next speaker is Christos Papadelis from Dallas-Fort Worth. Mm-hmm. Thank you for coming all this way, and we look forward to your talk.
Good morning, everybody. First of all, I would like to thank the organizers for the invitation. Today, I will present the work I'm doing together with my group regarding the development of non-invasive electrophysiological biomarkers of the levels of the GABA in children with Dravet syndrome. I would like to highlight that this is a study that is funded by Encoded Therapeutics. So these are my financial disclosures, and I will start with what is Dravet syndrome. It's a rare genetic form of pediatric epilepsy. It affects approximately 1 out of 15,000 live births. Patients with Dravet, they typically experience their first seizure at 8 months of age. This is typically triggered by fever, and within weeks to months, they experience additional febrile seizures and eventually temperature-independent seizures, which are severe and disabling. So the first mutations linked with Dravet syndrome were identified in 2001 in the SCN1A gene that encodes the A1 subunit of the voltage-gated sodium 1.1 channel. So the voltage-gated sodium channels, they play a critical role in the initiation of actual potentials in the neurons and in the regulation of their excitability. The sodium channels, they are located the axon hillock and across the length of the neural axons. And during the resting period, the channels are initially closed, so there's no any kind of ion influx or outflux across the membrane. So when a nerve pulse arrives, like the sodium channels, they are activated, allowing positively charged sodium ions to enter the cell. And following the activation, the channel does not open in response to a new signal for a brief period of time. And this critical sequence of events dictates the propagation of actual potentials towards one specific direction across the neurons. So studies on patients with Dravet syndrome have shown that mutations in the SCN1A gene, they typically produce non-functional sodium 1.1 channels. And these non-functional channels, they will cause reduced sodium current, and that's less sodium depolarization and inexcitability in the neurons. So the main idea is that Dravet syndrome is caused from what is called de novo SCN1 haploinsufficiency. It's actually a genetic term that indicates a situation in which the total level of the gene product produced by the cell is about half of the normal level, and that is not sufficient to permit the cell to function normally. However, the problem is that there's any kind of paradox here, so it's a surprise that reduced expression of a gene that encodes an essential depolarization current like cause epilepsy. And this is like because reduced sodium current like should lead to inexcitability rather than hyperexcitability. In order to explain this kind of paradox, alternative hypotheses have been proposed. Most of them, they're based on recent animal studies. One of these hypotheses is what's called the interneuron hypothesis. According to this hypothesis, like the seizures in Dravet syndrome, they arise as a result of selective defects in the GABAergic interneurons, which produce a network that lacks sufficient inhibitory tone. We can think that as a kind of a car in which like the brakes, they are broken and they don't work properly. So let's talk a little bit more about the GABAergic system. The GABAergic system is one of the two important amino acid systems in the brain. The glutamate system is responsible for what's called fast signaling excitation. The GABA system is responsible for fast signaling inhibition. So it's like the off switch in a large electrical circuit that responds very quickly and has a global effect, since like half of all synapses in the brain, they express some kind of GABA receptor. So the GABA neurotransmitter acts at the synaptic level. This is by modulating ion channel activity to two major postsynaptic receptors, the GABA-A ionotropic receptor and the GABA-B metabotropic receptors. So upon GABA transmission binding, the GABA-A receptors, they open, allowing negatively charged chloride ions to flow into the neuron, make it more negative or more hyperpolarized, and that's less likely to generate axial potentials. Upon GABA transmission binding, the GABA-B receptor is also activated, opens, and allows like positively charged potassium ions to flow out of the cell, making the postsynaptic neuron hyperpolarized and less likely to generate any axial potentials. So the proper functioning of the central nervous system depends on the balance between the excitatory and the inhibitory neurotransmitter systems. As we said before, the inhibitory system is regulated by glutamate. The inhibitory system is regulated by GABA through interneurons. So these interneurons, they control the information flow and the synchronization of the cerebral cortex, and can be seen in several brain areas, such as, for example, the amygdala, hippocampus, hypothalamus, and the prefrontal cortex. So this wide expression of the GABAergic cells indicate that this inhibitory neurotransmitter is involved in many functions in the central nervous system, such as the cognition and the motor control. The problem we face is that because GABA cannot penetrate the blood-brain barrier, that is, measures through a blood test do not directly reflect the levels of the GABA in the brain. There's recent interest to develop non-invasive biomarkers, which are based on neuroimaging tools, which can provide an indirect measure of the levels of the GABA in the brain. So there are several neuroimaging methods which can help us assess in an indirect way levels of GABA in the brain. And among today, I will talk about MEG, high-density G, and magnetic resonance spectroscopy, or MRS. So there are three basic neurophysiological mechanisms which are candidates to generate an electric and magnetic field that is measurable outside the scalp. We have axial potentials traveling away from the soma. We have postsynaptic currents at the apical dendrites of the neurons and back propagating spikes into the dendrites. And among these three mechanisms, like the postsynaptic currents are believed to mostly contribute to the electric and magnetic activity measured with the MEG and the EEG. However, there are recent evidence showing that axial potentials may also contribute to what's called high-frequency activities or high-frequency oscillations. I will not explain, of course, what is MEG and EEG, like all of us, we know what is that, but I will spend some time explaining what is magnetic resonance spectroscopy, or MRS. It's a neuroimaging technique that allows to examine the human tissue for the presence and concentration of different metabolites, including the GABA. So the MR spectroscopy is conducted on the same machine as the conventional MRI, and thus could be easily integrated with the conventional MRI scans. However, it has significant limitations. For example, it's almost impossible bring kids with Dravet syndrome or other neurological disorders and put them in the MRI scan. That's why MEG has some significant advantage. And what actually we measure with MRS, while with the conventional MRI use the signal from the hydrogen protons to form anatomical images, the MRS like using for this information to determine the concentration of different brain metabolites. So a first step towards tools that allow us like to measure the levels of the GABA in the brain non-invasively. It is the observation that drugs such as the benzodiazepines, which potentiate the GABA-induced chlorine influx, affect the oscillatory activity of the EEG signal. And although benzos, they are sedative drugs, like recent animal studies, they have shown that they increase beta and gamma activity in the EEG signal, possibly through their action on GABA-A receptors. So the question is now how we can induce this kind of frequency-specific changes in the human brain and how we can measure them with non-invasive tools such as the EEG or the MEG. So early studies on patients with implanted intracranial EEG, they have shown task-specific high-frequency activity that was between 40 and 150 hertz in the raw signals in response like to visual stimuli present to the patient. And by using MEG, recent studies, they have shown gamma band activity in frequencies around like 60 hertz induced by the presentation of visually stimuli in healthy adult subjects. And another group, like by using MEG again, showed evidence of gamma-induced activity in frequencies between 60 and 95 hertz in the primary somatosensory cortex of healthy adults in response to noxious stimuli. So in order to measure this kind of activity non-invasively, we need modern neuroscience experimental designs. Here we see, for example, a visual experimental setup we have with the MEG at Cook Children's Medical Center. We present to the child like several visual stimuli in the screen that is located in front of the child, and we measure like the brain activity in response to this kind of stimuli. So for the enhancement of the gamma-induced activity, we present like flashing checkpoints that is well known to generate the strongest possible cortical response within the primary visual cortex. In order to assess these weak brain responses, which can be easily recorded with implanted electrodes, we need to also increase the signal-to-noise ratio of the EEG and the MEG signal that we record non-invasively. In order to do so, we adopt a methodology that is based on the electric and magnetic source imaging and involve the solution of the forward and the invest problem. So we actually initially co-registered the location of the EEG and the MEG sensors with the patient's head scalp extracted from its MRI. And based on this information, we developed a head model that approximates like the patient's head electromagnetic properties. By using this model, we're able to estimate virtual channels at predefined locations in the patient's brain. And these virtual channels, they allow us like to estimate non-invasively high signal-to-noise ratio brain activity within this region without any actual invasive procedure. By using these two methodological approaches, like my team has shown in recent studies that we can detect non-invasively high-frequency activity between 80 and 250 hertz, what's called ripples, with scalp EEG and MEG. And this kind of activity was often overlain on interictal spikes and seen only in patients with refractory epilepsy. This is a study from Krish Singh's lab. He was the first to provide evidence that the amplitude and the frequency of gamma-induced responses to visually presented stimuli recorded with the MEG was positively correlated to resting state GABA brain levels, which was assessed with magnetic resonance spectroscopy. Actually, they performed a similar experiment with function MRI, and they found that the bold amplitude of the hemodynamic response in the primary visual cortex was negatively correlated with the GABA level and the gamma frequency. This is a study from Children's Hospital of Philadelphia. They showed similar findings in the primary motor cortex with the MEG by using the virtual sensors approach, like they found what they called movement-related gamma synchrony in the frequencies around 80 hertz in response to finger tapping at the virtual sensor that was located to the contralateral primary motor cortex to the active finger. So this activity was preceding the well-known event-related beta desynchronization and post-movement beta rebound, and the amplitude and the frequency of this gamma activity was positively correlated with the levels of the GABA, as this was assessed by magnetic resonance spectroscopy from the motor cortex of the same subjects. More recently, a group from Germany, they examined the correlation of the levels of the GABA with the gamma-induced response in the auditory system. By using MRS, they estimate the level of GABA in the healthy individuals on a large voxel that was located in the primary auditory cortex. And what they found by using EEG and the passive listening task, they recorded brain responses to auditory stimuli from healthy adults. Although they didn't manage to successfully record they managed to successfully record gamma-induced activity that was positively correlated with the sound intensity. They didn't manage to reproduce the findings from the previous studies that correlate the frequency and the amplitude of gamma activity with the levels of the GABA in the auditory cortex. That's a study with the Dravet kids. It's a recent study from Europe. They tested the hypothesis that brain oscillatory activity in the gamma frequency band will be altered as a result of GABA dysregulation in the patients with Dravet syndrome. They actually examined with EEG 13 children with Dravet syndrome using an auditory state-state experiment, and compare the EEG findings with them from a group of children with non-Dravet epilepsy and a group of healthy controls. The experiment was involving the delivery of auditory stimuli in the frequency of 1,200 Hertz, modulated in amplitude through earphones. And here we see the time frequency representations of the auditory evoke responses from a single participant from each group. So what they found, they found weaker or no responses in the frequency range between 1 and 120 Hertz for patients with Dravet syndrome, compared to both healthy controls and children with non-Dravet epilepsy. The healthy controls, actually, they showed oscillatory responses that were maximal in the low between 30 and 70 Hertz and high between 80 and 120 Hertz gamma range, both in the power and in the intertrial coherence estimates. So these kind of findings indicate that suppression of gamma activity in children with Dravet syndrome compared to healthy controls, and support our hypothesis of GABA suppression in children with Dravet syndrome. This is a paper my team published recently. It presents the findings from a project not on the Dravet syndrome, but in another GABAergic disorder called SSADSD, or succinic semialdehyde dehydrogenase deficiency. Don't ask me to say that again. It's a kind of an ultra-rare genetic epilepsy. It has some kind of similarities with the Dravet syndrome, and is possibly also caused by GABA deficiencies. The work was performed in my previous institute at Boston Children's Hospital and Harvard Medical School together with a PI there, Philip Pearl. So we hypothesized that patients with SSADSD will present altered somatosensory responses in the beta and the gamma frequency band compared to the healthy controls. And in order to test our hypothesis, we recorded somatosensory evoked potentials with high-density EEG from patients with SSADSD and healthy controls. In order to elicit the somatosensory responses, we delivered pneumatic air puffs through plastic tubes, which are attached to 4 fingers of their hands bilaterally. And based on the patient's MRI, we estimated their head models. And by solving the inverse problem, we calculate virtual sensors, which were placed at the primary somatosensory cortex contralateral to the stimulus site. So we also assessed the levels of the GABA in the patient's blood through plasma specimens and in their brain through MRS. Here we see the time frequency analysis plot among all participants for the 2 groups, the SSADSD group and the group of healthy controls. Each graph represents the power changes after the pneumatic stimulus in the virtual sensors, which were placed at the primary somatosensory cortex contralateral to the stimulated site with respect to the pre-stimulus period. So the red color in this graph indicates an increase in the signal power or what is called synchronization effect, and we observed a significant lower power or event-related desynchronization in the beta and the gamma frequency bands at different timings after the stimulus onset. We observed a very strong negative correlation between event-related power synchronization in the beta band and the levels of the GABA in the blood among all participants. And from this graph, we can see a clear classification between the patients with SSA-DSD and the healthy controls in terms of beta synchronization and the levels of the GABA in the blood specimens. We observe similar findings for the gamma activity at the early time window, a moderate correlation between event-related power synchronization in the gamma frequency band and the levels of the GABA in the blood among all participants. So based on all this evidence like that I present today, we have initiated a project at Cook Children's Medical Center, which is funded by Encoded Therapeutics. The main goal of this project is to develop non-invasive tools that assess the levels of the GABA in the brain and correlate these findings with the level of the GABA in the blood. In this study, we plan to use a combination of different neuroimaging techniques and correlate this kind of indirect measures of GABA with the ones derived from the measurements of the blood. So we managed to recruit so far 6 typically developing children and 8 children with Dravet syndrome and SCN1A mutation. We try to match the 2 groups in terms of age and gender as much as possible. And we performed 3 different experiments. The one was involving the visual stimulation. We actually deliver visual stimuli, flashing actually checkboards on top of some cartoon figures. An auditory experiment where beeps were delivered through speakers, and somatosensory experiment through compressed air puffs, which delivered to the child's fingers. These are the blood plasma findings. We observed significant differences between the 2 groups. In line with our hypothesis, we observed a significant suppressions of the levels of the GABA, but also suppression in other metabolites, like, for example, serotonin, aspartic acid, glutamic acid, and others. And in terms of EEG findings, similar to the results we obtained for the SSA-DSD, we observe a significant suppression of the gamma activity in the high frequencies, around 80 Hertz for children with Dravet syndrome compared to the healthy controls. By focusing on the entire gamma frequency band, we further estimate the relative power change with respect to the pre-stimulus period and estimate the peak frequency and the power in the gamma frequency band. And we observe a significant decrease in the peak frequency for children with Dravet syndrome compared to the healthy controls for the gamma frequency band, and a marginally significant decrease in the gamma band power for children with Dravet. MEG obtained similar results with a high-density EEG, significant suppression of the gamma power in frequencies slightly lower than the EEG, around 60 Hertz for the children with Dravet syndrome compared to the healthy controls. And again, by estimate the peak frequency and the relative power with respect to the pre-stimulus period in the entire GABA band, we found actually no difference for the frequency, but a significant reduction in the gamma power for the children with Dravet syndrome compared to the typically developing children. We're still in the process of analyzing the auditory evoked responses and the somatosensory data for the same cohort. So these are very preliminary data. We observed, though, the same phenomenon, suppression of gamma evoked activity in the gamma frequency band for the Dravet children compared to the typically developing children. So we can conclude that our findings may increase our understanding of the pathophysiology of Dravet syndrome, as well as other neurological disorders of GABA metabolism. We can validate non-invasive systemic biomarkers of brain GABA metabolism, and these kind of biomarkers, they can serve as essential tools for future clinical trials for patients with Dravet syndrome. I would like to thank my team for all the hard work and dedication, but most importantly, the patients and the families who participate in our studies. Thank you.
Thank you very much.
Mm-hmm.
Could I use chair's privilege and ask a question?
Of course.
So the peripheral blood markers showed a reduction in GABA in the children with Dravet. What's the mechanism for the blood GABA to be reduced when you said earlier in the talk that the GABA can't get across the blood-brain barrier?
That's a very good question. So actually, I think that it's kind of an indirect measure. We I think that like we're able to get this kind of correlation because like some kind of GABA modulation in the blood possibly like reflects in the brain.
Common mechanism.
But it's a common mechanism, but like the whole clear mechanism, how does this work? It's unclear. So, but that's an excellent point. Yeah.
Thanks.
Yeah.
Do we have time? I think we have time for one quick question. If anybody's got a pressing question, otherwise you can get Christos. Yeah, please. Yeah.
Hi, thanks. That was really interesting. I'm just wondering, is this? I missed it. I missed a bit in your talk where you mentioned it, but is it thought that the GABA deficit is purely kind of homeostatic? It's like a natural response to the reduction in excitability.
Yeah, that's correct.
Okay. But it's so, it's impressive. It's so profound that it can then lead to epilepsy.
That's actually the idea of the whole paradox. Like, so the idea is like, for example, for the Dravet syndrome, like we know that the basic mechanism in the sodium channels, and then we should expect that this kind of will cause like kind of suppression of the whole activity, but it causes epilepsy. That's why like all these other hypotheses have been introduced there with interneural hypothesis. So the main idea is that possibly that the GABAergic system is there's deficit there, and that's why like it's like what I said before, like the brakes there in the car, they're broken, then there's no inhibition actually.
Okay. And are there any other examples of homeostatic pathologies like this, or is this a unique?
I think there's also in other disorders, like I don't remember out of top of my head now to name some of them, but I think like Dravet syndrome, like it's I think one of the more common like GABAergic disorders. There are several others, but like they're more rare, and that's why I focus my work on Dravet, because like it's rare, but it's not so rare. Like so, there's some interest from the industry to present some evidence for biomarkers and then apply to also other like other disorders of GABA.
Cool. Thanks.
Mm-hmm. Sure.
So let's thank Christos again for a brilliant talk. Okay, everybody, we're going to start, and I'm delighted to say that our next speaker is Hanna Renvall from the Biomag lab in Helsinki. Hanna is actually head of the MEG lab there, so thank you for coming, and we look forward to your talk.
Thank you for the invitation. It's great to be here with you, and you there also on online. I just got back the reviews from my teaching at Aalto University, and the most negative comment was about my speaking speed, that I speak too fast. So I will try to now start this learning curve for myself already today. So I will not talk today about pharmacological interventions yet, but the patient group that I will address in this talk is and continues to be one of the core interest groups for drug development. So after more than 20 years, we now have first new drugs at the market, at least in the US, in trying to delay the cognitive decline in Alzheimer's disease, which is the most common of neurodegenerative diseases. So we are critically in a stage that we should be able to find those patients who are most likely to benefit from these new drugs, and especially find these patients early enough. So dementia is a huge burden to the individual themselves, but also to their close ones and to the society. Mild cognitive impairment, MCI, refers to a stage which is in between normal brain aging and dementia. And we have, at the moment, approximately 10 million people in Europe alone who suffer from mild cognitive impairment. And these numbers are expected to double in the next 20 years. So this is a especially interesting group, as we know that a huge percentage of these patients or subjects are in risk of developing into clinical dementia in 5 years. So not all of them, many of them stay stable or even get better, but we should be able to find those ones who have this huge risk of progression. Well, here MEG and EEG comes into the picture. And why is that? Well, we know that MEG and EEG can be highly informative on finding the first pathological changes that can be detected in the neural networks of Alzheimer's disease patients already years, even decades before the clinical symptoms. And here's, I think, really nice picture from a paper by our dear friend Fernando Maestro and his collaborators, who kind of made the picture, simple picture of these things. So the idea is that when we know that Alzheimer's disease is kind of mainly described at the neuronal level by the amyloid beta accumulation outside the neurons, we know that this amyloid beta accumulation causes disruption at the synaptic level, promotes first kind of hyperexcitation at the neural level, but then starts to imbalance the excitation inhibition levels at the small circuits at the neurons, and then finally result in real network dysfunction. So larger scale brain network level dysfunction, which in the end is supposed to cause the cognitive impairment in these patients. Well, we know that MEG and EEG measure neuronal processing, and we already know that MEG and EEG can reveal early alterations also in MCI. So one of the early seminal studies was by Nakamura and collaborators some 6 years ago using MEG, where they showed that actually fairly simple power spectral measures can already discriminate between mild cognitive impairment patients and age-matched healthy controls. So they were able to show that there are large areas around the cortex where there's an increased slow frequency activity in the patient group, while at some other areas, normal oscillatory activity is decreased compared to the controls. Another, maybe my favorite study on this subject, comes from the Spanish group by Fernando Maestro, so where they have been addressing MEG-EEG functional connectivity in these patients, and they have been starting bringing artificial intelligence, AI, into the picture. And what they have done, they studied patients who fulfilled the criteria of mild cognitive impairment. So they had clearly already some objective cognitive decline markers, but they were not demented yet. And they followed these subjects for 3 years, and they had MEG at the first point and also at the later point. But what was really informative, that when they looked at the first MEG results, and especially these functional connectivity measures, 3 years after the first measurements, combined with fairly simple machine learning algorithms, they were able to quite nicely discriminate those subjects who suffer from progressive state and who not. So as you can see from the picture, there is clearly overlap, but quite nicely they are already discriminated. Well, our colleagues in Italy went a bit further. They used EEG and combined this functional connectivity together with genetic information. So we know that there are certain genes, especially related to ApoE gene, which really increases the risk of Alzheimer's disease even to 3 times of that in those who don't carry that particular allele. And they were able to get into a really good classification accuracy between the controls and the progressive MCI patients, almost 100%. So you would think that, okay, it's done, we just go to the clinic and that's there. Unfortunately, this is not the whole truth. So then when you start taking these measures to new patient groups in a bit different countries, so the people are a bit different, their diagnostic criteria are a bit different, everything maybe is done a bit differently, then the numbers drop. So what we need large unified data sets. And this is what we have now been recently doing in a study called AI Mind. So this is a huge European Union Horizon 2020 supported study where we have been collecting data, EEG and MEG in Finland, from altogether 1,000 participants. We are just like 2 months ago, we reached this 1,000 participants. Of those, 221 are at our laboratory in Helsinki. So Oslo, Madrid, and Rome have been the other participating clinical centers. So what do? We have been measuring electroencephalography and in our laboratory also MEG from these patients. We are trying to find such measures from the EEG and MEG curves that would best predict the network disturbance in these subjects. Then we combine this information with other information like genes, blood biomarkers that are also emerging as good tools to predict cognitive decline, especially in Alzheimer's disease, and then also cognitive tests, not only the traditional neuropsychological tests, but also computerized, easy to do even at home cognitive tests. And the idea is to bring all this together into an artificial intelligent algorithm, and in the end, have a combined dementia risk assessment. Cognitive testing and these EEG, MEG measures are conducted 4 times within 2 years. And we are now at the point that the first subjects start to come for the 4th visit. So this has been a heavy thing, as you can imagine, to our research laboratory. And here's a picture of one of our younger subjects. And I couldn't help using Dennis's nice reference to dynamic duo about EEG and MEG. So we have been really collecting EEG and MEG simultaneously. And why to measure both? So we believe that it can benefit the source localization, but also that M can help in interpreting the EEG findings and maybe help to fine-tune the EEG measurements and our analysis in the future. And also, Dennis here said that one should not maybe put EEG and MEG against each other, but after measuring these 221 subjects, many of them many times already, I have to say, especially in front of this audience, that it's so nice to use MEG when you can just basically get the subject in 15 minutes compared to EEG. For example, here we have 128 channels, so it takes like 45 minutes to get a good quality cap on. So you can imagine that everybody's tired at that point, and that is not maybe the good thing for the data either. So here are some facts of this project, which is still ongoing. So we are kind of halfway, bit more. So we have 8 countries collaborating, 19 partners altogether. Russia is also now involved in this study, and it's really nice to start collaborating also with Dennis here. This project is also collaborating with, for example, eBrains, which is a large European digital research infrastructure, which is accelerating collaborative research between organizations and also researchers across different fields of neuroscience. So we are actively working there as well. And okay, 40 million sounds a lot, but I have to say that it's not that much in the end. Well, but here is our big goal. So if everything works, this is what we would like to have. So that when we would have subjects who show symptoms of mild cognitive impairment, we would have a clinical examination, then MEG or EEG, hopefully some kind of automated analysis to make it clinically easy and valuable. And then we would combine, if needed, this with other measures like the cognitive testing and blood samples, for example. So the goal would be early risk assessment, which could be done fairly rapidly, because nowadays the clinical practice is that when we see any symptoms, we typically say that, okay, there might be something, see you in a year. And this is, of course, not enough. If we know who are really in high risk, we can start a risk factor recommendation. We know that even for Alzheimer's disease, we can delay the progress if we control blood pressure, cholesterol levels, diabetes, and so on. So those are already things that we can do. And future, I think these are our primary targets for medications as well. But of course, this data is excellent for many other uses as well. So it's huge, and we have started looking at many other things from it already. So for example, it's quite seldom that you have 1,000 subjects who are kind of measured in fairly homogeneous circumstances. And so we have, for example, started looking at the within session stability of these features. You would think that this is kind of the first thing you need to do when you think of these as clinical tools, but like said, it's not easy to get big enough data samples for these kind of questions. And this is exactly what we have been, for example, doing lately. So it is clear that our really typically used simple spectral features like alpha band power, beta band power are highly stable measures, which is a very, very good thing. And similarly, the peak frequencies of this. So if you look at the intraclass correlation numbers, 0.95, 0.90, they are really, really good. And again, here, I have to say that MEG is a bit better than EEG, probably because there are a little bit less artifacts in the measures. But these are kind of important things to address also from these kind of data sets. Of course, we have already started a little bit looking at the real question as well. And for example, we have been looking at the first markers on combining functional connectivity measures with memory scores. And indeed, it looks like that even in really small cohorts, we start seeing a correlation between the MEG measures and memory scores. But this is all highly like work in progress. But this here only brings together two markers, so MEG and the memory scores. How about all the other stuff we have? We have the genes, we have the blood biomarkers, and so on. And for here, I would like to introduce you one way of addressing these things and one maybe solution for bringing all this informative data together. This is a model that we have been working for years already in Aalto University, together with Professors Riitta Salmelin and Samuel Kaski. And we have been implementing one type of machine learning model called Bayesian reduced rank regression on MEG data. I will not go into the details, but it's an example of a latent variable model which aims to explain kind of high-dimensional data like MEG with a smaller set of predictors. And it provides a framework to utilize other information, so meaningful priors as well. So for example, we can bring into the model other clinical variables. And this is, of course, really, really nice idea in clinical populations. And in healthy adults, we have used this model already for some years to, for example, show that functional connectivity and simple power spectral measures can be deducted to a few components that remain really stable in the subjects, independent of tasks they are doing or independent of the exact date when they're coming to measurements, shown here, for example, for 3 successive measurements from same subject over months. So these are kind of suggestive that there are kind of brain-level fingerprints which we can access with these kind of measures. We have already brought this together with some other information like genes, age, and working on some other fields as well. In this earlier study by Leppa and others, we actually measured sibling pairs. And we asked from the model that can we find features from MEG which are similar in siblings and different between families. And we were able to deduct that kind of informative components from the MEG activity, which clearly grouped siblings together in a 2-dimensional or multidimensional space. We have also brought age into this question, actually EEG data from 800 healthy children in Helsinki area, and their EEG was addressed so that we were both implementing also the age of the children into the data. And as you can see a kind of clear gradient of age there. So apparently this kind of model might be really informative, and maybe we will not have one measure in the end which will say that, okay, this is a patient, this is a control, but maybe some kind of group of measures would be brought out with this kind of approach. I'm coming to end of my talk. Of course, the big question that is that when MEG, EEG, all these models and dementia they are really used in dementia diagnostics. So when are these really in the clinical practice? And I have to say that it somewhat is about there already. So the Dutch already did it. So in Amsterdam, the memory clinic actually already uses MEG as a clinical tool. They don't have, it's not fully pipeline for everybody yet, but part of their cognitively impaired subjects already go through that line. And this is something that I wish we could also implement in Helsinki quite soon too. So thank you for listening. And then, of course, thanks to huge AI-Mind team in Helsinki alone, and then, of course, the whole consortium for this data. Thanks.
So very interesting talk. I was wondering, so they're using it clinically in Amsterdam, which I saw that paper, and you talked about it here. How do they use that information that kind of ties into the pharmacology of it, or?
So basically what they do that when they have the patient coming to the memory clinic, so then they invite parts of these patients that basically, I think they're evaluating only a couple of them per week, so only minor proportion of the whole clinical group. But they go to MEG early in the week. And then they have a kind of automatized algorithm which suggests whether they are progressive patients or not. So it's based on AI models from the big data cohorts. And then they have a clinical big group, which is kind of meeting. Then doctors meet together, first discuss the results, and then they decide how they will go through this with the patient. So the patient doesn't get a paper saying that you maybe have a increased risk or not, and so on. And I think this is, of course, a big thing that there's still, of course, big leap from these results to the actual patients. And also in AI-Mind, we are at the moment eagerly and thinking that what is the thing that we can provide our participants? So what kind of information should we give them? How far are we with the information at this point? So because, of course, we are not supposed to harm with either. So I think we are still in the phase that we need to define what is the information and how it's going to give into these subjects.
Yes. Thank you. Fantastic. It's fantastic to hear about the AI Mind. It's an extraordinary project. So I have a conflict of interest because I'm connected to AI Mind indirectly. And to get to 1,000 people on time, on budget from multiple countries is an extraordinary achievement. So first question is, how did you make that work? And what lessons in an MEG community can we take away to make such multinational data sharing studies work? Related to that is MEG versus EEG. You've avoided, which is better, arguing. It's more a question of a mature discussion about when you might use both. So what would you tell investors, for example, a drug company investing in one or other technologies, about when they should think about EEG and when they should think about MEG to help them forward? So one is practical, how do you make AI Mind work? The other is, what would you tell a pharma company where to invest?
Yes, it's indeed has been a big thing. So even our laboratory has been there almost 30 years, but we never had this kind of a study where you recruit the subject, but then you check that they fulfill all the possible criteria and inclusion and exclusion criteria. And I have to say that also the pipelining of the measurements. It has taken a lot of time, but I think now after 2 years, I think we could now make a book how to do it. So I think now some of my colleagues said that when she comes to the lab, it looks that you are like a machine. But it took some time, and especially when you are talking about this kind of patient, you know how this subject group is, but it's very fragile in many sense, and they don't usually, very often they don't remember when the appointments were. So you need many practical things as well. But so I think it has been a long effort, but 2 years it took to kind of learn to the practices. But as we go to that, I think here MEG is clearly showing its fingers, because the ease of its use, especially in this kind of studies where actually the patients need to come to the lab many times. They come for the cognitive testing, they come for interviews, they go to the lab. So it's a lot of things. And what they remember that it was a heavy day. So all the half an hours that you can get away from there, it increases their probability of coming for the next visit, I would say. And also, like now, when I completely appreciate the cost effectiveness of this that we have with EEG, but I think this is one of the points where MEG is likely to also have a new important field at the clinical practice, because it's completely different thing to measure a participant who is awake and not falling asleep after 45 minutes of putting on the cap. But I do think that at this point we do benefit from having both MEG and EEG. I said here that that we can probably make the social organization better with having both. How to do that in practice? This is of course also something that I know we will have to try. And there are ways of doing that, but this is hope that it will make it better. But I cannot, of course, know yet, because it's not that heavily studied either in really big cohorts and so on. So it's a bit question mark. But yes, and of course at this point we are also let's say that they're having the MEG and EEG together. It's not only easy. So we are still struggling with certain artifacts that we have also learned during the 2 years, that how to do this so that both of the measures are best possible. Unfortunately, it was not the case in the beginning, because we were not used to doing it this way. But now getting better.
Thank you. That was a really is a very fascinating project. You have this longitudinal component in it, and I'm wondering, is 2 years enough, or what do you really need? Do you have information if you really see changes over time, or just daily performance changes, or how long should these studies be?
Very, very good question. I think, yes, I could say that 8 years would be nice. So I think 2 years is really, really short. So this is the thing that we are constantly thinking that how to follow this, at least clinically, later on, because it's highly likely that many of our subjects they are in such an early state at the beginning of this project, that during 2 years the clinical changes will not be a lot. So we will clinically follow these 4 years. But I think when we think about other markers, and then going 5 to 8 years or something, then starts to be informative enough. But this is, of course, here comes now the money challenge, that you have a project for 5 years, you need to get as much out of it as possible. We have been, of course, looking at these measures, like now when they come every 8 months, that how stable do they stay? And at least, let's say, the 8 months it's not much of a difference. I think that's kind of, they don't really change. But I think that's also a good thing, because that shows us that these are reliable, stable measures to be used here. So of course, if they would be going like this, we could just throw everything to garbage. So that's a good.
Thank you.
Thank you. So I think we'll back again.
So our next speaker is Laura Hughes from Cambridge. Welcome, Laura.
Thank you. Good, fantastic. Thank you. Thank you so much for having me here and inviting me to talk about our work. So I'm in the Cambridge lab with James Rowe, amongst others as well. We have our very new TRIAC system, which we are loving because it's working really well. But the data I'm going to talk about today are from our previous system, which is the Neuromag system. And I'm part of the Cambridge Centre for Frontotemporal Dementia, so a lot of our work is really focused on looking at dementia and really focusing as well on symptomatic treatment. So is there a role for MEG for development of pharmacological treatments for the symptoms of dementia? So if we take, for example, the pathogenic cascade of dementia, the probable causes in the environment and the genes, and we look at the changes in the brain, the neural changes, you can see that MEG sits really nicely to measure these kind of downstream effects of the pathology. And it also then will link in really clearly with the symptoms, the clinical presentations of patients that we see. So changes in behaviour, cognition, quality of life, and ultimately survival. And we don't just take MEG on its own. It's one of our primary measures, but we can inform the MEG using information on synapses from PET imaging. We also use structural MRI and spectroscopy to inform us about the level of neurotransmission. And of course, we're looking at therapeutic treatments using different drugs. Can we probe the system? Can we measure these effects of drugs on neurotransmission, on the measures that we're going to see using MEG? And then the next step is, can we link these changes to changes in behavior as well? So it's kind of like a really two-step process to bring all of this information, pull these threads together to see whether we can actually end up with a possible target for treatment of symptoms for the patients. But the patients I'm going to talk about in the studies today have frontotemporal lobar degeneration. In particular, there's two types: behavioural variant, FTD, and progressive supranuclear palsy, PSP. These patients have frontal and temporal atrophy, as in the name. There's some overlap in the pathology. They're both fairly rapidly progressive diseases, and they're very common, one of the most common types of dementia in the under 60s. So it's really kind of impacting people with a huge burden on care and on health services as well. The symptoms overlap in these two groups in terms of they're very apathetic, they tend to lack insight into their own illness, and they can also be very disinhibited and impulsive, and that does go together with the apathy. It's almost like, I don't care, I'm just going to do it, and less thought is put into the behaviours, which can end up with quite a lot of risk-taking behaviours, which again adds to health problems. So in this patient group then, what are we going to target? So that's our first question, is do we have a target that we could even possibly treat? And the target that we're looking at is the neurochemical deficit, so the loss of neurotransmission in these patients. And we measure that using MRS spectroscopy. We've looked at different regions in the brain, but here I'm showing data from the right inferior frontal gyrus. So we've got a node there. And comparing with controls, you can see that those with FTLD, so the PSP and the BV-FTD patients, have a loss of glutamate and a loss of GABA. But probably one thing you can notice the most is that there's quite a big variability in the patient group of this neurotransmitter loss. So that's something that's really key for us to take from the spectroscopy into our MEG data to try and account for some of the variability in the patient groups. And this is backed up with a lot of post-mortem studies as well, showing a loss of pyramidal cells, a loss of neurons, and a loss of this neurotransmission in GABA and glutamate, amongst others as well. So it's not just selective to these. This is what our study is going to be focused on, but it's not selective to GABA and glutamate. There's actually fairly large, wide sweeping loss of other neurotransmitters as well. But the nice thing about the spectroscopy is it is contemporary to our MEG scan, so we can run the spectroscopy and the MEG scans within a close time frame and don't have to wait, obviously, for post-mortem results to come through. And as well, bringing in our key relationship with behaviour. So we want measures that we can change and that have an impact on behaviour. So this was run by Alex Murley, and he has showed that his measures of MRS in GABA and glutamate correlate with the stop signal reaction time. So this is a measure of response inhibition, so how good people are at stopping a very strong prepotent response. And those who were better at stopping or inhibiting their responses had higher levels of concentration of GABA and also glutamate. So potentially then we're looking at people who may benefit more or less from taking different pharmacological treatments. So as well as having a target for treatment, we also need to know what we're going to actually measure. As we all know, MEG has multiple measures. There's many ways that we can analyse our data. So we kind of really want to narrow it down and be quite specific about what our outcomes are, as well as our kind of clinical endpoints. We've shown in a number of different studies, and here I'm only going to show a few of them, that the patients with FTD have a loss of beta power. So here, Sarbasamy, he showed that using resting state data, the patients had a loss of beta power using local efficiency partial directed coherence. And this was in contrast to a similar group of patients who had Alzheimer's pathology, so those with posterior cortical atrophy or classic Alzheimer's disease had a loss of gamma frequency. So although I wouldn't say loss of beta power is specific to FTLD, it could be something about the FTLD pathology that's causing a loss, particularly of beta rather than perhaps gamma power. And we also see this across other tasks as well, so not under other paradigms, and not just resting state, but obviously a motor task when it comes to beta powered, you would automatically think that's going to be a very clear example to show changes there in our patient group. So we've used a number of different motor tasks. Here I'm showing go, no-go tasks and stop signal task. So on successful go trials, again, compared to controls, the patients with FTD have a loss of the beta desynchronization. So that's the blue blobs you can see up there. So the patients have a loss of their beta desynchronization for successful go trials. So they are pressing, so these are successful trials, but they're not desynchronizing to the same extent as the control groups. But it's still very closely related to reaction time. So the beta function is still the same function as the control. So those, so the peak reaction times for pressing the button, highly correlated with the peak time of the beta desynchronization for controls and for patients. If we look at the response inhibition tasks, so in our disinhibited impulsive patients, they were able to complete these tasks. They're very simple tasks. We haven't added any kind of extras to our no-goes or to our stop signals. So these are successful trials, so they're successfully inhibiting our response. But as you can see, again, they have less beta desynchronization. So these are prepotent, so the go trials are prepotent response. They really generate a need to go or the urge to press. So as soon as they see a stimulus, when it comes to an inhibition trial, a no-go or stop signal trial, they actually have to withhold a button press. So they do have, so controls and the patients will still have that urge to go, which will invoke a beta desynchronization, but less so in the patients. And we thought, well, why might that be? Is it related to their impulse control, their disinhibition and their impulsivity? So it does, we did show it related to clinical measures of disinhibition. So those patients who are more disinhibited, in order to have a successful response to a no-go trial or stop trial, they had to have very minimal desynchronization. If they had any beta desynchronization during these trials, they were far more likely to press the button to go. So they really had to have very little desynchronization. Whereas controls obviously can still have the beta desynchronization, still have that flinch or that urge to move, but they were still able to inhibit the end button press. So we've shown then that beta probably is a very interesting frequency for us to focus on with our drug trials. So what drugs can we use? There's loads of different drugs. So we have to really kind of focus what's going to be safe for the patients with dementia and what are we going to be able to measure using the MEG. We've used multiple studies. I'm actually on our 4th drug trial at the moment, which hopefully will have some results towards the end of the year for. But these 2 studies I'm talking about now, we've used tiagabine 10 milligrams, and luckily we've heard a little bit about that already. It's a GABA reuptake inhibitor. It increases endogenous GABA, and it's typically used as an anti-seizure medication. So it's used in treatment for epilepsy. Very rarely in our patient group, I don't think any of the patients are normally treated using this drug. And also, so that's focusing on the GABA side. So on the glutamate, we've got memantine, which is a non-competitive NMDA glutamatergic receptor antagonist. It actually works to preserve the function at the synapse by inhibiting the influx of calcium ions. And this is used in Alzheimer's disease. Again, rarely in FTD, but occasionally people will have been prescribed it. I wouldn't say it's not common, but it wouldn't be completely surprising if somebody did come in and was already on this drug. It's also not just specific to NMDA. So it also is an antagonist for serotonin, cholinergic receptors, and an antagonist at D2 receptors. So we kind of have to keep in mind that this isn't just targeting one neurotransmitter. Actually, it can be working on a whole system. Unlike tiagabine, which is a much more focused just on GABA. Okay, so we've run randomized, double-blind, placebo-controlled studies with 2 cohorts. separate cohorts of patients and controls on these two drugs. They come in 2 weeks apart. So on one session, they'd have the drug, and on the other session, they'd have the placebo. And this is randomized, so we don't know who's taking which drug and when. Although we usually do ask at the end of the sessions to say, you know, which drug do you think you're on, just to try and understand really how blinded everybody is to the study. And I will say that with memantine, nobody knew. With tiagabine, occasionally it was obvious, and the patients could tell. We've used a different type of movement task, really, to try to look at whether the drugs can help change behaviour in the course of the task itself. So we've called it the CAR task, controlled action response task, where they're presented with a little cue in the shape of a car, and they use a joystick to move that car into a target space. And the car changes speed from time to time unpredictably, so that they have to then adjust and adapt movement to get it into the target space as quickly as possible. And the measures we take from this is the error that for moving the car. So it's an error measure which takes into account movement time and how on target the car is. So we focus on our two groups here. So this is just on the placebo. So we've got cohort 1 on the tiagabine and cohort 2 on the memantine. And again, so we're just showing the time frequency plots in sensor space from time point 0, which is the onset of the cue, to the movement, which is the beta desynchronization. And again, you can see that controls obviously would have the desynchronization with the movement, but that's lost in the PSP and the FTD groups. More so in the PSP groups, it's barely evident at all. And this really is reflected in the behaviour. So if we look at behaviour over trial, so looking at how the beta desynchronization and the behavioural measures from the task change over trials. So for behaviour, you can see that there is some change. You would expect a bit of improvement over trials, and we see that in controls and to some extent in the FTD groups. But the PSP groups in red, you can see there's almost a flat line, almost positive, where they're actually getting worse over trials. So they're very impaired at both in the MEG and in their behaviour of doing this task. But they are attempting the task, so we are measuring them doing the task. It's just that their errors are much higher than the controls. So we start with the memantine. This here, we're just showing the difference between the controls and the patients. So obviously there was a loss of beta desynchronization in the patient groups. There was also a loss of beta desynchronization over trials and in relation to error, so in relation to task performance. So over trials and with task performance in the controls, beta desynchronization was enhanced, and this was lost in the patients. But there was no, in this sensor space, there was no group by drug interaction. So memantine wasn't changing the sensor space time frequency plots in beta or other frequencies. We take this into source space. We source localize the beta desynchronization, and then for the patients and the controls and the drug versus the placebo. Compared to the controls, as you might expect, the beta desynchronization is lost over bilateral motor regions. But if we look at the effect of memantine, we saw that memantine decreases the beta, so it enhances the beta desynchronization. And we always get a little bit confused about these kind of double negatives, but beta desynchronization here is enhanced by memantine in the FTD group, in what looks, what kind of localizes towards the left pre-SMA, so quite obviously a very motor relevant region. And also if we take now into account the glutamate measures, although there was no effects of, or there's no relationship of the beta with levels of glutamate as measured by the spectroscopy, there was a relationship between the change in error, so their behavioural performance, and measures of glutamate. So those with higher levels of glutamate concentration had greater benefit of memantine. So they improved their performance, reduced their error over trials more than people with lower levels of baseline glutamate. Coming to tiagabine, then again, so kind of show that there was a loss of beta desynchronization in the patients compared to the controls. But this time there was effects of drug. So here in the next two plots, this is showing the effects of drug in the patients more than in the control. So the drugs had a greater effect in the patient group and enhanced their beta desynchronization more than in controls. And this was enhanced more in the PSP group than in the FTD group. So tiagabine was enhancing beta desynchronization. And again, we can localize that using beamformer. So showing that the beta desynchronization was reduced by disease in frontal and parietal regions. And tiagabine was enhancing the beta desynchronization in prefrontal regions across all patients. And the difference between the two groups, the PSP and the FTD, was in the right parietal cortex. So again, kind of areas really key for motor control, response inhibition, and movement. But then as our really key question is not only can we change the beta desynchronization, but does it have any effect? Does it improve performance? In controls, there was very little effect and very little effect on behavior. In the patients, there was actually quite a substantial effect on behavior in both groups. So in the FTD group and the PSP group, their behavior improved more over trials when they were on drug. And this is especially evident in the PSP group in the red on the far right, whereas you can see on placebo, they had kind of got worse over trials. On drug, they actually improved their task performance over trials. So there was some, hopefully I've shown there's some benefit then for some patients taking memantine or tiagabine. And the last bit I'll go through as well is like, if we're going to go for drug discovery, we're going to understand, which drugs could be used. Because although these are quite safe drugs, taking something like tiagabine is not ideal for patients with dementia in the long run. And these are just single sessions that we're running. So we're not running kind of longitudinal data to see what's the effect of kind of chronic administration of these drugs. But it's still really key to understand what's the mechanisms in health and in our patients. How do these drugs work? And what are we actually measuring in terms of the MEG? So this is work by Amir Jafarian, and he took some resting state MEG data, and he used dynamic causal modelling, and Chris touched on this earlier. But what he's done is created a model. So this is a model of a particular node here, right prefrontal cortex again. And he's kind of created a model of the different cell populations, the pyramidal and the inhibitory interneurons, and the connections between them, as well as including the conduction and the ion dynamics of this kind of cortical microcircuit. We can then measure the how well this model can represent our data. And also he included measures of the spectroscopy again. So we're looking at the variance. Can we explain some of the variance in our baseline measures, even in health, using spectroscopy? And what he showed was that baseline measures of GABA, so this is in our healthy controls, also have some variability, but they're all ostensibly healthy controls, that the baseline measures of GABA were associated with the recurrent inhibitory intrinsic connection. So here highlighted in yellow. So those people who have kind of naturally higher baseline levels had stronger inhibitory intrinsic connections. And glutamate was associated with the excitatory connections of the superficial pyramidal cells with the deep layers and with the inhibitory interneurons. So again, those people who have higher baseline measures of glutamate had stronger connections here. So lastly, then, what about our patients and what about our drugs? Can we use these types of models that we can show work in health to explain the effects of drug and the effects of neurotransmission loss in the patients? So Tally Adams took a very similar model, again, this cortical microcircuits, and also included the baseline MRS of the patients and took the sessions from the GABA, the tiagabine and the placebo sessions to understand what's the difference in baseline in the patients and how does the drug change these connections. She showed that having a baseline GABA deficit was associated with the deep inhibitory intrinsic connections. So in those patients who had a greater deficit, a greater loss of GABA, they had weaker deep inhibitory intrinsic connections. And the effects of tiagabine really depended on the GABA levels of GABA concentration. So in those patients who had higher levels higher levels of their baseline GABA were more, had strengthened connections by TRGABA. So the drug affected those who have higher baseline levels more than those with the weaker connections. And she also showed, again, an interaction of drug and disease. So as I showed earlier, there was a difference between the PSP group and the BV-FTD group. And here, using this model, and again, it's a different data set, but they're essentially the same patients. She showed the interaction of drug and disease was at the stellate cells. So in the inhibitory connections there for the stellate cells. So these are cortical stellate cells, which will be excitatory, but it's the inhibitory connections and that are influenced by the drug in a different way between the PSP and the FTD groups. So it's those with PSP who benefited more from the drug, had strengthened connections at the stellate cells and strengthened inhibitory connections. So hopefully you can start to see how our work is trying to pull these threads together of kind of the what we can measure in terms of the oscillations and the dynamic causal models using our MEG, and bringing together our spectroscopy and the drugs to try and understand, who's going to benefit the most and which drugs are we able to use. And we've got more studies coming as well to try and kind of fill in this picture of the pharmacological effects. So thank you very much for listening. Thanks obviously to all of the patients who really take a lot of time to come in and take part in the study. We're not doing simultaneous EEG, but it is still quite a big ask for the patients to come in and complete a whole day. So the sessions really take a whole day by the time people are in and had the drug and so forth. Obviously, there's a great team. This is not just my work, everybody's work together. And thank you for listening.
Thank you, Laura. Do we have any questions?
Thanks for the fascinating talk. I had one question about the altered beta band signature in the first behavioral task, I think, in the go, no go. I was wondering how you think about the signature. So if I understand it correctly, there is a part of the response that disappears, and we see this nice correlation with reaction times. What kind of process are we tapping into there? Is this signature actually directly related to the production of the motor output, or is this something in between? I would be curious to learn more about your thinking.
Yes, it would be hard to tell. That's a very simple sensor space, all sensors together, to try and probably answer your question. What we are doing is more of the source localization and taking the beta band from different regions, from motor cortex, premotor, inferior frontal cortex as well, to try and understand the drive and the connections and the association between beta loss in one region, and is it just this broad, the whole brain, there's a beta loss everywhere, or is it very specific to specific regions? Is it preserved function perhaps in the motor cortex and more impaired in the inferior frontal prefrontal cortex as well? So I don't think I have an answer for you yet, but the question has been that we are asking that question ourselves as well, to really understand, what is the mechanism in the patients as well as in health? What does beta represent in the patients, and is it similar across the patients and the controls? But I think some of the key questions are starting to be asked, and that's something we can look at with using the DCM models as well, because we can not only look at kind of connections between regions, but look at the cortical microcircuit across cortical layers as well, to understand kind of the excitatory inhibitory balance as well. So sorry, I don't have an answer, but we do have the questions on the cards.
And that's going to be my question as well. Presumably you've got resting state data as well, so you could look at whole brain network in the beta band to see these drug by group interactions. If you have them right now.
We have started doing that. Yes, those methods are underway.
Great. So everybody, our next speaker is Professor Sri Nagarajan from San Francisco.
Also, you're going to be telling us about dementia, I believe.
Yeah.
Yeah. Thank you, Sri.
Okay.
Thank you all. Pleasure to be here. Thank you for organizing this, Chris and Megan. I'm going to talk about neurophysiological network alterations and trajectories in dementia with a focus on Alzheimer's disease, different from Laura's talk about FTD. So just disclosures, I'm a consultant to Megan and serve on a couple of scientific advisory boards and have worked with RICO and have ongoing research collaboration, which I'll talk about as well. So the outline of my talk, given all the prior speakers, I'll zip through the first portion of the introduction to MEG imaging. Just wanted to highlight some of the machine learning that's really enabled some of the imaging work that I'm going to talk about, then talk about resting state network changes in Alzheimer's disease, some biomarkers, promising biomarkers for AD that we could use with the MEG, and then thinking a little bit about neurophysiological trajectory mapping in diseases and using AD as a model that could perhaps be more broadly applied to other diseases. So just wanted to say about MEG imaging, just powerful breakthroughs have occurred in imaging, really pioneered by a lot of work on signal processing and machine learning, like Dennis mentioned. And in particular, I've been honored to have been involved in some of these efforts in the past couple of decades. So I've written a book with my colleague Kensuke Sekihara on some of the work that we've done in unsupervised machine learning algorithms, and we continue to publish. So I strongly encourage those who are interested in the ML or machine learning aspects to look at our recent publications in source localization, that we continue to do so. All of this work has sort of helped us be mostly thinking about source localization as a way to sort of get data from EG or MEG, the dynamic duo model, to basically think about in brain space so that we're not confounded by some of the questions that have been asked. When you're looking at a sensor, it actually has sensitivity to the entire brain. So we can sort of at least deconvolve that signal and be looking at specific brain signals. We can look at brain activity and synchrony. I'm going to focus this talk mostly on resting state and neural synchrony. So just thinking about looking at resting state network alterations, we can come up with multiple measures, and we have done a few of the measures that I list here, but other groups have looked at many other measures. We've already heard about, power fluctuations in alpha beta bands. We can also, I'm going to talk a little bit about this aperiodic slope, which is also gaining some interest in the field as an assay for excitation inhibition imbalance. And our primary hypothesis thinking about these network changes in AD is that physiological network changes are fundamentally reorganized in AD and at the earliest stages of the disease, and they can predict progression. I hope I can convince you about that. And that these imaging of these neural oscillations may be promising early biomarkers for differential disease diagnosis. And Dennis mentioned, like, maybe we shouldn't focus in terms of application of MEG and AI on just diagnosis, but just differential diagnosis might be an important problem. I'm going to show you some markers for that. Also progressing, how prognosis for disease progression, which might be a more important clinical last mile sort of translational piece, and obviously for therapeutic monitoring. So we've shown this in our recent paper a couple of years ago, that spectral changes are altered in AD. And this is, of course, a replication of many other studies in a large body of work, some of which Nakamura and others, and Fernando Maetsu and many others have already also shown this, that basically you have a spectral alteration. Notably, in Alzheimer's disease, you have this increase in delta frequency and a decrease in alpha beta frequencies. And so in our cohort of about 80 patients in each group, we find the same thing. This is just looking at the spectral signature across all brain regions, but there's actually a very interesting spatial pattern to these spectral changes. So if you look at the delta frequency, it's predominantly frontal, whereas the alpha beta changes are more posterior. And I'll tell you a little bit more about this. We also have been doing combining these with biophysical neural modeling. We've been using some of these linearized neural models that allow us to extract biophysical parameters that we can ask are altered. And we find that if you were to model each local spectral activity, a local field potential essentially estimate, as a combination of excitatory and inhibitory populations, you can come up with simple parameters of like excitatory time constant and neural gains. And we find that these are also abnormal in AD, that you have longer excitatory and inhibitory time constants and altered neural gains as well. The most interesting aspect of this neural mass modeling work was that we found in a group of patients where we also have tau PET and A beta PET, tau PET data shown in sort of yellow and A beta PET shown in purple here. And we find that these neural mass model parameters are in fact correlated with tau and A beta accumulation in the same regions obtained in the same set of subjects. We've also very recently found that this aperiodic slope is reduced, which is supposed to be a shift in excitatory inhibitory imbalance in the patient population towards more excitation. And so when you compare, a reduced slope is supposed to be indicating more excitation. And we find this sort of excitatory, hyperexcitable phenotype in Alzheimer's disease. And this is something new that we're seeing, is that this aperiodic slope actually has interesting spatial structure. If you look at brain regions, you find this sort of posterior parieto-temporal distribution of aperiodic slope changes in ED. A couple of years ago, we were very excited to see report that not only is this spectral power, which is an assay, I would say, of local neural synchrony, you can also look at long-range neural synchrony by looking at what is the, this is in our particular measure, we were looking at imaginary coherence, which is immune to sort of these volume conduction effects. And we find the same pattern. If you look at delta frequencies, in our case, we're looking at sort of delta theta in the slide, 2 to 8 hertz. You see the sort of frontoparietal pattern of increased delta long-range synchrony and more posterior temporal reductions in alpha. This pattern is very consistent. I'll show you in a couple of slides, but it's also very similar to what has been seen in the 2 standard of care radionucleotide imaging, PET imaging in Alzheimer's disease, both A beta PET and tau PET. And on first inspection of this sort of pattern of alpha decreases and delta theta increases, we find that it's somewhat qualitatively similar to tau PET patterns that are shown on the top right here. The alpha pattern seems to be somewhat similar, and the delta theta seems to be somewhat similar to this A beta PET patterns. So we wanted to actually take a deep dive at it. So we had about 20 subjects where we had MEG, A beta, tau, and tau PET. And tau PET is new, fairly new. So we were able to actually, in this set of 20 subjects, we were able to look at the spatial pattern and the code localization across individual subjects. And what I'm showing you here is a metric called the Goetz agreement coefficient, which is you have a MEG pattern and a PET pattern in the same subject. You can ask how similar are they, and you find that the sort of alpha patterns consistent with the sort of qualitative impression are in fact much more correlated with tau PET, and the A beta seems to be much more correlated with the delta theta patterns. Not only are they sort of spatial patterns similar, the level of tau accumulation is also associated with the neural synchrony patterns. So alpha beta patterns reductions are correlated strongly with tau, some also with A beta. Delta theta patterns strongly correlated with A beta, but not with tau. So the sort of take-home message is that sort of single MEG patterns looking at frequency specificity seems to be getting us information, what you get with 2 radionucleotide PET imaging. The other, so when that was sort of a proof of principle that this local long-range neural synchrony patterns have some predictive value. One way to test that is to look at differential diagnosis. So within Alzheimer's disease, there are 3 commonly accepted variants. The most common one is the amnesic dysexecutive variant. And there are two other variants. One is called the logopenic primary progressive aphasia variant, and then there's also posterior cortical atrophy. And the atrophy patterns of these are very different. So we looked at what is the long-range synchrony in these three variants. And you can see that the delta theta patterns of increased delta theta power, delta theta long-range synchrony is very similar across the variants, but the alpha reductions are very distinct. And this is actually very consistent with PET imaging data, where the A-beta patterns across these variants are very similar, but the tau PET variants are very different and distinct. And which is sort of what we see. If you look at the alpha patterns, they're very distinct across the variants. The delta theta are very similar. Again, telling you that the long-range neurosynchrony is actually a consistent biomarker. So is there a functional consequence of this? So we took the data and basically distributed it across many mental status exam scores and you find that these alpha reductions are in fact seen in the more cognitively impaired patients, but not so in the delta theta synchrony measures, which is consistent with this idea that while a beta PET is maybe a diagnostic screen for Alzheimer's disease, it's really not a good predictor of disease severity. So A beta is now the gold standard. Now there are blood-based biomarkers that hopefully will sort of replace A beta PET for diagnosis of Alzheimer's disease, but now which is known, I think, in the field that it's less predictive of disease severity, which is what this data is consistent with. So I'm going to tell you another phenotype that we see in Alzheimer's disease that was initially it was motivated from preclinical work where mouse models of Alzheimer's disease have a predisposition for hyperexcitability and epileptic phenotype. And Keith Wassel, who's a junior scientist in our group a few years ago, along with Leonard Mookie, we did a assessment of looking at subclinical epileptiform activity in patients with Alzheimer's disease. And we found that about 40% of patients, as opposed to 10% of sort of incidental subclinical epileptiform activity in controls, have signatures of this epileptic form activity. They don't have epilepsy, even though there is in the literature a sort of a high risk factor for epilepsy if you have Alzheimer's disease. The patterns of atrophy in these groups are very similar, but the cognitive decline, this is the finding that we, in the first paper we published on this, the cognitive decline is accelerated when you have patient with this subclinical epileptiform activity. This seizure semiology, I shouldn't say, some of these, only a very small portion of these patients with subclinical epileptiform activity actually had diagnosis of epilepsy. They just don't have diagnosis of epilepsy. They're not on anti-epileptics. But the pattern of the subclinical activity is actually coming from quite distributed sources. It's not like a single source. It's not like a hippocampal temporal lobe epilepsy pattern or anything like that. The neural synchrony abnormalities sort of mimic this, but there's also a gradation. So if you take our AD cohort and looked at this measure of long-range synchrony and split the patients with those who have the subclinical epileptiform phenotype versus those who don't, we find that the alpha reductions are greater in the patients with the epileptic phenotype. The delta theta are also there, different and more impaired in the epileptic phenotype patients. And these epileptic phenotype can be combined to actually predict patients better than controls in sort of a predictive analysis. Keith followed up the first incidence of epileptic activity, subclinical epileptic activity, with a trial, levetiracetam, which is an anti-epileptic, and found that if you were to look at the data and separate patients who had epileptic activity versus those who did not, he found in the clinical trial, which is mostly looking at behavioral outcomes, that in this case they were looking at Stroop test, and you got improvements only in the patients with epileptiform activity in this Stroop outcome, as well as also spatial navigation improvements were seen only in those patients with subclinical epileptiform activity. So but if you look at and subsequently there's been a study of levetiracetam in AD, and if you look at the primary outcomes, it's not very effective, is what you would conclude. But if you actually look carefully, it will depend on who will benefit from the drug. So this might be an important, I hope, a case study on pointing out the use of biomarkers to actually design the drug. So we actually looked at the MEG data in these patients. So we did, and here I'm just going to show you this aperiodic exponent that changed with levetiracetam treatment. So the patients were on levetiracetam or placebo randomized, and then they did a crossover. And so we can look at the drug effect over placebo, which is what I'm showing you here. And you see that the aperiodic slope changes are seen more strikingly and consistently in the patients with epileptic phenotype, and more diverse effects are seen in the patients without the epileptic phenotype. So this makes 2 points. One is that it sort of reinforces the idea about the clinical trial outcomes as being dependent on the biomarker phenotype that you need. So you can use biomarker phenotype as perhaps an inclusion criteria for who would actually benefit from the drug, which is sort of, I hope that sort of point comes across this, as well as using this biomarker of aperiodic exponent, which is perhaps a measure of excitatory inhibitory imbalance. I'm going to switch gears to the last part of my talk, which is neurophysiological trajectory mapping, which is when we see patients in a clinic the first time. Most of the time, we don't have longitudinal studies, and I know we've seen some very eloquent longitudinal studies ongoing, and that Hanna and others have shown. But when we see a patient in the clinic the first time, we have one time point of the measurement, and that time point is sampling different stages of severity in the disease that they are on. They're on some trajectory of disease. So it'll be nice to be able to map this time point of imaging into what's the stage of the disease. So the framework to do that, this is just a recent paper that's come out, I think this last month in eLife, that I'm going to be describing from Kiwamu, Kudo, and Kamalini Ranasinghe. So we looked at patients and controls who have a variety of hippocampal volume loss, so which you can quantify, and you can see here that the hippocampal volume in the AD patients is reduced compared to controls, and they also have reduced MMSE scores compared to controls. So we can sort of Z-score these distributions and then stage these patients as mild, moderate, or severe. So that's sort of just a simple quantification of this. There's a framework in statistics called event-based modeling that you can say, what is the likelihood of the sequence of these things? So we can, there's some math I'm not going to go over. You can just basically, it's a maximum likelihood estimation method, but I'm just going to show you how this would work in this cohort of patients with this distribution of, say, hippocampal volume loss and MMSE scores. Well, you can actually compute what is the probability of a patient belonging to each stage. And in this particular case, you'll have 7 stages, and you can see that the controls who are shown in CDR 0, which is clinical dementia rating 0, are in the early stages, and the patients with 0.5, 1, or 2 are in the later stages. Notably, patients in red have CDR 1 or 2 are in the later stages of the disease. So we have a mapping of, it seems like here you're just mapping disease staging from controls through MCI to AD, but you can also plot the time course of hippocampal volume loss and MMSE scores as a function of stage. Now, the question we were posing is, where does electrophysiological markers, where do they fall on this trajectory? And so we've done that in a couple of ways. First thing is to take the data here, because each patient is now mapped to a stage. We can just plot the electrophysiological mapping for each patient belonging to each stage. And so now we can get a time course. And you see the same thing across progression, across the disease stage progression stage is shown here. If you look at across frequencies, delta theta, you can see a progressive increase in delta theta activity patterns. And you see a progressive decrease in alpha and beta patterns. And it sort of seems to be mapping on to what we saw as in terms of a cohort level contrast. We also wanted to add the electrophysiology into the EBM or the event-based modeling and look at the relative time course of these trajectories. And that's what we did here, looking at long-range synchrony. We have a model that includes long-range synchrony, atrophy, and cognition. So we can plot the relative time courses of each. And what we find is that the long-range synchrony measures here, we're looking at amplitude envelope coherence corrected for instantaneous coupling. And you can see that the AEC time course is abnormal at the earliest stages of the disease, even earlier than any hippocampal volume loss or MMSE cognitive decline. So, and you can see that the pattern here, I'm showing you just the alpha staging of this alpha amplitude envelope coherence, and you can see this progressive decrease in the alpha amplitude envelope coherence. So to conclude this, so far what I've said, I hope I've convinced you that resting state neurophysiological networks are indeed altered in AD, and MEG imaging potentially can provide information consistent with both A-beta and tau PET imaging. And I hope I've given you some evidence for hyperexcitability phenotype in AD, as well as subclinical epileptiform activity that we see in about 40% of AD patients. And this trajectory mapping approach allows us to map onto disease stages, where we can see long-range synchrony as perhaps representing the earliest manifestation of AD progression. But I just want to say, and another thing, kind of borrowing Steve Jobs' tag line here, this one, because it's a Pharmaco MEG Symposium, I wanted to tell you a little bit about some exciting work we've done in brain tumors. Just one thing that might also give you some idea about network alterations in that might be valuable for biomarker targeting. So in okay, I have to come back to my slide on that. So we just had a paper where we looked at brain tumor patients with glioblastoma, and we found remodeling of neural circuits that predicted survival. But what governed this was actually MEG. At the heart of this was we were looking at lesion-specific connectivity. So the same measures I showed earlier, but here we're now looking at lesion or the brain tumor specific connectivity that were provided to the surgeon preoperatively. So this data was on an individual single subject level, was provided to the surgeon who did biopsies of the high functional connectivity and the low functional connectivity range regions. And this is coming from a hypothesis that there is a proliferative effect of glial neural coupling. And in fact, that's what we found. And this is looking at deep phenotyping of the biopsy specimens in high functional connectivity versus low functional connectivity brain regions, and show that tumor-infiltrated circuits have synaptic remodeling that are consistent with high functional connectivity. And these high functional connectivity regions, in fact, are proliferative. So we found that you have increased neural glial coupling when you look at these co-staining immunocytochemistry. And in particular, there was one thing that came up is that the high functional connectivity regions had higher expression of TSP. So there was actually a gabapentin trial. You can also show that if you have high functional connectivity, it decreases survival time in tumor progression. And also that the serum TSP level was very consistent with this high functional connectivity markers. And gabapentin was introduced to the biopsy specimens in an organoid model and shown that high functional connectivity biopsy specimens with gabapentin had, it seems to show a reduction in sort of this synapse formation between glial and neurons. So all coming together is, in addition to dementia work, I hope I've convinced you that sort of looking at network alterations in dementia or brain tumors can be powerful biomarkers that hopefully could be inspiring for pharma companies to think about clinical trials in this arena. So with that, I'll stop there. I forgot about the slide. The slide got mixed up when I added this thing, where, of course, this is thanks to an incredible team. I have to particularly thank Kamalini and Kivamu, as well as Keith Bossel and several others in our team who have enabled this work. Thank you.
Thank you, Sri, for a great talk. I'm going to use chair's privilege and get my question in first. So, so two related questions. So, first of all, what's the rationale between for pooling delta and theta into one marker? That's the first point. And the second is, by that signal, do you think that that's a true oscillatory signal, or is it more related to the steepness of the aperiodic slope at that low end?
Yeah, great question. So the delta theta, we have found that we mostly did it for increasing power and reliability. We didn't find very big differences between the two. So that's why we started pulling them to get a little bit more power. I don't think, so the second part of your question is, could that just come from the aperiodic slope? Actually, I have a whole talk on this. I don't think that the spectra can be fit with a single aperiodic slope. So in our aperiodic slope calculation, we actually calculated from 15 to the gamma range, 15 to 50 hertz. Right. Because I think that the spectra that we typically observe, like I can show, I think I showed this. Sorry, I'm zipping through to this slide on the spectra. Here, you can't fit this to a single slope model. What we found is that you can fit this to, I can even use a cartoon, this one here. So you can fit it to, actually, I don't know how to do that in PowerPoint. But it's typically, you can, and other people have shown that the aperiodic slope, you have one slope here, and you have a second slope in the low frequency range. So you can't fit it to a single slope. I actually think that that's why there was a reviewer who asked us, why did you not subtract the aperiodic slope from your calculation? And I think that's from the Donahue and the Brad Wojtek work. They were only using a single exponent model. It's actually not a good fit for the data. So you're actually going to creep in offsets into your so-called periodic component. And so that's why we didn't do that as one. I think that the delta theta, you can see here, it's a low frequency. It's really like a band pass signal. So you can't really model it as if it were sort of this 1/f slope on a periodic spectra. I think, anyway, that's my take on this. I think the offset is also very difficult to interpret. So I think like some people will say, well, it could be the offset. The problem with offset is that it's very sensitive to DC, and we don't do true DC. So it's very sensitive to filter setting and where you are in terms of the measurement. So I think the exponent is interpretable, and the offset, I think, is questionable interpretation potentially. Yeah.
Any other questions?
Yeah, I actually had a very similar question, but I would like to turn it around. So rather than asking if it is the slope, I was wondering, how do you distinguish the slope from the power? Because another way of looking at this is that the beta peak is gone in a sense, and the alpha peak goes down, and there's the slowing. So obviously, if you think about it in terms of a slope, it will change your slope, but you could also just say that there is really a change in power. How do you distinguish these two things?
So the first way is that we actually measure our slope outside. We only measure it from, like, say 15 to 30. So we're outside sort of the classic alpha beta frequencies. So we're looking at really the tail of the slope. So that's kind of the one way to address that issue.
And okay, sorry if I can just mini addition. Have you looked into making an additive model or something like that to really tease that apart, to see what is added information that comes from the slope that is not explained well by local spectral power?
Yeah, so it's a slightly different way. This slide is cropped up. So we are doing it in a slightly different way. It's a paper in the works where we're actually asking whether the slope can arise from these neural mass models. It's sort of trying to answer this question: is the slope an epiphenomena of, and that's kind of what we're finding, is that the slope is kind of an epiphenomena of these neural mass models. You have spectral power, and the bandwidth of the spectral power can kind of account for this. So if you actually look at models for aperiodic slope, the GABA et al. Is the one model. It's not really a biophysical model. It's kind of just like, it's not a true biophysical model in that in a traditional sense. So initially, I was actually quite skeptical about the aperiodic slope sort of finding or the body of work, because it was sort of put out there as a marker of excitation inhibition, but it was coming from this one model that wasn't a true model. But I think I've been more convinced with the recent studies with propofol and ketamine that actually show that you can modulate this, and our own data that shows that you're actually finding this. But we also find that this is correlated a lot with neural mass model parameters. That's a paper that we're working on. So I think there's more. That's kind of a different way to look at it. Is Laurie?
Yeah, thanks, Ray, for the great talk. You advocated studying long-range synchrony or functional connectivity, and well, as we all know, we have a bunch of metrics to do that. Do you think it matters much to pick the proper one of those, or should we be able to see the same thing with pretty much all of them? And then a related question: now that the oscillatory power changes, well, in patients, in aging, and whatnot. So, do you think that's a worthwhile confound to take into account in connectivity estimation?
Yeah, I'll take the second one first, because it's actually very part of the parcel of the work we're currently working on. In all the data, especially in the trajectory mapping that we did, we looked at age residues. So we sort of regressed out age effects. But in a recent paper, we just got rejected from Journal of Neuroscience, but we're working on, we're looking at age effects. So there is clear age effects in all these measures in healthy control population that you definitely need to account for. And so it's a very integral part of this effort. So the first part of your question, I think there's a short and long answer. I'll give you the short answer. The short answer is I don't think it matters that much. And actually, there's both empirical data that there are subtle differences, but there's largely similarities. But there's also very strong theoretical data. It actually came from, I'm forgetting her name, just blanking, a student of Richard Leahy, who actually showed that many of these linear metrics are really if you think about modeling this as an underlying stochastic process, they're just different instantiations. And given Gaussian and underlying process, it doesn't matter which metric, you're going to ultimately measure the same underlying statistics. And then there's also work from Guido, who showed that some of these things are really related to, say, bispectral estimation measures as well. That's sort of the long version of that, is that I think it doesn't matter that much. There are some differences in reliability that people would argue. I would also argue that people say, oh, imaginary coherence is not that reliable. We actually find pretty good ICC, and I think that it's just measuring something different from amplitude envelope coherence. And so when Matt Brooks came over and we published this paper on AEC versus IC, I think they're not measuring the same thing. They're just measuring two different timescales. One is looking at an envelope timescale, and the other is faster timescale. So I think these are the connectivity measures allow us to give multivariate measures of the potentially multivariate biomarkers. But there's a challenge with how do we convince pharma industry and the regulatory industry of embracing multivariate biomarkers. But that's where we are with MEG. I think we're ultimately making multivariate biomarkers. How do we sort of bridge that gap? Sorry.
Yeah. Thanks, Ray.
So I'm going to treat myself to one extra question.
Yeah, great.
So I noticed in the trajectory mapping, which I thought was really striking, that you switched to the AAC measure for that.
Oh, I just showed you that. In the paper, we have the IC as well.
Did I see that right at the early stage thing that actually it switched, the alpha had switched out hyper?
So in this case, we were looking at age regression. So I think what you're pointing out.
That one then.
No, this is atrophy. But I think here you're saying, yeah, at the earliest stage, this is one of those. So this is not this actually in the paper. There's a statistical contrast, which is the stage 5 versus 1. So these are just for this stage. What is the representation? It's the unthresholded representation, and then the statistics are on the right.
Yeah.
So it's hard to, what do you compare the early stage to? The only thing you have to do is what Laurie is asking. You want to compare across age, which is sort of, there's actually an age effect for alpha, which is going up. And then delta theta goes down with age and alpha goes up. So there's sort of very interesting effects that we're finding with age.
I got a little bit excited because we published this paper looking at young people with and without ApoE4, and we showed that in the alpha and beta bands, there was hyperconnectivity in the ApoE4 carriers.
Yeah, we have some preliminary data. We don't have high samples with the ApoE versus non-ApoE contra. We have some preliminary data on that that also mimic effects that you've published. Yeah.
Great.
Great.
Rick. Go on then, Rick.
I'm not a dementia expert at all, so I thought the first sign of it was usually in the hippocampus. So what is this widespread brain-wide alpha disconnectivity all about?
Okay. So there's again a short and a long answer. I think it's related to spread. So our hypothesis currently, which is grant we just got funded, is that I think the alpha reductions is really, I think the underlying process is hyperexcitability. There's a hyperexcitable network on which I think tau spread occurs. Because when we actually, in the small set of patients where we also have A beta, what we find is that A beta actually spikes much earlier than all of these. So A beta, but that's not surprising because A beta is the diagnostic indicator. So all the patients with MCI or AD, they just have to have high A beta. So A beta is almost an irrelevant, it's just a diagnostic marker. But to look at progression, I think that the tau spread has to occur. And this is a hypothesis. It could be wrong, but it's falsifiable. Well, that's good. But I think that our hypothesis, working hypothesis, that tau spread occurs on the hyperexcitable network. So one of the competing hypotheses that people have is that function is an epiphenomena of tau spread. But we think that perhaps the more provocative hypothesis is that function is actually the skeleton on which pathological spread occurs. So hope that's gives you a
super interesting. Thanks.
Thank you.
So MEG's the early marker. By the time you see it structurally in the hippocampus, it's too, it's all over. Yeah. Okay, so now coffee break. Let's thank
you. Thank you.
I've been reliably informed that there's going to be football players in this talk.
Yes.
So look forward to it. Thank you for making the journey.
Thank you. Thank you guys for having me. I'm excited about this. So yeah, we're gonna watch a little bit of football. Maybe not as entertaining as the Super Bowl, but we'll get there. So I'm going to talk a little bit about pediatric football players, but also Alzheimer's patients. So this is a really good timing right after Shree's talk. So first of all, just a few quick definitions. TBI or traumatic brain injury often gets categorized into 3 different categories. You have severe, where essentially you're in a coma, moderate, where you have a Glasgow coma less than 12 or loss of consciousness more than 30 minutes. And in terms of imaging, you're going to use a CT to rule out any sort of bleeds like a contusion or a hematoma. And then you're going to find my mouse really quick. And then you're going to do an MRI with susceptibility weighted imaging to look at diffuse axonal injury. This talk, we're actually going to focus on mild TBI or concussion because part of the definition of concussion is that the structural clinical imaging is normal. And so the diagnosis is purely right now based on symptoms reported in neuropsych testing, which we heard a little bit of earlier this morning about the DSM-5 and how a lot of the psychiatric illnesses are based purely on symptoms. So concussion is you can either strike an object, you can have acceleration deceleration like whiplash, or a blast injury like you would experience in the military from a grenade or something like this. Loss of consciousness for concussion is not required. That's often a misconception. And again, that diagnosis was based on the symptoms like headache, confusion, kind of visual disturbances, things like that. So fairly generic. And then when we're working with our physicians, there's really no quantitative metric to tell when these people are better or how to recover. So they're essentially told, go back to your normal routine. If it hurts, stop, take longer to rest. And so we don't have a great biomarker for knowing kind of the severity of concussion or even when to say how to recover. And if you continue with these symptoms for longer than about 3 weeks, you get
kind of bend into post-concussion syndrome. And often you have an exacerbating symptom, like you have a migraine that's really bad, or you have a really bad sleep disturbance, and it can fall into a lot of different categories. And then the physicians are essentially treating the symptoms. So if you have a headache, you may go on migraine medication. If you are having depression, you may go on an SSRI. If you're having sleep disturbances, they'll work on you with sleep hygiene. But essentially, they're just treating the symptoms here, not really the underlying cause, because it's not very well understood. Now, what about longer than this? What about kind of lifetime experiences of post-concussive syndrome, or people that experience multiple concussions in their lifetime? So a good example of this, or a good population for this, is American football. They often get multiple concussions in their lifetime. They're consistently hitting their head repeatedly throughout their lives. And there's a very interesting pathology that comes out called chronic traumatic encephalopathy, which was published by Bennett Omalu in 2005. It became a blockbuster movie in 2015 with Will Smith, which I think is kind of every scientist's little bit of a dream, right? To be played by this actor in a movie. And then it's, of course, the subject of a couple of books. One of them is a good one on League of Denial, about how the NFL really pushed back against this concept that hitting your head would cause brain injury, which is amusing, but still, it was a big deal in the news for a long time, and still is. And then in 2017, Dr. Ann McKee's group at Boston University published this finding where 110 brains out of the 111 brains that were donated to their brain bank by NFL players, so American pro football players, had CTE. They found it on autopsy. Now, again, this is a little bit of a limited sample, because if you're donating your brain to a brain bank, you probably had symptoms that led you to do that, but it's still a striking finding. Now, what is CTE? CTE is actually very, very similar to Alzheimer's. You have neurofibrillary tangles, tau proteins, beta amyloid proteins, cortical atrophy. So part of the Dr. Omalu's story about that first diagnosis of CTE in the NFL player's brain was that he took it around to a lot of his pathology friends, and they said, yeah, this is Alzheimer's, but it's a very weird spatial pattern. So, Alzheimer's disease, the tau pathology occurs in this kind of Brock staging, and it starts deep and spreads out, whereas CTE, it actually starts in the depths and the sulci of the frontal lobe, which is very interesting. So it's this different localization, but very similar pathology, which we'll talk about in a little bit, and why MEG excels in this genre. And so we've gone from having an unremarkable CT, your kid gets a concussion, playing soccer, playing American football, whatever, to this very scary neurodegenerative disorder where you're having tau accumulation, much like that of Alzheimer's. So how did we get from one to the other, and how do we measure what's happening there? Because right now we're kind of in the dark. We don't have a good biomarker for this. And I will say as a disclaimer that concussion and CTE are related, but this relationship isn't very well understood. Having a concussion does not mean you're going to develop CTE, which is good news for most of us. So what we do know about concussion is that there's a neurometabolic change in the brain. And this has been well documented by Dr. Giza. I think they're on their second version of this now. The biomechanical forces initiate this ionic flux. And this, essentially the brain is a sponge, and it squishes, it releases this indiscriminate release of neurotransmitters. And then you have neuronal suppression, impaired metabolism, and neurotransmission for about 3 to 10 days. So in those first 72 hours, you have a lot of repair going on in those things. And so those neurometabolic changes often correlate with a specific rhythm of an active neural population. And one thing I forgot to say here is that these neurometabolic changes, we know this because of invasive animal models. We're not doing this in humans for obvious reasons. And so the spontaneous or the resting state is typically in the alpha range, which we've heard this morning. And then the EEG of these severe TBI, we know from people who are in a coma or in the hospital, they get an EEG. Their EEG frequencies shift to lower and slower frequencies. So in that delta theta, a lower shift in the alpha peak. There's some suggestion that this might be healing and that there's a metabolic cleaning mechanism associated with these slow waves. But again, this mechanism isn't really well understood. Now, so functional changes obviously MEG is we're at the MEG conference. So MEG is very sensitive to chronic MTBI. So it's significantly more sensitive to the abnormalities in patients with post-concussion syndromes than MRI or EEG. It also outperforms SPECT. The slow waves in the temporal lobes from the first paper in the late 90s correlated with memory problems. One of the most important things I want to say here is that MEG can localize. And that's important because we know the localization of CTE pathology differs from that of Alzheimer's. So if we're able to pick this out, it's very helpful. And then kind of later, a couple of years after the first publication, Dr. Huang and Dr. Li at UCSD demonstrated clinical viability. So they have this automated fast Vestal method that compares the delta power of their adults with chronic post-concussion syndrome to a normative database, highlights the areas with abnormal increase in delta power. It's 85% or more sensitive to mild TBI and 100% sensitive to moderate TBI. And again, this is in adults with chronic symptoms. What's really interesting is that these areas correlate with their post-concussion symptoms. So here in the frontal lobe, this person had trouble concentrating, which makes sense. Now, we've talked about this kind of chronic post-concussion syndrome. Now we want to study the concussion itself. We want to look at that 3 to 10 day range when they first get the concussion. This is hard to do because how do you just blanketly tell everybody like, hey, if you get a concussion, we want to see you within 72 hours. You have to get them you have to know them before they get their concussion, so it's easy to bring them in. And so how do we do this? We study an at-risk population, which is American football in our case. And we are actually doing this in kids 8 to 18. So this is the imaging, telemetry, and kinematic modeling study that's based at Wake Forest University, where I did my dissertation. And so, like I said, we're recruiting this population at the beginning of the season before they engage in this risky behavior. So they have cognitive testing, MEG and MRI before the season. We measure their head impact exposure throughout the season, and then we measure them again postseason. And we're also doing this with a non-contact sport control group. And I spent my 5 years of my PhD collecting this data in the mud. I was there for all of this, so it's a passion of mine at this point. So the HIT system hardware, it actually fits in a natural gap of the helmet padding, and we're able to have accelerometer measurements of the linear and rotational acceleration that these players are experiencing. And this was done with our biomechanic colleagues at Wake Forest. This data is collected wirelessly for every game in practice. And so we're able to capture potential concussions. And so I'm going to show a video of one of our concussions, and we're going to watch this player here that's standing up. And there won't be audio because it's kind of loud, but they're going to go for a play, and this is middle school football. So I think he's 13, something like that. And so he gets tackled and hits the ground. And then as he gets up, he's a little dazed and confused. He's kind of stumbling around. His coach at this point notices and calls him off of the field, and he's like, no, I'm going to the huddle. He doesn't really know where he is. So we can watch it one more time, just really quick. So here he gets tackled and falls to the floor. So that's actually not a helmet to helmet collision, but a helmet to ground collision that he experienced. And it was around 72 Gs, which is a moderate impact for football. Now, I'm on the sideline, and our concussion protocol is concussion suspected. We have an athletic trainer that's paid as part of the study. They assess them on the sidelines of the field. In his case, his parents actually elected to take him to the ER. And then the study team was alerted. We had an appointment with our sports medicine physician to confirm the clinical diagnosis of the concussion. If it's confirmed, we schedule the post-imaging appointment, and then we have the post-concussion imaging. And all of this we want to happen within 72 hours because of that neurometabolic cascade that I talked about. So again, this is why it's so important that we engage them before they get their concussion. And so with all of that, we were able to capture 8 concussed football players that had good baselines, plus we had them within that 72-hour time frame. And then we had 8 age-matched football players and 8 controls. So we had 8 minutes of eyes open resting state data. We processed this with Brainstorm. We took 3 minutes of clean data, filtered it, removed artifacts, co-registered it with their specific T1 MRI, and then source localized all in these different frequency bands. Now, what's really interesting is that if I look at this individual subject, now this isn't the same subject that was in the video, but it was a very similar head impact exposure concussion to what that player experienced. So pre-season, we see some delta power. Post-concussion, we see delta power that actually looks a lot like the coup contra coup pattern that you would expect. So they had a very similar tackle. They hit the back of their head and kind of bounced off. And then post-season, you also see some residual delta waves. So that's an interesting pattern that we're seeing there. And this is even the post-season scan, they're no longer symptomatic. They've returned to play. They're totally fine, kind of exteriorly. And this player, I will also say, has had concussions in the past. This is a high school player, and they had previous concussions. And then we can also look, and I want you to note the spatial differences here. So this player is more occipital, kind of parietal area. This player, 15-year-old middle linebacker, was an open field tackling drill with a helmet-to-helmet collision that was a 92 G head impact. And so you see more frontal delta power, increased delta power, and especially in the depths of the sulci. Now, I will say the biomechanics data that we have, you see the highest number and the highest severity of head impacts in the frontal lobe, or in the frontal areas of the helmet, which I think says a lot to that CTE pathology pattern. So just if we look at this blanketly, just kind of whole brain delta power, just very crude, you see that compared to the controls and the football players, all of our players with concussion had an increased delta. It did tend to correlate with where their head impact exposure was for that concussion too. So it's a very heterogeneous disease, which makes it a little bit more difficult. And as Sri talked about, there's more and more research on this aperiodic slope. And so if we look at this was work done by Kevin Yu, who's a graduate student there, and then Alex Weissman has been helping advise him. And so you see this increase in the frontal delta. If you correct it for that aperiodic slope, it kind of goes away, and then they pull back in the head impact exposure, and you see, and it reappears once you've corrected for the aperiodic slope. But we're still working on understanding this a little bit more. But I did want to point out that work is coming out soon. And then this is the UCSD group again, and they've done children with post-concussive syndrome. So 3 weeks post-injury, they're still having symptoms. And then they also have some orthopedic controls in there. And with their machine learning classification, they're able to tell with 95% accuracy which kids had a concussion. And then the resting state MEG data actually predicts the symptom recovery for 3 months after. So this is very important, especially for a kid who's really looking forward to getting back into play, or even for an adult who's had a concussion as part of a car crash. Knowing that recovery trajectory is very important. And I think something that you would want to use clinically. So I don't have data to show for this yet, but we have 4 patients that have gone through our long study where we're taking them. We've keyed in with the athletic trainers, and we get MRI and MEG within 72 hours of a concussion of high school sports athletes. And then we do 1 month post-concussion and 3 months post-concussion, mainly because that player that I showed earlier that had the residual delta power in that post-season scan, that was not quite 3 months out. And so we want to see longitudinally what happens with this, and is there something that can be done to better inform that recovery. And then lastly, I'm going to touch on repetitive head impacts. So these are head impacts that don't cause a concussion. High school football players receive 400 to 2,000 head impacts per season. There's limited literature on imaging biomarkers to inform how much is too much. DTI shows changes with FA, fMRI shows changes with altered connectivity. But what's really interesting is that there's a recent paper showing that repetitive head impact exposure, kind of a retrospective look at those players that donated their brains to the brain bank. If we look back at when did they start football, what positions did they play, how kind of like what was their hours of limitation of practice and things like that. And we calculate out what we would expect their head impact exposure to be throughout their lifetime. That is more predictive of CTE than concussion itself. or the severity of the concussion. So, and also it correlates with that NFT burden in these male football players that have donated their brains to brain banks. So this is a very informative metric that we need to pay attention to. And so we've prospectively looked at this with our database of 72 football players and controls. And we said, okay, head impact exposure compared to number of abnormal voxels, again, because it's a very heterogeneous disease. So we just said, okay, just in general across the whole brain. And what we see is that the more head impact exposure you have, so here on the x-axis, the more increase in slow or delta theta power you're going to have. And in this particular population, this is the 8 to 13-year-olds. And other studies have shown increased delta after concussion, but those studies were in adults. These are in younger kids. We know that kids, that their delta power is decreasing as they age and mature, and with a very steep trajectory. So this is our age range here, 12 to 18, delta is going down. Theta is also going down as they mature, but at the, I'll point out the y-axis there is very different. And so it's informative across this broad span of ages, from children all the way to adults, but making sure that we understand the neurobiology of the kids that we're studying is important. And lastly, I'll point out that the military personnel is exposed to subconcussive blasts from their own weapons and from breacher training. So this is when you see them kind of line up to go and, knock down a door with either a grenade or some C4 or whatever. They see actually increased amyloid on PET. And as we heard from Sri, that this amyloid compares well to the some MEG data. So here on the right, you see these are instructors. So these are breacher instructors that are exposed to a lot of subconcussive blasts throughout their lifetime compared to controls. And you see this increase in amyloid on those that are doing instructing in the breacher training. Now, I'm going to skip over kind of the psychiatry behind this, but I encourage you, there's the episode by The Daily that talks about the findings in this ex-military personnel donated their brain, and looking at CTE in that brain and kind of the psychiatry impact of this disease. So I encourage you to listen to that because they did a much better job explaining it than I can. But now, MEG for PTSD in the military is also another important topic, and I really encourage you to look at Ming Huang's work as well as Ben Dunkley, looking at PTSD and how MEG can be a biomarker for PTSD, as well as potentially informing treatment for those with PTSD. So in summary, concussions as well as repetitive head impact exposure in adolescent athletes are linked with neurodegenerative disorders, CTE, but this is very poorly understood. MEG can see concussions where other imaging modalities cannot, and it's very obvious. So you can just, that image that I showed earlier with the 3 preseason, post-concussion, postseason, that could be pushed to PACS, that could be pushed out to a radiologist to read. Not covered today, there have been multiple studies with DTI changes, but it's more in groups. It's harder to see on an individual level, again, where MEG excels at that individual level. And then increases in repetitive head impact exposure correlate with increases in MEG measured theta in our youth group. This can be used to help inform rules, practice structure, technique. I feel like there's also an application there for military breacher training. And then CTE and Alzheimer's share a pathology as well as MEG features. And I think this is a very interesting thing to ponder as we talk about pharmacological interventions for both diseases. And with that, I'll acknowledge my collaborators at Wake Forest, as well as my new lab at UT Southwestern.
Thank you, Liz.
Thank you.
Brilliant talk. Thank you. Do we have any questions for Liz? I think Craig was first, right at the back, right?
Yeah. Does the CTE show up ahead of autopsy with MEG?
So does CTE show up ahead of autopsy with MEG? We don't know. We would very much like to know. I know that there have been some studies of PET tracers looking at CTE, and it can show up on the PET tracers. So I think there's a very high probability that it will show up with MEG, and we're hoping to do that with some retired athlete studies.
Yeah, thanks, Liz. Really great talk. You showed the delta band changes, now that you had the very nice reference baseline recording from these people, which of course normally we don't have. And I think you have some correlation between the direction of the impact and then the sites where you have the elevated delta band.
Yeah.
We have also seen, in a sort of a more generic manner, also changes in other frequency bands, which probably aren't specific about the direction of the impact. And well, we don't know that, so then because we have no information about the impact. But did you see something like this also in your data?
There's a trend, I believe, in the alpha and the gamma bands, but we only had 8 players. And so with our new study, the long study, we're hoping to get about 20 players with concussion and increase our power to look at that. But I do think that there is something more in the gamma bands, and that's definitely been shown by the UCSD group, that the gamma is informative for predicting post-concussion symptoms and recovery.
Yeah, that's compatible with what we have seen. And also, if you train a machine learning model to pick up the MTBI patients, then the delta band doesn't show up because it's so different from each individual, but then rather it goes for alpha and gamma activity. Yeah,
I agree. Yes.
Yeah, okay, thanks.
So I have a question.
Oh, okay.
So obviously, if you're talking about the delta as a biomarker, that's obviously a signal from the grey matter that we're picking up with MEG. I wondered about the relationship with potential white matter injuries, maybe diffuse axonal type stuff in concussion and or CFD.
So we've actually, my graduate student, Natalie Bell, just finished that analysis, and we did a voxel to voxel comparison to see if maybe there was changes in white matter close to where we were seeing the delta. And we actually didn't see a very strong relationship there. And so what we're looking at doing now is a more advanced analysis to look at trictography and seeing if there's a more distant relationship where there's where there's kind of this white matter change in the tract that then terminates at an area where there's delta power. And so we're working on figuring that out statistically right now.
Is it your feeling that these sort of electrophysiological measures will end up being more sensitive than
I think they are more sensitive. Yeah, so the paper that Natalie's coming out with soon, that she just finished that analysis, we showed that the MEG was more sensitive than the DTI or the DKI even. So yeah,
great.
Thanks.
That's great. Oh, James, I'll show you hand up.
Just on studying the biology and the imaging and the clinical syndromes, just think about linking the biology and the syndromes and the clinical data. The change in that delta, does it? I may have missed it. Does it correlate with the biochemical measures of brain injury, like the NFL, neurofilament light, that could surge in the days following concussion? That gives another index of collective damage.
I love that you asked that question. We don't have that data on the study that I showed today, but the study that we are just started recruiting for in January, we have blood biomarker data on, and so hopefully I'll have that next year.
Are those inflammatory markers?
So you can be inflammation or brain injury. So neurofilament light, it's released by damaged and dying neurons, and it surges in the days after concussion and then eventually dies down.
But I will say the players that we've had 8 people come through so far, 4 controls, 4 concussed athletes. MEG, so easy to do. Blood draw, they're terrified. So
that's great. Let's give Liz a hand.
So for our next speaker, we've actually got a local speaker, Professor Pergola, who's going to be, interesting, going to be talking about schizophrenia. Thank you.
Well, thank you for an introduction. It's nice to know that I'm local already. So I'm actually working in 2 institutions in Bari, Italy, where I'm associate professor, and also at the Lieber Institute for Brain Development in Baltimore, Maryland. She's affiliated to the Johns Hopkins campus, and I'm professor at the Department of Psychiatry and Behavioral Sciences. So I will go back to some of the things that Professor Singh and Professor Harrison said before, and this will be from a slightly different angle. I'm enjoying very much this symposium because I'm learning a lot about the technicalities of MEG that I have not dealt with in the depth that you're presenting today. So I'm very excited about that, and I will take a slightly different angle and tell you a little bit about the genetic risk for schizophrenia and why I think it matters to the search for biomarkers, and the personalized treatment of psychiatric disorders. So this slide, the first few slides, please bear with me. There will just be primers about some of the concepts that we will discuss, and then I will only have a few slides. This talk will be very short and simple, and there is one idea that leads this talk, and that is heterogeneity in schizophrenia might not be just about the heterogeneity of symptoms and of brain markers, but it may be about the heterogeneity of causes. So in this slide, you see there's some risk factors, and there are many. They happen at different stages of life, prenatal life, childhood, before the onset, and then at the onset, you have different risk factors, and they can be genetic risk factors or environmental risk factors, for example, stress, substance use, but also perinatal complications and so on. And there's genetic factors that we will deal with in a little, and there's some neurobiology. Now, the point that I'm trying to convey is all this list of factors doesn't really need to apply to every single subject who has a diagnosis of schizophrenia, but there may be several of them applying, or maybe one of them, or maybe none of them in some rare forms of the disorder. And these lines that you see here, these are hypothetical because every case is different in terms of how symptom severity, how the disease course goes over time. So this slide serves to try to introduce a concept that not everybody is familiar with, but psychiatric disorders, and some more than others, run in families. They run in families not that much because the familiar environment is corrupted somehow, but very much because there is a genetic predisposition to vulnerability to some psychiatric disorders. And you can see this here. These are identical twins. One of them has schizophrenia. How many times does the other twin, does the co-twin have schizophrenia? It's about half of the times. And this is much more than fraternal twins. They are born in the same moment as identical twins, but they only share 50% on average of their genome. And therefore, the difference that you see here is used for the computation of the variance within the population that is explained by genetics, which is a measure commonly called heritability. Now, the heritability of schizophrenia, you have to bear with me, telling about heritability of a trait is not completely correct, but we may, if you're curious about this, we can talk more, but it is generally, you can see this in this meta-analysis across many different studies. This paper from 20 years ago established that in the general population of many different studies involving thousands of people, the heritability of schizophrenia was at 80%. And this little cartoon that you see here has a purpose, and that is to reconnect a bit to what Professor Harrison said about the dimensions in psychiatry. So there is this idea that patients can have different dimensions of symptoms, and they can vary from one patient to the next, even though they have the same diagnosis. And there can also be environmental impacts and different genetic predispositions to these dimensions. When we are saying genetics, we are saying here something very different to what has been said before about, for example, the Dravet syndrome or particular epilepsy types, because they are rare disorders, so they have rare genetics behind them. What we are dealing with when we talk about psychiatric disorders, in the majority of cases, actually the overwhelming majority of cases, because 99% of the causes of the cases with schizophrenia have no rare genetic variant. So only 1% of schizophrenia cases is probably connected to a rare genetic variant. So these are not. So this you see here, this on the x-axis, this is called Manhattan plot because it resembles Manhattan skyline. And it's simply the DNA from chromosome 1 to chromosome 22. And this is the X chromosome. And this is so it's just laid over the x-axis. And this is a measure of statistical strength. So it's a p-value. The higher this is, the more significant a certain variant is. And so this that you see here, they are variants. Why are they called variants and not mutations? Because mutation is something that happens, usually you call them de novo mutations. They happen there is a change from parents to children. And in the case of variants, they are called so because every one of us has a certain setting at a particular place in the genome. And so I may have a certain nucleotide and somebody else may have another one. And what we can do is, well, what does this plot stand for? You see this line here? This is the line of significance after correction for multiple comparisons. So this is about 300 variants that have been discovered to be associated with schizophrenia. And this is probably just the tip of the iceberg because these 300 variants probably impact on thousands of genes. So we don't really know which genes we should target as treatments for schizophrenia. But what we have done so far over the past 10 years has been just summing up all these variants under the assumption that what we're seeing is additive genetics, so-called, and we can compute a score, and this is called polygenic risk score. So the people in this right extreme of the score have a very many variants corresponding to the risk allele for schizophrenia. And the people here on the left, they have protective variants. So they have a very different genetic background, and these people can be diagnosed. Well, we have observed in independent samples that they are diagnosed much more often with schizophrenia than the people here in the first decile. So these people actually, if you want the numbers, they are in the last deciles 8 to 20 times more likely to get schizophrenia than people in the first decile. So this seems to be useful, but it doesn't actually have that coverage in terms of variants explained that is really needed to make it a clinical tool. You have to think that all this work, which involves, so this one involved, I think, about 40,000 cases and about 200,000 controls. So with all these numbers, we still only explain 7% of the variants in schizophrenia diagnosis. And if we expand a little more to what I showed you before, so the psychosis dimension, this explains 9% of the variants in cases, so not much. What does this have to do with MEG? So this presentation is really about the failure that I'm showing you today. So the failure is we have had an MEG an electrotriox in our lab in Bari in Italy for 8 years, and we have collected a few hundreds people, and we gave them a task that we chose because it's been shown to be strongly associated with schizophrenia, which is the mismatch negativity task. It's a series of sounds, and these sounds are all equal. They're standard sounds. They come in the majority of cases, and then there is a deviant sound, and this deviant sound has a stronger response. And then when you plot standard minus deviant, you see a negativity. And one interesting aspect is that this negativity is quite heritable. So a 0.68 heritability is quite a lot. You have to think that the heritability of height is about 72, 73%. So the heritability of MMN is not bad at all, according to this particular study. They range a little bit. So we began studying mismatch negativity, and we said, well, maybe we will first ask if this is specific to schizophrenia or if it's found at large in the psychosis spectrum. So we had schizophrenia patients, bipolar disorder patients, first episode of psychosis, and these are subjects who are non-psychiatric at the moment, so they are clinical high risk. That means that these people have some distress from their psychological condition, and they will see a doctor to talk about this distress. We do have an early treatment clinic. So what we're trying to do is catch these cases quite early and see if they transition to a diagnosis. So what we found in this paper, it was published last year, is that schizophrenia patients have a significant decrease in the mismatch negativity amplitude, but so do bipolar disorder patients and first episode of psychosis, and also people at risk. So this is quite widespread in the symptom spectrum. And then the next question is, okay, but now if we know who is more at risk for schizophrenia, maybe, for example, in these cases who are manifesting some subthreshold symptoms, we can say something. Maybe we can help determine whether they will transition or not. And so the thing that we did was we will measure genetic risk in healthy controls. These healthy controls do not have all the confounders like medical treatment not working, familial distress, and so on that patients with schizophrenia have. And so we will see if we can approximate this phenomenon in healthy controls with genetic tools. And it turns out we cannot. So we had this not much interesting result where we had 150 people. This blue are patients, orange are controls. And this is one of the scores that we use. That's not very important in this context, but this significance is not enough to make any claim. And it's also in the opposite direction than what we expect, because we expect people with higher risk to have lower mismatch negativity. And this year, the group of Ole Andresen in Oslo has published a paper. So we did not publish these results, but they published a paper where they failed to associate the polygenic risk score with the mismatch negativity derived from EEG. We derived it from the MEG. They go in the right direction, but it only works for patients, doesn't work for control, so there's nothing here. And the question is then, why do we not see anything from a heritable, so a trait that in principle is heritable? Why don't we see anything that has to do with schizophrenia when not only schizophrenia, but the whole spectrum of psychosis seems to be implicated, and we should see something. So we're thinking, well, this is perhaps a biomarker and perhaps it also co-segregates with the disorder. Why don't we see anything? And we have published a review last year in Biological Psychiatry claiming that probably the reason is that the polygenic risk score is too diluted in terms of biological content. The polygenic risk score has been developed comparing cases and controls. But if you buy for a minute the idea that schizophrenia has different causes, so there's different biological pathways that could be implicated and not only one, and they end up giving the same symptoms, then it doesn't really make sense to compare all these different boxes as they were one box with controls. And the idea here is that we can take the studies that have been performed in these hundreds, thousands of people, we can perform a study in postmortem brain and use this information. So we do data mining on postmortem brain. Specifically, this is RNA sequencing quantifications. We can use different tools to derive pathways, and we can have multiple methods. We have used, for example, parsed risk. Parsed risk means I only take the genetic variants that belong to a specific biological pathway, or we can take a biology predicting pathway. For example, take a genetic score that predicts how much a given gene is expressed in a brain, given the genetic background. And we can associate this with biomarkers and perhaps predict treatment response. We have tried to do so for a decade now. We have shown that a score predicting the expression of one of the genes that Professor Harrison cited, the DRD2 gene, this score is associated with greater prefrontal cortex activity during working memory. If you take the co-twin of DRD1, which is called of DRD2, which is called DRD1, it will show the opposite effect because physiologically it has the opposite effect. We have shown that we can use networks to predict treatment response, and this does better than standard polygenic score. And we have shown that we can derive from the post-mortem brain collections, we can derive different scores based on age and brain regions. And this is the paper that we are about to publish this month or the next in Nature Communications, where we tried to leverage this work that we have done before to show that a score, as a risk score that is enriched for biological content, can identify the association with phenotypes that you cannot identify with the whole score. So this is a case where less is more. Previously, I've heard from Professor Harrison that DRD2 does not really predict much in terms of genetics. In this particular work, what we did was postmortem analysis. We derived this co-expression context. This was significant in the caudate nucleus, so in the striatum, and this particular set of genes appears to be related not with DRD2, but with dopamine synthesis in the striatum. Dopamine synthesis is also regulated by DRD2 because DRD2 has an autoreceptor form. But without going too much into this, so basically what we did later was using 2 PET datasets and 2 fMRI datasets. And here you can see the results. So we basically derived a polygenic score where greater risk for schizophrenia, but only in this pathway, was related with greater dopamine synthesis in the striatum. And we replicated the result in an independent data set. So these were overall about 200 people tested with PET. And these were 2 independent data sets with fMRI. And we showed that people with a genetic predisposition for greater dopamine synthesis who will also have greater F-DOPA PET by the previous association. They also have greater reward response in a reward task. And this was, again, significant in 2 independent data sets. So what does this have to do with today's symposium? I think that the range of phenotypes that we can use for an RDoC association or for connecting symptoms with biomarkers can be predicted much better if we add biological information to the polygenic risk scores. And the underlying idea is that instead of finding subtypes, because subtypes can be phenocopies, they're called, so they have different causes, but they have the same phenotype. So instead of parsing by subtype, which is what you observe, we could parse a priori using the parsed biological risk. And if we do so, then the idea is that we will be able to find different markers, and they can all be associated to schizophrenia, but in some cases and not in others. So it may be that some cases with schizophrenia will show a tendency to decreased mismatch negativity, and some others will show a tendency to different, for example, alpha peak in the alpha power. And then if we do obtain a parsed genotype to parsed phenotype association, then it will make sense to develop drugs that specifically target one pathway and not the others. So there will be a second talk, not today, about how to transform this information about pathways into actionable drug targets. So that's what I'm doing in Baltimore with the data mining of postmortem data sets. But for today, this is what I wanted to tell you, and that this is the group that is doing this wonderful work. Thank you, everybody.
Thank you, Giulio, for an excellent talk. Do we have any questions? Yes.
Yeah, thank you so much. I have a bit of a question because I've been out of the loop for quite some time, but my understanding is that we have a lot of GWAS studies out there, a lot of cohorts, but very few of them, maybe none, have neurophysiological measures associated with it. So I'm wondering, we have a plethora of genetic advances, but how do we manage to plug in something like mismatch negativity? Because we have a different power in terms of sample sizes, and whether this is something that you thought about in your case.
Yeah. So great question. Thank you. And I think these very large numbers are required when you want to discover which variants are associated with the phenotype. So for example, you will need hundreds, well, at least tens of thousands to 100,000 data sets to identify the genes that are associated with delta connectivity or with alpha power, alpha peak range. So that will require so many subjects. What I'm proposing is different. I'm saying, let's pick the data already obtained on a disorder, schizophrenia or depression, you name it, as long as it is a heritable thing. And we try to identify phenotypes that co-segregate genetically with the disorder. So what you need in that case is only a genetic correlation. And the genetic correlation, although it will always be imperfect, it can be very high. So for example, neuroticism is correlated 80% with major depression, which can be, quite significant. You wouldn't need to run an entire study on that, if you can count on that correlation. So I guess the real question is, if we can, if we knew the code beforehand, so we have to decode this richness of genetic variants. But if we knew which variant belongs to the mismatch negativity phenotype, and if we could make a perfect mapping, then the correlation between the genotype and the phenotype could be very high in principle. The problem I see is that we're not dealing with one schizophrenia, but schizophrenia is probably the end product of a trajectory of development that for some reasons looks similar. And it doesn't even look so similar as Professor Singh was saying, because there's many different symptoms, but they look similar, but they're probably not the same thing. Much like you can get diabetes from multiple completely independent genetic causes. And that's probably, well, I guess what I'm saying is we should probably, instead of finding new drugs, find the right people for the drugs.
Any other questions? Yeah, Rick.
Thanks. Really interesting talk. I was just wondering what other kinds of gene co-expression patterns have you been looking at? I'm just wondering, because in a sense, the dopamine one is maybe the lowest hanging fruit because there's not much dopamine in the polygenic signal, but there's loads and loads of NMDA and pyramidal cells and interneurons. And I'm just wondering how do you pass that into a smaller smaller subsets? Do you find kind of pyramidal cell and interneuron specific ones, or are they localized to specific areas?
No, that's, as I said, that will need a second talk. But so I think there's multiple ways. So in this review, we argued there's at least 3 approaches. So for example, gene ontologies have been used to this purpose to generate pathways, and also co-expression has been used. And the regulome, it means, for example, you have TCF4 is a transcription factor, and you take all the targets of TCF4, for example, as a gene set of interest. Now, the problem that you highlight, and you very correctly do so, is that co-expression is simply correlation of expression, but we don't know what is behind co-expression. It could be cell types, could be co-expression in certain contexts in time. But cell types are the most popular thing right now. So what we do is generally one thing I have to say is we have observed that when you analyze independent of cell specificity, you do find certain genes that are co-expressed regularly with schizophrenia risk genes. But we, much like you, we think this co-expression actually reflects different biological content. This is not only cell type, but you have to think that within one cell, so even if you only take neurons, and you only take glutamatergic neurons from layer 3 of the prefrontal cortex, you're still going to have certain proteins appear in the dendrites and certain other appear in the soma, for example. And if you think of mechanisms that can support physiology, you will need to know that even within one specific cell type. So what we are trying to do at the moment, because we have 2 cores a resolution in terms of biological resolution, I'm saying our granularity is not enough to look at specific cells in large enough samples, and let alone specific cell districts. But what we are trying to do is asking the question, is the best target, so are we targeting the best gene in that mechanistic network? So for example, if we want to target the dopaminergic synapse, Is DRD2 the best option we have? We don't know that yet because DRD2, for example, is considered a risk gene, but there may be another gene. And if you activate that gene, it will not only affect DRD2, but also, I'm just making this up now, serotonergic receptors in other, as you say, in other brain regions, in other cell types. So that's called network pharmacology. And that's what we're trying to derive from these co-expression networks. For example, what is the most important actor in this network? Or for example, what is the most specific that doesn't affect the rest of the cell? We're asking these sorts of questions and we are probing these substances in stem cells for now as a model of what might happen in patients' genetics.
Cool, thanks a lot.
Have you looked at how some of the biologically informed past genetic scores might affect specific symptomologies and disease severities?
So we currently have a study where we try to do subtypes from symptoms, and we associate that to risk for different disorders. And we found that one subtype of schizophrenia seems to have less risk for schizophrenia and more for bipolar disorder type 2. That is not published yet, but the next step will be what you suggest. So trying to understand if that distribution of risk is actually reflecting pathways and not just the aggregation of risk. People generally underestimate how much time it takes to do this data mining of big data sets. So we haven't been able to get there yet, but it's definitely one next step.
Thank you. Okay, let's thank Giulio Pergola again for a great talk. Okay, so our next speaker needs no introduction, but perversely, I'll introduce him anyway. It's Professor James Rowe from Cambridge. Thanks for coming, James. No, it's a great pleasure. Thank you. And thank you to Megan and to Krish for organising this. It's a great day. I'm aware it's the end of a long day packed with information and ideas. Let me use that. So it's one of the joys about talking at the end of the day is lots of the ideas and themes have already been introduced. But I'm going to really extend some of these and talk about our work on Alzheimer's disease. For the sort of long-term record, these are my disclosures, but none, I think, that are relevant to the material I'll talk about today. The work I'll talk about is really all set in the context of the Dementias Platform UK. This is an academic industrial partnership in the UK seeking pre-competitive, high-trust relationships and multilateral solutions. We all face the problem that if we were lucky enough to have a drug that works for Alzheimer's disease and dementia, how do we show it's going to work? And the DPUK team focuses on processes rather than single molecules. We're not on amyloid or tau or whatever, but we're focused on processes around inflammation, vascular health, and synaptic health. And it's synaptic health that is most directly related to the physiology, the cognitive physiology, as to how the squishy stuff between the ears generates thoughts and memories and behavior. So we have a number of technology platforms. The one I'm closely involved with is for experimental medicine. There's also a trial delivery framework and a really valuable data portal for disseminating and providing access to data now from around nearly 4 million people with multimodal imaging, cognition, genetic information, etc. So I'm going to focus on experimental medicine work and MEG in particular. So what might MEG have to offer? Why are we interested? Well, the challenge is to open up the bottleneck in drug development. We heard a little bit about this from Dennis this morning, that funnel that tapers very rapidly as you go from drug discovery through early phase trials. And if you're very lucky, on into clinical applications with 1 or 2 examples. It's a very tight bottleneck and it's expensive. So we need to de-risk and accelerate this whole process, and can try and do that with a range of new assays, MEG-based assays. We might ask the MEG to, in some way, be sensitive to the presence of our disease, presence of Alzheimer's, to be sensitive to the progression of Alzheimer's, in order to be sensitive to the effects of a drug to slow that progression. Hopefully, on the way, we might learn something about disease mechanisms, building bridges between preclinical animal models and the human condition. But also something I've learned a lot about from industry partners and not from my academic training is really about trial readiness. Is it reliable? Is it scalable? Does it do what's needed for trials? Okay, not about high impact paper. It's not about a small p-value. It's a rather sort of hard graft around trial readiness. So the MEG data that in the UK, and it would be accessible to really anyone here, but derived from the UK that might be relevant to aging and dementia risks, includes the Cambridge Centre for Aging and Neuroscience, the CAMCAN study. This is now a multimodal imaging data set with a lot of MEG, now available through, freely available to download worldwide. The same protocol was mirrored in what's called CAMCAN Frey, and I'll talk about those in a moment, and in those with Alzheimer's disease and mild cognitive impairment through memory clinics. We mimic the CAMCAN protocol into the clinical context. There's also the BioFind data set, over 300 people with either controls or MCI. There's a very exciting study called the Deep and Frequent Phenotyping Study that will link MEG and EEG to tau PET, amyloid PET, optical tomography, MRI, cognition, blood genetics, CSF, I forgot. Pretty much anything you could put a living human through in a study of dementia, but all in the same people, head to head, and frequently, baseline 4 months, 8 months, and 12 months. That should be finishing later this year. I'll talk a little bit about this study called NTAD. This is really following people with either MCI or early mild Alzheimer's dementia longitudinally. And there's a non-Alzheimer study, which Laura Hughes has talked a bit about, and we'll hopefully make that available later in the year. Most of these data are available to access through the Dementias Platform UK. So today, though, I'm going to talk really about how we've been running the NTAD study and the CAMCAN-12 study. I'm going to focus on task-based MEG. You can touch on this resting state later. I'm going to focus on the evoked responses, although there's a very high dimensional rich information in the induced responses that mustn't be overlooked. I'll touch on both activity and connectivity. And while we often talk about this division between sensor-based and source-based, we should also think about model inversion to generative models. Okay, so understanding how the MEG signals are generated through formal biophysical models, which I'll illustrate today. We've heard about the mismatch negativity task, and I'm going to use examples from this. I like this task because, unlike resting state, you have some cognitive interpretability. We understand the nature of what's causing these evoked and induced responses. The resting state, goodness only knows what's going through somebody's mind while we're getting the data, okay? And that is a problem. So you control, you parameterize, experimentally take control of the brain responses with MEG, with a mismatch task. But it's basic. Anyone can do it. You can do it in your sleep, frankly. I'm literally in a coma. You can get mismatch studies in coma study patients. So, and it's minimal training. You get a response in everybody. So it has a really nice sweet spot for translational applications. There's different varieties. I'm going to draw on what we call the roving mismatch, which has a series of tones repeated, and we vary the frequency every so many tones, between 3 and 10 tones, or shift frequency. So it sounds a bit like beep, 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 boop, 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 beep, 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 beep. It just goes on and like that. It's pretty dull, but it's, you can watch a film while you're doing it. You get nice responses. There's another very nice study from Finland, former mismatch from Finland, which compresses many different types of deviance in a highly efficient way. So you can think of each tone having multiple dimensions: its volume, its laterality, its temporal profile, its loudness. And on alternating tones, one of those many dimensions becomes deviant. So it sounds a bit like beep, boop, beep, 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 beep. And it's incredibly efficient to pack in lots of deviants, almost simultaneously deviants. Very good for clinical studies. And there's a visual associate oddball where the unexpectedness is around the pairing between visual and auditory stimulus, and that depends on hippocampal associative learning mechanisms. So really nice for Alzheimer patients, but still has this automaticity and a minimal training, a minimal performance aspect. So let's take some of these tasks. We're going to take that last task initially and just ask the question, what happens in Alzheimer's disease? Does it damp the mismatch response, the response to unexpected stimuli? And if it does, is it specific? Or is it just a marker of an unhealthy brain, a brain that can't perform optimally? So we can look at that with people who perform in cognitive terms similarly poorly, but not due to Alzheimer's disease. So the data set has patients who are, or participants who are either healthy controls, cognitively normal, no symptoms, community dwelling, MMSE 26 or above, ACEs 85 and above. Or people with Alzheimer's disease who have symptoms, they've come to memory clinics, they've got Alzheimer's disease with clinical impact. But in between are the two interesting groups. We have a group called MCI, mild cognitive impairment, that have come through symptoms, memory clinics, mild cognitive impairment in that classic diagnostic group. And you'll be familiar with those perhaps. But the cognitively frail group are interesting. These are community dwelling, asymptomatic individuals, but they've got low MMSE. Their MMSE score and their Addenbrook's cognitive exam score, slightly more sophisticated cognitive screening test, is as good or bad, is the same as those with MCI. Okay? So if you look there, look at the green and yellow boxes, they've got low MMSEs and low ACEs scores to the same extent as patients with MCI. What's different, though, if you look on the right-hand side, is hippocampal grey matter volume. So the top row shows normal volume and controls. You can clearly see reduced and highly variable hippocampal volume in the Alzheimer's patients, slightly milder but still significant hippocampal volume loss in MCI patients. But those who have this cognitive frailty, who are scoring poorly on cognitive tests, but community dwelling, asymptomatic, they have relatively preserved hippocampi. So let's see what happens physiologically when we run this oddball task. So you get a series of images, simple cartoon images there, each paired with a tone. So the sort of green and yellow star goes with a beep, let's say, and you hear that repeatedly. The pink and blue goes with a boop. But every so often, we might play the boop when you see the green and yellow sort of flower. Ooh, that's the wrong sound for that picture. So it's unexpected. It is an oddball. The nice thing about that is it's dependent on hippocampal function for dissociative pairing, but the expectation is a tone. So the readout is going to be driven by auditory cortex, which, unlike hippocampus, is readily amenable to MEG. Okay. So you're actually getting a signal that you're going to read out from easily visible auditory cortex, but it's dependent on hippocampal function. So what we see is we get a very, on the left-hand side in controls, clear difference between the waveforms in response to a standard tone, which is when you see the right, you hear the right tone to the right stimulus, and when you either get that DA, the associative oddball, deviant based on its associations, or occasionally play a completely new tone. So it's a novelty deviant. So look on the left, controls, you clearly get differences in the MEG waveform. On the far right, you see an Alzheimer's disease. They've lost that ability to differentiate unexpected from expected stimuli. You still get an evoked response. It's just that you haven't learned what is being repeated, what is predictable versus what's unexpected. What's then interesting is to look between the two, those cognitively frail people with scores down in the MCI range, but no symptoms, normal hippocampal volume, they're effectively normal in their physiology, even though they're cognitively in the MCI range. Whereas those with MCI who've got symptoms that come through memory assessment services, they've lost those responses, the differentiation. They've got a mild version of the Alzheimer's disease, in effect. Okay. So the automatic cortical response to unexpected stimuli is reduced, but it's specific to AD and MCI. It's not merely a feature of a cognitively underperforming brain. Okay. So you don't see it in those with cognitive frailty. Well, let's stay with those who do have Alzheimer's disease. I'm now going to come into a second study. This is called the NTAD study, New Therapeutics in Alzheimer's Disease. This is building a trials platform. The challenge that was given to us by colleagues in AstraZeneca was, can you show us in under a year for under $1 million that our drug is not useful and we should cancel the program? Or say it's likely to be engaging relevant neural systems, we should invest further through 1B and 2A studies. Okay, so that was our challenge. So MEG, I think, is ideally suited for this. We're going to take patients who are biomarker positive and clinically diagnosed Alzheimer's disease or MCI. We're doing this roving paradigm, so beep, 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 boop, 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 beep, 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 study. And we're looking for that response when you change frequency. You go from a standard beep, 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 and you change it. Boop, 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 boop. Or that first deviant, something unexpected has happened. And if you compare the repeat to the standard, so the deviant to the standard in controls in blue, you get a very large mismatch response. Okay, I call it mismatch negativity. I mean, it's not EEG anymore, it's MEG, but I'll call it negativity for habit. Around 150 milliseconds, exactly where you'd expect that initial mismatch response to be. But our patients with mild to moderate Alzheimer's disease at baseline in yellow show a significant reduction. And we follow them then for a year. It's about 75 patients, I should add, sorry. And we follow them for just over a year. And even in a year, 16 months on average, you see a further reduction in pink. Okay, so the MEG response is not only reduced by Alzheimer's, it's progressive in a way that we can identify with modest numbers of patients. Okay, 50, 75 patients, under 100. So you've got a good enough sensitivity to think about for looking at experimental medicine studies. So yeah, Alzheimer's affects this mismatch response and its progression, and it's detectable in pretty much everybody. So but it's a very robust, robust response. It's simple. It's a nice heuristic biomarker, perhaps, but you really know nothing about the mechanism, either the mechanism of the mismatch response or indeed the mechanism of Alzheimer's. So we need to think about a way to move beyond these simple evoked responses, sensitive though they are, to think about understanding those changes, understanding mechanisms. There's a number of different sort of tools available. We heard about NeuroSolver this morning and dynamic causal modeling. So I'm going to use this process called dynamic causal modeling. These are biophysically informed generative models. In other words, it's a simulation, if you like, but it's a simulation where you optimize the parameters of the model so that you best reproduce the MEG of each and every subject. And the way that to conceive of that is to say, well, across the cortical networks, the macroscopic cortical networks in frontal and temporal cortex that generates a mismatch response, in each region, you've got cortical column structures. They're highly regularized. Remember back to your undergraduate days, you've got this nice laminar structure. Inputs come into stellate cells and layer 4, and they come up into superficial and deep pyramidal cells, and they're subject to inhibitory neuronal control. It's a highly regularized stereotypical structure, which we can model. So this figure is thanks to Alex Shaw, who's worked with Krish for many years. We now have a model where we can represent for each cortical region how these neuronal populations and subpopulations and different laminae may be interacting. And we fit that or invert it to MEG data, and we can get parameters for the strength of connectivity. We can get parameters for the overall goodness of fit of the model, which is based on the model evidence, which is looking at the goodness of fit, correcting for model complexity. And we can also, in some of the later models I'll show you, actually parameterize the individual receptor classes, AMPA, GABA, and NMDA receptors that are underlying these microcircuit connections. So I'm going to give two examples of this initially. One is around the effect of Alzheimer's disease on pyramidal cell function, particularly the pyramidal cell gain. Now, why am I going to focus on that? Well, we go back to preclinical mouse models. This is work from Sherby et al., looking at the superficial gain in actually rats, I'm sorry, not mice, superficial gain under pharmacological manipulations. So they showed there's a dose dependency between cholinergic agents and anticholinergic agents in the gain of superficial pyramidal cells. Cholinergic agents have been the mainstay of symptomatic treatments for Alzheimer's disease for many years, drugs like donepezil and rivastigmine. So this shows that that cholinergic effect may at least partly be achieved by changing the gain in superficial pyramidal cells. Now, if you are really into DCMs, keep looking. Otherwise, you'll allow yourself a blink. I'm just going to show a more technical slide. Really, this is for Krish and others. If you're used to working DCM, this is a canonical microcircuit model using a convolution model for evoked responses. At the first level, in other words, for each and every subject, we specify 4 cell populations: superficial and deep pyramidal cells, inhibitory interneurons, stellate cells, at 8 cortical regions, symmetrical in the brain. We optimize the model for each subject, and then we go back with a method called parametric empirical Bayes, which provides group level priors to then understand what governs individual differences and individual similarities in the function of these microcircuits. And at all times, we find the best model by looking at model evidence. We estimate a thing called the free energy estimate. It's a model evidence estimate, and we want the models that have the best fit, provided we penalize for complexity. Okay, because these can be complex models. We have to have a rigorous penalty for complexity. So let's just look at the basic MEG data. The middle panel is very similar to what I showed you before. This is a mismatch negativity response. In the top of panel B, the blue and yellow is patients and controls, showing that clear difference.
The patients that came back after a year are shown in the lower panel. And again, you see this progression of the mismatch response. On the left-hand panel A, what we've shown is the response, MEG-detected response on each repetition. So the first line is when you change frequency, beep, 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 boop. You get a very large mismatch. And then we show over the successive tones, first, second, third, fourth, fifth repetition, and so on. If we look, though, at this first mismatch negativity, what's shown in panel B, and we take the same subjects and we bring them back 2 weeks later to do a test-retest assessment. Okay? Not long enough that the disease has moved on. We just give them a few days off to rest, and then bring them back 2 weeks later and do the whole experiment again. I hope you can see the dots there. There's a very, very strong correlation between day 1 and 2 weeks later. This is an incredibly reliable metric. Your ICC is above 0.9. Okay, so it's a reliable measure on an individual subject basis, and that's critical for them being able to detect the effect of change or the effect of drug or time. But it's the MEG response that we wish then to sort of invert back into our biophysical models. So which is the best model, though? How might these 8 regions be best connected? Well, there's many ways that 8 regions can be connected. So we need to actually, first of all, think, what is the best model, the best anatomical model? And we set up a family of alternative models representing different potential connectivity patterns. I won't go into the details here, but you can search over model space. You find the models that have the highest model evidence. So in C and D, the top rows with multiple bars is the model evidence is for each of these different models. Okay, we want the highest value. The difference in that free energy corresponds to the log of the base factor. So we can convert effectively those free energies into a relative probability and say which is the most likely model. And there's only one model that really wins, hands down. It's model 6, model 6A. It's the same model is most likely in controls and patients and at the beginning and end of the year. So it's a good model. It's a best fit throughout the entire experiment. And you can't take that for granted, but fortunately for us, it was the same model at all time points in all groups. So now we're going to take that model. Okay, so this is a biophysical model representing in each of our regions the properties of the superficial and deep pyramidal cells and other populations. We're going to focus here, for that background reason I talked about, focus on the gain of the superficial pyramidal cells. Okay. So what happens that, we can say, first of all, does that differ between groups? And the answer is yes. So shown in panel B in the red bars, in Alzheimer's disease, that gain is reduced. Okay, in multiple brain regions, symmetrically, it's reduced. So just as we'd have expected from those rat studies, we see a reduction in the gain of superficial pyramidal cells. Take those same patients a year later, and it's further declined. So the effect of time, a year, 60 months, I should say, is to further reduce the pyramidal cell gain. So we now not only have the MEG can detect changes, we can start to attribute those changes to individual elements of the course, of individual cell classes. We can also look at the connections between regions, frontal to parietal and parietal to temporal and so on. And again, we see many of those connections are reduced in Alzheimer's disease compared to controls. And of those, many again change over a year. So it's now not just the evoked response, but it's the evoked response understood through the changing macroscopic connectivity. So both micro and macro connectivity are changing over time in as little as a year. So this is just trying to introduce the idea we can get some cellular and laminar specificity, okay, to understand the MEG changes. Well, we want to go a bit further. In a mouse model or in the vervet monkey we heard about earlier, you go further, you say, well, what about receptor subtypes? What about the synaptic dynamics? What about the time constants of NMDA? What about the NMDA blockade? What about the strength of AMP or strength of GABA connectivity? You could take that for granted in an animal model, but in humans that's difficult. In practice, you can't do it directly. You can't stick electrodes in and do iontophoresis and so on. It's just not realistic. But we want that same level of specificity in humans as we get from the animal models. And these biophysical models do allow that. So we can start to ask around, for example, the example here I'm going to give is the effect of Alzheimer's disease and the effect of a drug like memantine on a specific receptor class, the NMDA receptor. So Again, just for the sort of, if you're into DCM, the specificity of this is we're using the CNM NMDA conductance-based microcircuit model, inverting from the mismatch responses over successive repetitions. We fit these models individually to every subject, and we add a parameter that allows for variability in the degree of NMDA blockade. But we allow the modeling to say for any given participant, how strong is their individual blockade? What that individual blockade does is it changes the conductance for any given membrane potential. Okay. And this is a nonlinear dynamic, sort of summarized in the bottom right. You get these nonlinear relationships between the conductance and the membrane potential according to that degree of blockade. The bottom line is the higher the blockade, the less conductance across that channel. Okay. But it's nonlinear. So we can take these models, we fit to every person in our group. There's about 75 patients here and 30 controls. And we can say, is the MEG difference between patients and controls, is that explicable in terms of the model based on the AMPA receptor dynamics, the, what I've got here, AMPA, the GABA, the NMDA time constants, or the NMDA blockade parameter, the extrinsic connectivity between brain regions with H, or some combination of all those. Of course, the combination gets more complex, so there's a penalty. Yeah. But essentially, when we compare model evidences, it comes very clearly through that the models where you attribute the MEG differences to the NMDA blockade parameter is the best, simplest way to explain difference between Alzheimer's and control participants. Okay. So hold that thought. So the NMDA blockade becomes very important now for understanding how we might take that forward with drug studies. Is it clinically relevant? Well, I've shown you the difference between groups. Interestingly, in the patients, that NMDA blockade, the mismatch amplitude, I can't block in a minute, the mismatch amplitude correlates modestly with MMSE, disease severity. Okay. So the mismatch amplitude is related. But what's really interesting then is the relationship between the NMDA blockade and clinical severity. So the modeling's estimate of individual NMDA blockade parameterization correlates with their clinical severity. Okay. That's at a group level at baseline, and we look at that NMDA blockade effect over 18 months, and we see a further change over time. Okay, so progression of the disease as expressed through changing NMDA dynamics. That's with a known drug. Okay. Is it reliable? Well, yeah, it has to be reliable if we wanted to study the effects of disease and progression of drugs. But these are complex, multivariate systems with high covariances amongst parameters. There's incredible redundancy really at the synapse. You put one parameter up, you might compensate with another parameter, and the net function is the same. So it's very covariant. So traditional measures like just a covert correlation, ICC, do not work. We need to rethink reliability. And the lay way to think of this is, if I did the same experiment twice, would I get the same answer? That's kind of what we mean. Okay. You can formalize that to say, are the inferences under hypothesis testing the same, or is the Bayesian parameter estimation equivalent? So we've looked formally at this. We've shown, well, we've taken this approach. We said the reliability should be really, really high if I take the same subject in the same session and just take on and off epochs or odd and even trials. It's like the same person, same setting, same day. It should be extremely high. If I took the same person 2 weeks later, it's not going to be perfect, but it should be good. It should be high reliability. If I take two different groups of people, same type of patient, but different individuals, the reliability will be lower. If I started to measure people at different times or different centers, it might drop further. So there's a hierarchy of what we'd expect. And so we've looked at this formally. We've taken subjects with Alzheimer's disease, we've brought them back 2 weeks later, and we've either looked within the session or between sessions, and then with a separate group of same subjects at the same center. So a hierarchy of equivalence. And if you remember, I said these are complex models. So we have in these biophysical models about 150 parameters. There's a lot going on. How many of those are the same or different when we come back 2 weeks later? So in the top row here of 150 parameters, I think it's 156, only 4 differ within sessions. So I take same person, same session, and I just divide their data up. And I do the whole modeling procedure twice. 2 or 3% of parameters vary between that, 97% are the same. Take those subjects back 2 weeks later, then is it 4, 5 parameters differ? So 97% of those parameters you're interested in, you get the same answer. It's brilliant. Of course, if we take the same patients, but not the same individuals, we start to see many more differences emerging as 8 or 9, but there's still a very strong disease effect. So I'll skip over this. So can we de-risk it? Well, we've shown that MEG is sensitive to the disease. It's sensitive to progression over time. We can start to elucidate disease mechanisms. We can start to attribute and identify drug effects. What we're interested in is failing drugs fast, as we heard about earlier. All I've shown you here is about confirming a drug effects, but the real idea is to go to new drugs and fail them quickly. And we've shown it's reliable. It's also highly well tolerated by patients. So we're starting to meet some of the requirements for drug scalability. And with that, I'll thank you for listening and thank the fantastic team that have done all the hard work. Thank you.
Thank you, James. Sorry to be this.
I realise no way of getting on late. Yes.
I think we're just going to squeeze in one question if anybody's got a pressing question for James. Okay, could be a beer.
I can see them both switching. Who's going to?
If it's short, but we may be.
A beer is good.
Yeah,
Cabaret.
super concise. Yeah, fascinating talk. Thanks, James. I was wondering, I thought there was quite a space between the full-blown biophysical model and the average mismatch amplitudes, where you ignore the spatial dimension. Has anyone in your team tried running topographic decoder on this? Trying to use really the entire source geometry and using all the sensors?
We haven't. We would be very happy to make the data available to anyone who wants to do that. We skipped that step because we really wanted to understand the mechanism at a cell and neurochemical level. But you could, yeah.
Go on then, James, I'll ask the final one and keep everyone away from their wine. Mine was really about the variability, and your hierarchy had within or between subject within site as being less variable than same subject between site. Was that correct?
Same subject, different sessions.
Different sessions.
So if I bring you back 2 weeks later, you're a little bit different. You're a bit more chilled one day, an extra cup of coffee, a bit more relaxed. There were legitimate reasons why our brain dynamics differ slightly day to day. So it's okay to have a few parameters changing.
But what about different sites then? I mean, on different scanners? I mean, is that...
Different sites we're coming to. Yeah, we've now just got the second site was delayed because of refurbishment, but we now have data running the entire paradigm, including test retest at a second site. So we're really looking forward to that. It's going to be important to know. Yeah.
Thank you so much.
Thank you.
We have 2 more talks from the local lab. So Olivier is the first speaker.
Thank you very much. It'll be quick, I promise. I just going to represent the platforms at Campus Biotech here and just say a few words about us, about what we are and what we do for the community here and what we can potentially do for you. So we have an MEG here and we are part of the Human Neuroscience platform, but it is only one of the many platforms that are actually at Campus Biotech. So as you can see on the slides, we have some platforms that are specialized in neural microsystems, organoids. We have a genome center, the Human Neuroscience platform that we are part of, but also a gene therapy unit, preclinical neuroscience, and a system integration platform. And the services that we provide are to support the entire workflow of neuroscience research and development by providing cutting-edge technology, but also expertise with staff, which are all experts in all of these different techniques. And also a common point of all these platforms is that we are open to everyone and accessible to all, whether you are academics or a startup or an industry. And so if we zoom in inside the Human Neuroscience platform, you can see that we have different units. So we have now an MEG system, but there's also a virtual reality platform, a clinical and sleep research unit, a non-invasive brain stimulation unit, an EEG lab, an MRI platform, and recently a biomarkers analysis lab, which is on site. And so the Campus Biotech is pretty much this biotech hub, this meeting point. And we firmly believe that by having all these technologies
together here accessible at Campus Biotech makes it a lot easier for all the researchers to combine them together and complement all of these informations. So there's now an MEG on campus, and it's been operational for more than a year. And this is part of a very ambitious project that was called Ambition Neuroscience that kept it quite busy for a lot of time. So it was a huge partnership between major local universities from EPFL, UNIJ, and HUG, but also with academic partners such as the NCCR Evolving Language, which you'll hear a bit just later on with Alexis, and the CIBM, which is the Center for Biomedical Imaging. And this project resulted in the acquisition and installation of two major systems. So the very first MEG of Switzerland from MEGIN, a MEGIN 3X Neo on the left, but also a Siemens Terra X 7 Tesla MRI. So we've heard that MR spectroscopy and MEG, they go well together. So it's a good thing that we have both systems here. This is just a small teaser, but those of you who are interested will be most welcome to follow us after the end of the session, where we can show you the actual MEG on site, which is now, like I said, operational. And the last slide is to show you what are the different services that we typically provide the community. So basically, we do support for the entire community, and this means for the whole human neuroscience platform, more than 450 users on site that belong to 30 different research groups from the local universities and also actually national and European ones. Since the creation of the platform in 2018, there's now more than 115 publications using the type of equipment that we have. You can see the faces of our team members on the slides. And what we do is basically the maintenance of all the equipment. We can train the users, we can design and do the setup of new experiments or replicate whatever they want to do. We can develop new tools for the community, and we've done that plenty of times, especially when it was about combining different modalities together. So we are, for instance, one of the few centers where you can do TMS inside the MRI or virtual reality inside the MRI. And now we can also play with the MEG to make it compatible with virtual reality or other things, depending on the wishes of the community. Of course, we welcome multimodal imaging, and our team members are experts in all of these techniques. What we do is we have a strong expertise in all of these techniques. So all of our team members are either engineers or PhDs in the respective fields, so that even if you have a good idea, but you don't have the means in terms of human resources to do the project, we can actually also help you with data collection and analysis. We have technologies to guarantee optimal data quality, experimental support. Like we did with Ambition Neuroscience, we can support new large-scale projects. And then, like I said, we're open to everyone. Of course, the MEG has a strong interest for neuroscience, but as we've seen today, there's also a huge potential to be used by the industry. And so we're open to everyone, and please do not hesitate to contact us if you have any questions, any ideas during the tour, after the tours, whenever. Thank you.
All right, so I'm here as a representative of the National Centre for Competence in Research, Evolving Language. The National Centre for Competence in Research is a great mouthful, so I'm going to refer to it as NCCR, or Evolving Language. And first, why? Why am I here? As Olivier mentioned, the NCCR was instrumental in pushing for the acquisition of MEG, the first MEG in Switzerland, and we're super happy to have that here. Why do we exist as a group of researchers? Here are my justifications. Language is a fundamentally defining human characteristic, which is central for communication, obviously. It structures our thought, or at least we believe so, and it creates important social bonds. The way that we communicate is fundamental to being how and who we are. Now, the important part of the evolving language here is that language may define us as a species, but it's constantly undergoing profound changes. So there's now challenges we have to deal with new amorphous, depersonalized interlocutors when we talk to our digital assistants, for example, like our Siris and Alexas. We're in a realm or a time of neurotechnology. We have potentially very ethically challenging promises to deal with, so reliable brain decoding interfaces that might rehabilitate people who lost the motor capacity to produce speech are opening doors for brain reading, but we know that not all technology is necessarily used for good. And there is globalization as a continuous and ongoing threat to linguistic diversity, which comes with an attendant threat to knowledge, the loss of indigenous knowledge and local knowledges. So these challenges, these threats, these opportunities are some that require a radically transdisciplinary approach, and that's what the NCCR hopes to provide. So we are a diverse community of researchers spread throughout Switzerland in a huge number of fields. I'm not even going to read them all because this is probably not an exhaustive list. We're spread throughout the country. There are 6 centers with over 40 research groups implicated using various kinds of methods. And I'm only going to talk about the kinds of things that we're interested in with MEG and why. So my summary of why MEG is so important here is the brain is the limit. Our brain is fundamentally the biological constraint on the human language capacity. And we need to understand, at least those of us interested in the brain and language, feel the very, very burning need to understand how language is implemented upon that biological substrate. So being the constraint, we have to also ask what makes the human brain uniquely suited to support a complex communicative system. This is an evolutionary question. Fundamentally, we have to ask, how does the brain differ from our nearest neighbors in the animal kingdom who do not have the same language capacities that we do? I was super excited to see a vervet monkey in an MEG. We're not ready to do that here yet, but maybe one day that would be super cool. They have great communication systems. So what do we do with MEG to address these issues? Just to get back to why MEG has arrived, why I'm so excited that it's here. Communication is rapid, it is multimodal. Speech signals unfold. Recently, it was published at 39 bits per second. It's quite fast. And speech and sign language and all communication, but they're really fundamentally multimodal. There is a lot of information happening in parallel. I'm speaking, but some of you are doing other things, but some of you might be looking at me, watching me wave my arms. You'll notice that I'm modulating my voice. These are all things that we have to integrate. And we need high temporal resolution in order to be able to understand what the brain is doing when it is integrating dynamic signals at this kind of pace. And we're also interested in the networks in the brain. We want high spatial resolution, or the possibility of high spatial resolution, in order to get insights into where we should be looking when we want to look at the differences between the human brain and non-human primates, in order to understand what it is about our neural architecture that makes us able to have this capacity. So I'm just going to briefly outline some of the projects that we're running, and I'm doing it at speed because your brains are fast and language is obviously dynamic and fast. So this idea of multi-channel contexts, there are postures, there are gestures, there are facial expressions, all of which modulate the way that you understand a signal. Our voices themselves constantly betray things like cues to identity. Who is saying what is absolutely essential. We have to track one another. We have to track the person that said something. The truth value of a statement is fundamentally altered if you know that your interlocutor is a habitual liar, for instance, or if you know that they are a trustworthy interlocutor. And then there are things like prosody, and the way that prosody is used to flag communicative intentions. So irony, sarcasm, you can tell things about my mood. You know that I'm going fast because I know you're all thirsty. Lots of things that you can derive from this signal. And many of the projects that we have underway try to uncover the mechanisms that underlie the integration of all of these parallel streams of information that can all serve to somehow change the way that the linguistically encoded content is processed and perceived and integrated by the receiver of information. We're also very interested in the neural basis of hierarchical structure building. Keeping track of the words that I say in your memory serially will not lead you to be able to understand everything. And not everything you hear or read. So bear with me for a brief example. Have a look at this slightly quirky sentence. Some of you will know this. Those of you who don't know it will have experienced a puzzlement when you came across the final word. If I enunciate this with a bit of prosody, the horse sped by, the barn fell. So even though you may have struggled to understand how fell integrates into the sentence, you eventually managed it. And we know that the brain deals with these things with a process called syntactic repair. It takes about 600 milliseconds for a big P600 signal to be noticeable on an EEG or an MEG. And this is happening all the time. So when we're understanding sentences, we have to build hierarchical relationships between the words that we read or hear. And that process is continuous. It is rapid. Here's a little picture culled from a recent article by Helena Hösling and her colleagues, showing that in about 350 milliseconds, your brain is discriminating between words that are in context and words that are out of context in scrambled sentences. A huge amount of the brain is dedicated to integrating words into their local situational linguistic context. And in order to decode this, we need MEG. And composing meaning, of course, is fundamental to what language is. So this is really a precious thing that we can do. We can decrypt what the brain is doing when it's making sense. We're very interested as well in conceptual space as defined by language, how the words that you have actually modulate the way that you represent the world. And so temporally and spatially resolving the mapping of forms to meaning when we're retrieving words in order to do speech planning, to actually utter something, to actually understand, for instance, the interference of shared forms between languages, or the perturbation of access to words in disease states is something that we can look at, and we can try to ask questions about how the different organization of concepts within languages might change behavior and how that ramifies into the signals that we're able to look at in the brain. And finally, just a slight applied thing that we are very interested in from our medical colleagues, the idea of using MEG in neurofeedback, so rehabilitating patients and improving training or improving performance in healthy participants. And as has been evoked already today, MEG is very well suited to patient applications due to a reduced setup time. And we're interested in network-based approaches. So the high spatial resolution is, again, super important to us. And some of the domains that we're trying to do this in are aphasia rehabilitation and cognitive training. And some of these lines of research are really, like I say, interested in a network-based approach to the problem, therefore having real-time readouts that we can actually invert, look reasonably well in the source space is going to be extremely helpful for this kind of approach. So I have rushed, therefore I was less clear than I would have hoped. Thank you very much. Enjoy the rest of your time in Geneva, and I'm available for any chat afterwards.
Thank you, Alex.
Thank you.
A quick question.
Of course.
You mentioned studies of particular diseases. Do you have a particular disease in mind that would benefit from a deep dive into?
So typical focus for us at the moment would be aphasia. Obviously, that's a very applied case. There are also questions that I was asking myself in relation to the Alzheimer's talk that arose earlier and how the change in oscillatory dynamics might actually be relevant to potentially impaired communicative strategies, but that's a very speculative thing. For the time being, really, the approaches have been on some forms of dementia. So we have a colleague over there, Valentina, who will be glad to talk about dementia in great detail. So that's one of the things that we're particularly interested in the NCCR because we have the expertise, and rehabilitating aphasia patients is the main, so post-stroke, obviously, kinds of approaches.
I wondered a little bit about maybe schizophrenia with the sort of deficits in predictive coding.
So there are some colleagues in Zurich who are working on computational models of the content of expression in various psychiatric states. And that is definitely something that is of interest, although at the moment it remains at the computational linguistic side rather than the MEG.
Thank you.
Thank you very much for the questions.
I just wanted to say it's been a really fantastic day. We looked at this about 8 months ago and made it happen. So thank you very much for supporting us on this. It's been amazing to uncover all this work that's going on in the pharma space. So we would like to continue collaborating with you. We'd like to continue discussion groups, and we'd like to continue making more of this in the future. So we will be in touch, and we will follow up with you on further potential events. Quick few thank yous. Krish, thank you very much. I've already said it, but you've been amazing, so thank you. It's been great. All our incredible speakers, you come from far and from near, so thank you very much for doing that. And also we will be in touch with hopefully more collaboration with all of you too. It's been an incredible array of talks today. Thank you to Campus Biotech for letting us use your facilities today and also do a MEG lab tour next. And I know it's been a very long day for you, and we've been badgering you every single week with some requests. So thank you very much, and thank you, Ben, for helping us today with this.
Yeah, sorry, we're late and made you stay.
Yes, sorry about that. And then also thank you to the online audience. I think we've got another 25 still there, so they lasted all day too, which is amazing. Thank you.
Yeah, well done.
Yeah. And then lastly, Nicole and Zoe, if you're still online somewhere in the virtual world, incredible organization, incredible way of getting everyone to do what we need them to do. So thank you again. And Nicole, you can relax now. So, that's it. I'm hoping you're going to enjoy your drink. Thank you very much.
And thank you so much to the speakers as well, I think.